Okay, welcome to all of you in Ancona for this cost meeting. It is very nice to see all of you, with most of you for the first time in person, and we are confident that it will be the first of a long list of times. With some others we already met in other meetings, and this is a good opportunity to meet again and to have some exchange all together. But before the start, there is the welcome of our director, Professor Davide Neri.
Good morning to everybody. I really welcome you in Ancona University on the behalf of our rector, Professor. I like to say you that these kind of meetings for us are very important. It's a way to interact, a way to spread the knowledge and overcome the barrier. So for young people and old researchers, this will be a way to stay together. So the topic is very important. Today we start the cost action. We have the kickoff for the cost action. Yesterday we had the final meeting for the Prima project, and together they are a conference that is innovation in food loss and waste management. So that is a key topic, how to make this improvement of circular economy. And this is very important for our university, and we are working together with the other faculty to create the way to mix the approaches from engineering, from medical school, from economy, from life science, and from agriculture. These 5 faculties in Ancona University are joining for making this kind of projects. And we have also a master that is prepared for circular economy. So that is a key point for us. The fact that here we have speakers from 30 different countries means that this is a topic that is important also for other countries. And so that will create the possibility to make a food chain that is more reliable and with lower losses. So a few words about our university. So our university is a medium small size. It's about 15,000 students and 5 faculties, as I told you, scientific faculties. And we have agriculture that is more or less 1,000 students. And every year, this year, we had new coming students for about 200 people. So that is good numbers to make research and to create the basis for the bachelor system. Then we have master system for our 3 topics that are agriculture, environment, and food science. So these 3 are the courses that we have, and we put up from the bachelor up to the masters, and then to the doctorate school. That is quite good and important, with more than 10 new doctorate students every year. So I think that we can wish as a university, as delegates for the rectors, that this meeting will be very profitable, and the stay in Ancona will be a pleasure for you. Thank you.
Thank you, Davide, and for the support that you provide us through our activities and for the admin duty that you took in the behalf of all the department. And we are here all together thanks to an initiative of European Union, that is this COST, that is a project that support and foster interactions. We are within the food waste stop cost action, and we have the pleasure to have the welcome of the cost officer, that is Marco Carulli, that is involved in other activity in Brussels, and he takes care of several cost action, and it is a pleasure to have him for a welcome address.
Thank you very much. Thank you very much indeed, the director and Gianfranco, for organizing this meeting of the Food Was Stop action. As Gianfranco said, I'm working as science officer at the Cost Association. Cost is. Can you hear me?
Yes.
Excellent. Very good. COST is the biggest researchers networking program in Europe. We have almost 300 running actions in any scientific domain. I would like to thank the Università Politecnica delle Marche and the action chair, Gianfranco Manazzi, for organizing this meeting. And I apologize for not being there. Unfortunately, we are going through the Open Call 2023 evaluation of the proposals we have received after the last cut-off date in October, and now we have the consensus phase where basically the triplets have to meet the review panel members and agree on the scores to be given to the proposals that we have received. Open Call 2023-1 has been very successful. We have received more than 500 proposals, and this is the record number since 2016 and the highest number under, let's say, the Horizon Europe period. So we are quite happy with that. We are really working hard in order to have all the processes put in place, respecting the formalities, to then communicate the results of the evaluation in June this year. Regarding today's meeting, Food Was Stop, we met in September indeed, together with some of the management committee members. I have to say that these first 5 months have been quite productive so far, at least from the central level, from the cost perspective, since the number of countries at submission level and now have increased. Now we have 33 cost countries represented with 60 management committee members, and we have 354 participants, which is a quite relevant number compared to other just kicked off actions. This is very promising. It's very good. We know that there are actions which can even reach 500 participants in their working groups. So well done so far. Let's proceed on that track. It's also very good that the first call for short-term scientific missions has been launched. I'm happy that this has taken not too much time and that we have dive into it very quickly since the start of the action. Just a motivation for the future, you might have heard that 3 Nobel laureates have been communicated recently in the physics. Actually, the 3 of them have been participating in cost actions in the past. So this is what COST is about having researchers networking, coming together, talking to each other, better communicating the results. Keep in mind that you might also achieve these results in the future. Go on like this. Very good that you have implemented the cost policy plans in terms of inclusiveness of young researchers, of participants from inclusiveness target countries, and also female researchers when it comes to, for example, the leadership positions. As you know, these 3 policy plans and these 3 dimensions will be assessed at the end of the first year of the action. So continue this way. We want really our actions to be inclusive and open as much as possible. I had recently an exchange with Gianfranco, who mentioned this first month as a very nice adventure. I'm very happy to hear this, and I hope that also the remaining 3 years and a half of your action will be a nice adventure, not only for the action chair, but also for the other researchers involved in the action. Have a look at what we have selected for the Open Call 2022. So the previous call, we see more and more projects, proposals related to food, to waste, to reuse, to circular economy. So I know that Gianfranco has already been active in creating some synergies or potential links with another action. Bear this in mind, we support networking among actions, among running actions as well. So please take this in consideration. I don't want to take much more time, also because there are a lot of presentations. The agenda is quite full, and I look forward to talking to you tomorrow, at least with those who are members in the management committee, to clarify questions you might have and to support the action running and being implemented in the smoothest and most effective way. Thank you very much, Gianfranco.
Thank you, Marco. To you, to Cost, and you provide us a very good support for the starting of this action. That was a nice journey, and in the behalf of the Vice Chair, that is Fernando, that you will hear later during the first session, and of the core group of the Cost project and all the members, it is a pleasure for me to start this day, this first plenary meeting. We had a kickoff meeting in Brussels, in where the management committee started to draw the program for this first year, and we planned this day. We planned some short-term scientific mission that were already addressed to 4+1 young researcher, and since Cost has to promote the knowledge and the involvement of young researchers, further calls will come soon. We will have a training school in Cranfield this summer, likely in July, and Natalia will organize it, and we will hear about. So there are some funding opportunity. A bunch of you used those, and remember to sign the leaflet with your presence. And you know that there are 6 working groups that try to work and to discuss and to present results on the prevention of food loss and waste, because this food loss starts from the field and go to the harvest and the consumer. So we will see some case studies that starts from the field and go along the chain. But after prevention, there is the management along the chain. So we will have the second session that we will start after the coffee break, after a small portion of the first session. Then we will have the third session on the quantification of food loss and waste. That it is not easy, but we have to do, and we have the option, having 33 countries, as Marco just said, we can have a broader picture of this quantification. And all of you, it is a volunteer project. All the ones that wants to be involved in this activity will express their wish to participate, to collect the data, and to contribute with those data. So I'm confident that we will have this journey along the next 4 years, and we will go on with the new tools, and we will see what there is close to the consumer. So to have the feedback from the supermarket, the retailers, the feedback from FAO. And so this is a very important approach for us to have the view from other institutions or for the people that are involved in closer relationship with the consumers. Then we will talk about communication as well, because we have to do our best, but we have to communicate what we do. And so we will have the chance to meet with the working group separately in presence, and I say hello also to the people that are connected online. They will have the opportunity to have the question in the chat during the presentation. Thanks to all of you that took your data with the presentation or with the poster. It is a very valuable contribution that you are taking to this meeting. And we are so many, so for the presentation, the moderators will suggest you to take the time, and the timers will help in this. And so I wish you a good interaction, a fruitful interaction, 2 days of meeting, networking, and connection for new project, new activity, and new joint research. So I welcome to all of you. And now I give the floor to the vice chair, that is Fernando Perez Rodriguez, and to George Carao Glanidis, that will moderate the first session.
So good morning. Let's start. We have a tough schedule today.
Morning, everybody. I would also like to welcome you here in this first COST meeting. We opened the first session of this meeting dedicated to presentations that belong to the working group number 1, that is related to the prevention of food losses and waste. And our first speaker is coming from UC Davis, Barbara Blanco Ulate, that she will speak to us for improving shelf life while ensuring fruit quality using gene editing. Barbara, the floor is yours.
Hello, everyone. Let me set up this presentation. Okay. Got it? Perfect. Okay. Can you all hear me well? Well, thank you very much to the organizing committee, of course, Gianfranco, Fernando, and George for inviting me. I'm going to try to be brief with my presentation as we have a full agenda, as it was mentioned, but I would like to take a moment to also thank everybody. It's been a great time in Italy so far. I had great food, great conversations, and I hope we can continue these interactions. So my name is Barbara Blanco Ulate. I'm a professor in the Department of Plant Sciences at UC Davis, and I'm also the co-director of the UC Post-Harvest Center. I will take just a moment to tell you about the center because I think it's important for this opportunity. And this is not working. Okay, there you go. So the Post-Harvest Center has been in function for 45 years. You may know about it. We used to work a lot on extension and education, but we're starting to reach out more to research. And we have become the co-directors, myself and my colleague, Irwin Donis Gonzalez, since July 2023. As we're kicking off this new era of the center, we're going to have different types of education, extension components, and newsletter, and different projects. So if you're interested in knowing more about the center, I recommend you to go to our website. We have a lot of resources related with fruits and vegetables that can be explored and used and will be updated frequently, as well as more information about our newsletter Oh, I don't know why this is going so crazy. This is not working. So let me see if I can go back. It was true you were rushing me, huh? Okay. So yeah, so if you want to join us in that, you can scan the newsletter and get more about us. And then before I start talking about science, I would like to acknowledge the people that do the research in my lab. And this is a photo from last year. You may recognize a face there. Simone Piancatelli, who's there, has been helping with the meeting, was in my lab. And some of this work, actually, he was involved. And in my lab, we work with 3 different topics: fruit ripening, fruit cell walls, and also fruit pathogen interactions, with a special focus on California crops, as I work for an university from the state, and we use different approaches. So I wanted to take just this moment to tell you about what I do, as I'm going to just talk about a little bit of my research, which is focused on gene editing. I know that I'm in Europe, so people may be skeptical about using genetic modifications or even gene editing technologies, but I think it's important for us to recognize that this is a tool and is available for us to understand gene function, but also can be brought to the market. It is currently accepted in the US as a non-GMO, and a lot of companies that do work in Europe and even based in Europe are approaching us in the US to commercialize this type of products. So with that being said, let's talk about science for a moment. When we talk about post-harvest, and this is linked to this meeting, we have 2 big challenges. Obviously, we need to reduce crop losses and waste. That's why we're here and discussing. But also, we need to improve and maintain crop quality. And these 2 challenges, which I also call them opportunities for innovation and research, are linked. What that means is that if you can improve quality, you can also ensure the reduction of losses. If you reduce loss, you can also have more inputs and energy to improve quality as well. So these 2 things are important. They have a mission, not just for sustainability, but also a mission for feeding the people. A lot of the breeders always say we need to increase the amount of food that we produce, but actually we can argue that we need to improve the quality and also avoid losses. And specifically talking about quality, I focus on fruit. So we focus on the ripening process and how all these beautiful changes happen in a fruit that is hard and green and not so tasty and become a delicious fruit with time. And by understanding these processes biochemically and genetic pathways, we can start thinking about improving quality from different standpoints. I'm talking about shelf life, and is depicted in this image as the green circle, which encompasses also other important traits such as color, nutrition, flavor, and texture. Even if you have a delicious fruit, if it doesn't have shelf life, it's not valuable, it's not worth it, because we cannot make it accessible for more people. You can grow it in your garden, but you cannot send it to more people in different markets. So shelf life, even though sometimes is considered to be antagonistic to quality traits, is really important for us. And as I was saying, understanding ripening, we can find targets for fruit improvement. These targets are generally ripening genes or regulators, and we can use different tools to approach this improvement. For instance, just conventional breeding that we do all the time, but we can have other biotechnology and molecular tools available, as I mentioned, gene editing and some others. And we have discussed this work in a publication that I mentioned there below. When we talk about fruit ripening regulators, I'm going to be focusing mainly in this bottom part of the pathway, which are ripening genes, not so much big hierarchical regulators that may be transcription factors that control many pathways, but specifically ripening genes downstream that control the softening of fruit. And these have been studied for many years. I'm not the first one. I'm not going to be the last one. But we have been using some of the knowledge that have been gained for many years to move this process forward to a more applicable solution for improving shelf life. Specifically working on tomatoes, I do work with many crops, but in this specific example, we use tomatoes because they're important for California, for the world, and they are really beautiful system to work, and people care so much about tomatoes. So gene editing has been used for many different instances. You might be using it in your research. We took advantage of knowledge that we had in fruit softening to study pectin degrading enzymes. These are mentioned here: pectin-lyase, polygalacturonase. You might have heard about them if you're in the biology field. I'll be calling pectin-lyase PL and polygalacturonase PG. These enzymes act during ripening and they break down the cell walls to make the fruit soft. And they work in tomatoes, but also in other different crops as well. So by understanding these enzymes, we first just use this tool and RNAi technology to knock down pectin-lyase and figure out that this is the most important enzyme controlling softening in tomatoes. We published this back in 2016. Obviously, this is a transgenic tomato, so we wanted to approach this same strategy but using gene editing. And we created single CRISPR knockouts of these PL that we knew it was really important. We had published it before. And then the other enzyme that is important in tomato softening, that is polygalacturonase, or PG. PG is more of a secondary enzyme. So we went through a more mechanistic approach that we published, in which we look at how the cell walls of this tomato were. And we figure out that actually the pectin was changed in these knockout lines, especially in the CRISPR PL, making fruit firmer. Now, when you have a firmer fruit, you also have a fruit with potential of having a longer shelf life. And we knew that from our research, right? We see the fruit, they start lasting longer, but we did not measure that so quantitatively in this work. And we decided to do that in the subsequent research. The other reason I was extremely interested in cell walls besides shelf life is that we know that the cell wall is an important compartment that limits pathogen growth, especially to avoid infections of fungal pathogens that can cause losses in post-harvest, fungal pathogens such as botrytis, alternaria, etc., Coletotricum. And we knew that pectin-lyase is a susceptibility factor in tomatoes. When we knock down or knock out this enzyme, just this enzyme, we have fruit that is more resistant. We had published that. And so we knew these tomatoes have better shelf life. They're more resistant to pathogens, but it's only single CRISPR line. So what happens if we have two CRISPRs together and we decided to do a double knockout between these two enzymes? We made it pretty simple. We just crossed the tomato plants, and actually Simone helped with those crosses back in 2019. And by just crossing these two lines, we had a double knockout mutant that we can use in the field in the US right now as a non-transgenic tomato. And just quickly going through the data, we found out that in addition to having just slightly better firmness, the double knockout had no differences with the wild type in terms of quality, meaning that you can extend shelf life without impacting quality. And actually, we saw the increase in some volatiles that are good for flavor. So it's even either the same or better than wild type in terms of quality. But the most interesting part is that we had a dramatic effect in shelf life. And this was maybe obvious in this image, but maybe made more obvious in the follow ones, in which you see the double knockout is in the bottom and the fruit looks much better after 16 days after harvest. But and we have that quantitative data to validate that. But I don't need to convince you. Just looking at this image, you will see that this fruit can gain a dramatic shelf life even at 36 days post-harvest, in which they look marketable without any damage. And this was not obtained in the wild type or the single CRISPR line. So we have a tool in which we can create mutations in these genes that increase shelf life, but we don't have any effects on quality. Specifically, we have these even better flavor in the CRISPR double mutant. And we just recently published that work as well. One other little thing is that the CRISPR PG double mutant is also resistant to disease as it was the single CRISPR PL. And another question that seed companies always ask me is, how is the seed of these mutants? Is there any other impacts in the plants? So the genes that we use are genes that are ripening specific, so there's no impact in the growth or yield of those plants. And in addition, we don't have any impacts on seed quality. We actually saw that the seed had a more uniform maturation process compared to the other lines, even the wild type, meaning that we don't have any problems with seed quality. That is, really important when you're commercializing this type of lines. So with that being said, we are moving this example that I show you in tomatoes shelf life or fresh market. But in California, we also have the tomato processing industry that is interested in other traits, such as consistency or extended field harvest. And we're moving these CRISPR mutations into other tomato lines, including processing varieties, as well as heirloom varieties that are important because of their different traits. So with that being said, I hope I'm still on time. I would like to thank everybody. I would like to just show you that using gene editing, as I mentioned before, can be a tool for research, but also can be a tool to achieve some improvements in quality that will take way too long through breeding, or then may have some potential negative effects, because a lot of the shelf life related genetic basis of shelf life can be negatively correlated with quality traits. So it's almost impossible to have a perfectly delicious fruit with the best shelf life. And we've seen this in strawberry and other breeding programs. So thank you so much for listening to my talk. And I wish to talk to you and have more questions later on. Thank you.
Thank you very much, Barbara, for your very interesting presentation. We have no time for questions, but just one small question from the chat. Maybe you're going to have the chance to discuss with the people that are in the room. So the question that comes from the chat is whether genetically modified tomatoes are allowed on the EU market. If genetically modified tomatoes are allowed in the EU market?
Well, they are not allowed right now. There's a lot of limitations, even for gene editing technology that is not considered a transgenic in the European markets. There has to be a change in mind and approach for that to happen. But as I was saying, even companies, European companies, seed companies that are based here in Europe and have operations outside Europe are interested in this technology because it's really promising. So it's just sometime maybe Europe will change their mind about using different approaches. I'm not a gene editing pro person, it's one tool I use. We just have to have options, right? So the more options we have, the better we can approach these problems of food loss. Thank you.
Thank you very much, Barbara.
So let's move to the next speaker. This is David Gramaje. David Gramaje is coming from the Instituto de Ciencias de la Vid y del Vino from Spain. He's going to present today the fungal tract diseases, the global threat to grape wine health. David, the floor is yours.
Thank you. Okay, so thank you. Thanks, Gianfranco, for inviting me to be here. Good morning, everyone. So I'm going to give you the first example of crop losses, in this case due to diseases. So I'm going to talk about grapevines, probably one of the most important, economically important woody crops in the world. I'm going to talk about grapevine trunk diseases, so one of the main phytosanitary problems right now worldwide. So grapevine trunk diseases are caused by fungi that colonize the vascular system and also the pith, causing this kind of symptoms, so a broad range of external symptoms. You can see on the top left, like reduced vigor, retarded growth. Also, you can see death spores, death arms, chlorosis, shortened internodes, and sometimes discoloration of leaves typical of ESCA disease. So we have a look at the internal symptoms. We have also a broad range of internal symptoms, going from sectoral necrosis, central necrosis, white rot, silent spots, so spots in the silent vessels. So obviously these kind of symptoms will compromise the translocation of water and nutrients from the root to the aerial part of the plant. But let's talk about numbers. Let's talk about the economic impact. For example, in Australia, you can see here that 47% of the Syrah cultivar is affected by one of the diseases, U-type diabag, generating a cost of 2,800 Australian dollars per hectare. In California, these two diseases, Botrytis and U-type diabags, generate an economic impact of 260 million American dollars per year. And in France, researchers consider that 12% of the vineyards are affected by ESCA disease. In La Rioja region, in my region, northern Spain, so we estimated the cost of replanting 1% of plants affected by GTDs, and the cost reaches about 7 million euros per year, without taking into account the losses in wine production until the full yield. But grapevine trunk diseases are not new, so we have a look at the Italian and French viticulture manuals. We see these beautiful photos of ESCA symptoms. Actually, some Italian researchers like Laura Mugnagna and collaborators pointed out the disease currently known as ESCA may be as old as vine cultivation. So why there is an increase of grapevine trunk diseases worldwide over the last 25-30 years? So there are several factors that explain this increase worldwide. So the first factor would be the grapevine planting boom experienced during the 90s. You can see some graphs here from Australia, Chile, US, South Africa. There was an increase in the demand of planting material during the 90s, and the nurseries couldn't supply the first quality planting material, and they sold also potentially contaminated propagation material. And now these vineyards of about 25, 30 years old, they are experiencing this kind of symptoms I showed before. The second factor is the drastic changes in production methods. I mean, intensification, like increase of wounds, and sometimes overcropping. This can favor disease infection. And the third factor is the phasing out of some broad-spectrum fungicides, such as sodium arsenite or methylbromide, and some also specific fungicides, such as the benzimidazole group, which were very effective against grapevine trunk diseases. So this pathosystem is really complex. So when we talk about powdery and downy mildews, we have one pathogen associated with one disease. Here we have more than 100 species associated with 35 genera. We have diseases affecting nursery, young vineyards, other diseases affecting mature vineyards, and we also have diseases affecting all the scenarios. The second factor that explains the complexity of this pathosystem is that we have fungi with different biology and epidemiology. I mean, they live in different places and they can be spread by different ways. We have diseases affecting the roots. Other pathogens are spread aerially and they penetrate into the plant through the pruning wounds. And we have also pathogens infecting the propagation material during the propagation process in nurseries. And the third reason that explains this complexity of this pathosystem, probably the most important, is that no curative measures are known for controlling GTDs. So we have to apply an integrated disease management strategy using different tools, giving less priority to the chemical methods, as we will see in my presentation. So this strategy, this integrated management strategy, should be applied in all lifespan of the plant, in this case, from nursery mother blocks to mature vineyards. And now in this presentation, I'm going to identify the strategies used by researchers, growers, and nurseries to reduce the impact of GTDs. I think later, Florence and Ersebe will talk deeply on this topic. So in the propagation process, we have one main problem, is the conditions in nurseries are favorable for fungal trunk pathogen infection. So the wet and humid conditions in hydration tanks, high root density, and close spacing of plants during causing stage and during the rooting stage. So the second factor involved in nurseries is that practices increase infection risk. We have wounds in the rootstock and scion planting material in the mother blocks, also in the disbudding stage, and also in the dramatic stage of putting together the rootstock and the scion in the grafting process. And the third factor involved in nurseries is that the external symptoms are quite tough to identify. Here you can see a photo with probably a nice thickness of the rootstock, nice aerial part, probably very good root system. But when we cut these plants, we observe these black spots in the xylem from which we isolate vascular pathogens. So they are considered latent pathogens affecting asymptomatic tissues, and they become pathogenic following biotic and or abiotic stress factors. So nowadays, it's not possible to ensure GTD planting material, free planting material by non-destructive methods. So during the propagation process, chemicals are almost forbidden in many countries, especially in Europe. So biological control works very well using Trichoderma treviride or Bacillus subtilis, so these two strains. So for those that are not familiar with biological control, so it's a living organism, in this case, fungi or bacteria that compete with space and nutrients, basically, against these pathogens. Another method would be the hot water treatment. So this strategy has been used worldwide against several pests and diseases, especially phytoplasmas, but some things to consider when using hot water treatment is that some Vitis vinifera varieties are more sensitive to the treatments than others. So the tolerance of plants to this treatment is affected by the climate in which the cuttings are grown. And finally, this strategy is not completely effective in eliminating fungal trunk disease pathogens growth. Regarding disease resistance, so much research have been done on evaluating planting material. So regarding rootstocks, none of the rootstocks tested have shown complete resistance to the main vascular pathogen associated with grapevines. And regarding cultivars, so there is no one single cultivar able to tolerate all broad range of pathogens. For example, here in these graphs, you can see that Concord cultivar is tolerant to Neophysococcus parvum, but susceptible to another pathogen, Pseudomonas chlamydospora. So recently, there is like a trending topic about the relationship between the mean diameter of the xylem and susceptibility to vascular infection. And some researchers pointed out that the small xylem vessels are less susceptible to fungal infection. Regarding the new plantations, we have the same problem. So we don't have chemicals available as a pre-planting treatment, and much research has been done on the use of biological control agents. But in general, the results in general under field conditions are that we were not able to recover the biological control agent after 1 or 2 years after inoculating and after plantation. So, and in general, these studies pointed out the inefficacy to prevent fungal trunk pathogen infection. Finally, in nursery model blocks and mature vineyards, so we have to have a look at the disease cycle, I mean the epidemiology of the diseases. Here I show you in this graph the epidemiology of Botrytis diabac, which is similar to the other diseases I showed before. So basically, from the pruning debris and the infected dead wood present in the plant, this pathogen can produce fruiting bodies, I mean resistant structures. You can see in this graph on the top right. Then when they get good conditions in the spring, high humidity and temperature, spores are released from the fruiting bodies. These spores land in a susceptible pruning wound. The spores germinate, produce the internal cankers, and after some time, we don't know exactly if it's 1 week or several years, we see the external symptoms. So one way to stop the disease cycle, we believe to eliminate the pruning debris or the infected dead wood present in the plant. So there are several ways to eliminate this pruning debris and the dead wood. So burning probably would be the most effective, but environmentally not friendly. So there are other strategies such as mulching, grinding the pruning debris and leaving the pruning debris on the floor, or grinding and covering this pruning debris. So with these strategies, we don't know exactly the impact on the phytopathology. I mean, we don't know if these strategies are enough to destroy the fruiting bodies and the spores. You can see here the size of these spores and fruiting bodies is very small, so much research needs to be done on this topic. And the last one probably is the most interesting one, is the composting. It's much more expensive than the other ones, and growers don't have usually access to these composting places. But here you can see an example. French researchers from Bordeaux demonstrated that composting is effective in reducing to 0% infection of grapevine trunk diseases. So let's think that the pathogen will continue the disease cycle. So the pathogen can produce, as I said before, the fruiting bodies on grapes, but also an additional problem is that this pathogen can also produce the fruiting bodies in other woody crops. Here you can see in this photo a typical Mediterranean scenario where grapevine is planted along with other crops, olives, almond, and whatever. So there is like a hypothesis that there is an inoculum exchange between grapevine and these woody crops. So we reach this point, the red arrow, the pruning wounds. So especially winter pruning is the main port of entry of this pathogen in the life cycle of the plant. So regarding the strategies, the most effective are the use of mastic and paste plus fungicides. We don't have chemical control, basically, so not in Europe. So some products, but they will be forbidden soon. So the alternative is the use of trichoderma again. Trichoderma is a fast-growing fungus and can colonize this pruning wound and compete with space with these pathogens. Trichoderma works very well under controlled conditions, greenhouse, nurseries, but under field conditions in general, all the studies done under field conditions show that these products, this fungi in this case, show variable results for preventing infection by TTD pathogens. So finally, to finish my presentation here, I'm gonna show you a list of strategies that are investigated by researchers right now to reduce the impact of grapevine plant diseases. So the mixture of cultural practices with biological control, also biostimulants, resistance inducers, biofumigation in nursery soils, the use of nanomaterials. For example, the group of Alex Achmeyer in Mendel University is working hard in this topic, ozonated or electrolyzed acid water during the nursery propagation, and also natural compounds. And in this sense, I would like to show you the poster we present here in this construction, the Green Beeties. This poster is in collaboration with my colleague from ICBB, Tere Garde. So we are using the grape pomace, the extracts from this grape pomace to combat the main fungal and oomycete diseases in grapevine. Here I'm showing you some results in vitro with promising results right now. We are going to try to test these products in plant and under field conditions. This will be the next step and probably the most difficult step. So that's it. This is my presentation, an overview about the grapevine plant diseases. And now Florence and Dersevel will talk a little bit more about that.
Thank you. Thank you, David. Unfortunately, we are run out of time, so maybe you can catch up with David later during the coffee break.
Okay, now we will continue, but remain in the same topic more or less. Florence Fontaine from the University of Reims in France will provide us a talk on the strategy studied for an eco-friendly management of grapevine trunk diseases. Florence, thank you very much. The floor is yours.
It's okay for you, no? Not. Oops. How I can share the screen with you? I have something to do, or no? You can help me because I have my presentation, but it's not sharing with you.
Maybe you have.
It's okay now? Yes. Okay, so good morning, everybody. As you present me, I'm Florence Fontaine. I'm a full professor at the University of France Champagne-Ardenne, and I'm a supervisor of a research team working on grapevine trunk disease. And so after the nice presentation of David, I will not present you again the grapevine trunk disease, but just to present you the different strategy we have in my research team and to manage this grapevine trunk disease. So for that, as David said, it's very difficult to manage grapevine trunk disease, not in nursery, but especially in vineyard. And for that, I will only focus on the part of the management. And on the part of the management, according if you have a strategy in the nursery or in vineyard, you have to adapt because the dissemination of the pathogen and also to manage a pathogen in vineyard inside the plant is not the same, but to manage the pathogen in the nursery and in the different bath. So for that, the best strategy is to adapt your strategy according nursery and vineyard, but also not to forget to combine this strategy with the prophylaxis, because we know that in one growers, you can give a minimum of advice on the prophylaxis, but for sure, the prophylaxis is not enough to manage grapevine trunk disease in vineyard. And so you need more help, and especially you need biocontrol agents or natural product. So for that, I will develop the different strategy we have in my lab. Is relatively known strategy, but other people working on that. And we have the first one by using a biocontrol agent. And for that, from the vineyard, we isolated different microorganisms from the soils or from the plant. And after the identification of the pathogen and the cultivation of the pathogen, we work on it. And I will focus especially not on all the pathogen we identify in my lab, but I will focus especially on Bacillus subtilis, and we will see the different combination or not with Trichoderma atroviridae, because Trichoderma, as David said before, is the main biocontrol agent which are marketed to manage grapevine trunk disease. And on another side, I will present you another product that we work in my lab, developed by an Italian company in Bologna. And we have some publication on this product, which call it LC 2017, is composed of a low concentration of copper with plant extra and also a carrier, which are hydroxyapatite. And so I will just show you the different strategy we have, independently that we work on BCA or a natural product. We first evaluated the potential of this product by in vitro test against the pathogen, and we focus on some pathogen. And then we have in planter experimentation using some plantlet or some cuttings in the greenhouse. And finally, if we can, and according the advance we have and knowledges on the product, we can continue the field essays. So at first time, I will present you some results on biocontrol agent and especially on the use of Bacillus subtilis we identify in the Champagne area. And in the first time that we have as a test, we have antagonistic activity, and we could see that Bacillus subtilis is able to stop or to limit the growth of some pathogen associated to grapevine trunk disease. In the second time, we evaluate also the potential of this bacteria to detoxify some compounds produced by the pathogen. As you know, most of the pathogen, for example, Botrytis cinerea, or pathogen associated to grapevine trunk disease, are able to produce some phytotoxin, which are involved in the expression finally of the disease. In this case, especially in Botrytis dieback, we know that some of pathogen produce this kind of toxin. And one of the strategy, why not, is to use the BCA to limit or to degrade the toxin produced by the pathogen. So in your case, we evaluate the potential of Bacillus subtilis to eliminate and to degrade this toxin. And you could see that in vitro assay, Bacillus subtilis is able to concentrate this kind of compound and to degrade this compound. And in the third time, what we evaluate is the potential of this bacteria to stimulate some defense of the plant. And due to this induction of specific defense, we could see a better protection of the plants in the case of an artificial infection by some GTD pathogen. Finally, we evaluate the potential of this bacteria to decrease the expression of some symptoms, especially in the case of the use of the pathogen Neophysicoccum parvum, a pathogen associated to grapevine trunk disease. And that we observe that when we have some cuttings infected and in which we had Bacillus subtilis on the soils, we could see that we have a significant decrease of the expression of the dieback of the cuttings. In parallel, we observe that for the plant infected by the pathogen and inoculated with Bacillus subtilis in the soils, we have also a decrease of the necrosis induced by the pathogen. So due to this positive result, or for sure interesting result with Bacillus subtilis, we decided to associate and to combine the effect of Bacillus subtilis with Trichoderma atroviridae. The strategy is to bring Bacillus subtilis on the soils and to apply Trichoderma on the wound produced by the pruning wood. And why we use Trichoderma? As I said before, Trichoderma is able to direct attack the pathogen, have also an illicit plant defense, and is also able to promote the plant growth. Due to this combination managed by Katarina Lea during the PhD thesis, what we observe at first during the in vitro test, we observe that Bacillus subtilis is at first able to limit the growth of Trichoderma, is there together on the same place. In the second time, we observe that if you put Bacillus subtilis and Trichoderma, we have some strange relationship, but on the same petri dish on the first time, if you put on the same petri dish Bacillus subtilis, Trichoderma and GTD pathogen, for example, Neophysicoccum parvum, you can see that the combination of Bacillus subtilis plus Trichoderma limit the growth of the pathogen. So due to this interesting result, we continue by in planta test, especially in a greenhouse with cutting. And in this experiment, we work on two cultivars. We work on Chardonnay. Chardonnay is considering as less susceptible to GTD, and Tempranillo more susceptible to GTD. And what we observe that due to this combination with Bacillus and Trichoderma, we have a decrease of the expression of the disease by the expression of the symptom and the size of the necrosis. But the answer is different according to the cultivar. And according to the cultivar, we observe that Bacillus subtilis is able to more protect Chardonnay than the use of Trichoderma or the combination of Bacillus and Trichoderma. And for Tempranillo, we observe very few effects or beneficial effect of Bacillus subtilis with Tempranillo compared to the use of Trichoderma alone or the combination of Trichoderma and Bacillus. So you can see that the strategy to use this or this BCA is totally dependent to the cultivar. As I will not develop, as David present too, we use also this combination of Bacillus and Trichoderma during the nursery process. And so during the nursery process, on the different step of the production of the young plant, we added the combination of Trichoderma and Bacillus, or Trichoderma alone and Bacillus alone. And what we observe at the end, and the main conclusion of this experimentation, is due to the combination of both BCA, we have a better protection by a decreasing of the contamination by the GTD pathogen. Now I will present you a short presentation on the part of the natural product. So the second part we work on with my research team. So as I said to you, this product LC 2070 developed by the NDG company is composed of hydroxyapatite associated with a low concentration of copper and with some different plant extra. What we observe at first is by fungistatic effect in vitro test, and we observe that these products are able to limit the growth of the pathogen, not to kill the pathogen, but to limit the growth of the pathogen. On the second time, during the in planta assay, unfortunately, we not observe a decrease of the expression of the symptom or a decrease of the size of the necrosis after an infection of the pathogen. But what we observe is that this product is able to elicit or to stimulate the plant defense response. And probably over the time, this plant defense response could help to manage the infection by the pathogen. After several years to more characterize the effects of this product, we have in parallel some assay in vineyard. And during the vineyard assay, after 5 years of treatment and the different foliar application of this product, what we observe? We observe a trend to the decrease of the foliar symptom expression, especially on the apoplectic form of this disease. We observe that this product have no effect on the vine physiology. When I say no effect, they not decrease the vigor of the plant, and so the stature of the plant, it's exactly the same as the control plant. They have no effect on the juice, and so that it's interesting for the oenological parameters. And finally, what we observe also that there are no changes on the vine microbiome of the plant compared to the control plant. So due to this result, we decided to manage also a study on the use of this product and trichoderma. And you could see that we have only 2 years of experimentation, and it's not enough to conclude if they have a beneficial effect or not of this combination. So just to say you that related to this product, we decided to have a European project. And so we have now a live project, and the supervisor, are the Italian company, so the Natural Development Group from Bologna. And this project is to define the potential of this product, again, gravinculating disease, but also Donimedio. And we will have essay in Italy, in France, and in Portugal. So just to have a link to the waste and GTD and circular economy, that we know is that gravinculating disease pathogen, and especially Botrytis dieback, are able to produce some carbohydrate enzyme. And this enzyme are able to degrade cellulose, hemicellulose, lignin, and so on. And we have a part of my research is to characterize this production of enzyme in the case of gravinculating disease and to better understand the aggressiveness of the pathogen. But another part is to use this good side of this pathogen to produce Kai's enzyme, this enzyme to degrade the lignocellulose, hemicellulose, and so on, and like that to use this pathogen in bioraffinery. So this is a part of a work of my PhD student, Restrepo Leal. So I will thank all the partners associated to this work, and I will thank you for your attention.
Thank you very much, Florence. You are precisely on time. So we have time for maybe one question. Yeah, Dov.
Yeah, we work on lacase, cellulase, and so on. All the casim are the Botrytis pathogen, such as Neophysicoccum parvum, are able to produce all these pathogen. And according to the strain that you have, you can see that they have different degree of activity of this enzyme, but for sure, xylanase, lacase, cellulase, and so on. And due to this enzyme, if you put the pathogen on the good condition, they can produce more this enzyme, and like that, they are able to decrease this compound. And so it's very interesting for the industry part.
Florence, thank you very much.
Thank you so much.
And doctors, sorry, of plant protections. She's going to speak about endophytic trichoderma strains, increase pre-harvest quantity and quality of grapes.
Thank you. So thank you for the invitation, and it was introduced that this presentation will partly connect to Florence's presentation and David's presentation, but I'm more concentrated on the trichodermas, whether they can increase the pre-harvest quantity and quality of grapes. So it was introduced in the first presentation that to improve the product quality may reduce the pre and post-harvest losses. So we also have to concentrate in the pre-harvest strategies. European Green Deal within the farm to fork strategy would like to decrease the pesticide usage till 2030. There are also consumers' requirements to lower the pesticide residues and their chemical pollutants in the environment. And there are further problems with chemical pesticides with emergence resistance among the pest, which is also going to be introduced in the posters or present in the posters. And there is toxicity, not only towards the microbes, but some of the pesticides may have toxicity for the plant, the host plant itself. So there are increasing demands for alternative treatments, and trichoderma is one possibility to be used because it's mycoparasitic antagonistic activity not only on basidiomycetes but ascomycete fungi, and their mycoparasitism is very known from a very long time. As it was told previously, they produce secondary metabolites which prevent the spore germination and growth of the pathogens. The other action is cell wall degrading enzyme production, which destroy the pathogen cell walls, and they can make a leakage and as a predators just eat the pathogenic fungi. And based on the rapid growth mentioned by David, they are also act as a space parasite and space competitors and nutrient competitors. They also act on the plant with inducing the resistance, increase the resistance toward the pathogens, as well as promoting the plant growth, development, and nutrient uptakes. So they also act as a biostimulant. I would like to introduce a little more deeply our endophytic trichoderma strains, which were isolated from Hungarian commercial vineyards with a very high grapevine tongue disease incidence, more than 34%. It was an old vineyard, and only within a half a hectare, we could isolate 10 different strains. And they were isolated from woody tissues, so they are endophytic ones and not from the soil and not from the surface of the plants. They were identified first based on the ITS sequences and refined this identification based on the TEF1 sequences. So finally, we identified 6 different species, and those were the first properly identified endophytic trichodermas from grapevines from Europe. One of them belonged to Longibrachiatum clade, Longibrachiatum orientale, I'm sorry for the error, and which is a potential human pathogen, and it was underlined the potential human pathogenicity by the increased growth at 37 centigrade. So we concentrated to other strains, and among them, Trichoderma simonsi, Trichoderma afreharsianum showed the highest growth rate at ambient temperature. So they may be the best space competitors. And within the Trichoderma simonsi, TR05 strain had the highest and most rapid growth, not only at ambient temperature, but as low as 5 centigrade, which may have a potential during the pruning wound cover at very low temperature, which is in Hungary. Those 2 strains are in a marketed product called Trichoimmune. It is now marketed in Hungary, but maybe gonna be more widespread. It was registered as a biostimulant. The biocontrol potential were tested in confrontation tests when several cases, 100% of biocontrol index was achieved. It means that the strains completely overgrow the pathogens. This picture about Eutipalata within 4 days of growing in a confrontation test, sporulated under mycelia, killing the pathogen colonies, and by microscopic, we also could detect the mycoparasitic activity by hyphae culling and penetration. Those 2 strains had a very high biocontrol index against not only the GTD pathogens from Vitis vinifera, from grapevine, but also from other host walnut. We also tested, and we tested some mycota, and they also had a very high biocontrol index against them. The biostimulant activity was checked on field research. They could decrease the plant loss. The treatment happened pre-planting, so the nursery, the plants from the nursery were soaked in the spores of the trichodermas. The biostimulant effect was shocking for the producers. He sent me the picture 5 months after the planting and this planting treatment. And the biostimulant effect could be detected even 4 years after one single trichoderma treatment and bud burst ratio. And also the yield was increased between 10 to 20% after 2 years. Similarly, 2 years after the trichoderma treatment, the quality parameters of the fruit juice, so the mast parameters, also increased. Higher sugar content, which represented in a higher Brix value, Brix as well, and glycerol content, which is important for wine production, and potassium content was also elevated. This biostimulation effect was proved by official field tests on white varieties and red variety as well at different wine regions. And the plant colonization up to 4 years were detected both on local white varieties and international red varieties. So the conclusion, we have to take care about the isolates with potential biocontrol agent and with biopotential biocontrol activities and biostimulant activities, because there may be some potential human pathogens against them. So there is a necessity of proper identification and characterization even for biostimulant agents. And the advantages of endophytic biocontrol strains with biostimulant activity isolated from commercial fields are that they are adapted to agricultural practices, or strains were from commercial vineyards and were tested in commercial vineyards, and they could survive the pesticides usage. They adapted the climatic conditions, so it was important in Hungary. They could survive the cold conditions, and they are active at low temperature. They adapted to host and its microbiome, so they can stably colonize the grapevines, and they have a long time effect. They have a lower risk for soil microbiome because they have a tendency to invade the plants. And because of these endophytic characteristics, they have lower pesticide, they have a resistance from further pesticide application and application of antagonists just like bacilli, because they colonize the plant and they are in a protected region area. So thank you for your attention. Those results were summarized in the article, and there is, as I mentioned, a product in the market. I'm sorry. Thank you.
So thank you for your interesting presentation. So also thank you for stick to the time. So any question from the audience? So I have a question for you. So I mean, I think that your results are so promising. So you are able to get from the environments and some species that can be used for improve and also to reduce the food waste. So do you think what is the time frame when you want just to implement this into reality in a kind of commercial?
It is in the market as a biostimulant now, and we are about to find some support, financial support, to develop as a biocontrol agent as well, because it needs time. and it needs a lot of money for the registration. But the results are promising, and they are acting not only on grapevine, but on different crops.
Okay, very good. So any other question? So thank you.
Thank you.
So now let's move to another field after 3 presentations on grapevine diseases. We will continue with Piotr Kulavik from the Department of Animal Products Processing in the University of Agriculture in Krakow. And the title of the presentation is the use of multilayer heterosanfusular nanoemulsions with oregano essential oil for preservation of perishable food products.
Yes. Hello, thank you for having me. I know it's a difficult, let's say, topic to pronounce, but it's actually relatively simple. So I represent more the carnivores here because we mostly talked about the plants, but I will represent more about the meat and the fish because we also are saying a lot about and the problems of food waste in those terms. So what are the objectives of this presentation, of this work that we did? We actually developed a new type of emulsions of the coatings, of edible coatings, that we applied on the perishable food products, such as pork loins or Atlantic mackerel. And so we applied them using the multi-layer system. So we applied different layers of the emulsions, and then we stored the products for 18 days at 4 degrees and wanted to check what's happening to the shelf life. So to show how we actually did it, so the emulsions actually consisted of either 0.1% of hitozan or 0.1% of furcellarum and 0.06 of oregano essential oil. And we applied them using the electrospraying technology. So the basics of the electrospraying is just that we put the product onto the between the electrodes, I would say, and we spray them, and there's a current activated. In this particular case, we used the 20 kilowatts. And so the idea of the electrospraying is that when you spray it, the layer of the coating is much more evenly distributed because of the electrical resistance. The droplets will actually go to the nearest side to make the thicker, I would say, layer. So first we use the first layer of the emulsion. We use the hitozan and oregano essential oil mini emulsion with the size of approximately 220 to 150 nanometers. And the idea of this actual layer is that it has antimicrobial properties. So we use the first layer to actually cover it with antimicrobial properties of hitozan and oregano essential oil. And then we left it for drying, and then we applied a second layer of coating, which consisted of furcellarum and oregano essential oil nanoemulsion. This actually emulsion doesn't have too high antimicrobial properties, but we use it actually to improve the barrier properties because it improves the water vapor permeability and different types, so makes the coating more, let's say, less permeable for different gases. And then we left it for drying, and then we applied the third layer of the coating, which is again the hitozan and oregano essential oil nanoemulsion. Again, the idea in here was that it's antimicrobial. So this way we had three layers. First, antimicrobial, which is supposed to destroy the bacteria which are on the surface of the product. Then we make a second layer, which improves the permeability of the whole layer. And then we make the third layer to actually preserve from the cross-contamination that can go from the environment. This was actually based on different preliminary experiments that we checked for 1 layer, 2 layers, 3 layers, 4 layers, etc., and we decided that 3 layer actually is one of the best. Then we took the product, either pork loin or Atlantic mackerel, and we used the tray sealing machine to seal them in polypropylene packages. And then we stored it, as I said, for 18 days at 4 degrees and checked for microbiology, sensory. We actually did a lot more color, texture, oxidation, etc., etc., etc. But in terms of microbiology and sensory, because those are the two, I would say, most important in this case, we checked for total viable counts, psychotrophic bacteria, yeast and molds, pseudomonas also, and we did a sensory evaluation. The sensory was done by the eight panelists, and we also checked the consumer acceptance on different consumers. And all the experiments were conducted with 3 independent repetitions. So we actually did this 3 times to ensure that the results are proper. So first of all, I would like to say what are the antimicrobial properties, because I said that Hitosan oregano mini emulsion that we developed has antimicrobial properties. And yes, those are the minimum inhibitory concentrations we did on in vitro testing with those emulsions. So we can see the percentage here is actually the percentage of emulsion in the solution that resulted in complete inhibition of growth of the bacteria. So as you can see, the Hitosan emulsion is actually effective against all of the tested bacteria, a little bit less effective, but still effective against fungus and yeasts. So in terms of furfuran, on the other hand, the furfuran doesn't have, I would say, antimicrobial properties. There are some antimicrobial properties against yeast and fungus, but those are mostly done by the oregano essential oil, because we also tested furfuran on its own, and actually furfuran not only does not have antimicrobial properties, it actually served as a, I would say, a nutrient for the bacteria, because it improved the growth. So that's the basic idea. This is how we developed those 3 multi-layer coatings. Now, for the results, just to show you, first the Atlantic mackerel, the gray one, the gray line, I think it's in your screen is the gray line is the control. Meanwhile, the brownish is the coated samples. As you can see, the figure A shows the total viable counts, so the basic, let's say, aerobic bacteria that are growing in 30 degrees. And we see that there's a high inhibition rate done by the coatings. The black line indicates the limit of acceptability, I would say the critical limit, which is 6 logarithms of colony-forming unit in terms of Atlantic mackerel. We use the same for pork loin. As you can see, the control samples, somewhere approximately on day 10, the shelf life, the microbiological contamination already exceeded or was on the limit of the critical limits. Meanwhile, after even 18 days in the coated samples, we still did not observe higher than 5 logs of colony-forming units. So that's quite a lot. The difference here is more than 3 logs in terms of, for example, on day 10 and day 14, we have 3 log difference between one samples and the coated samples and the controls. We also checked the yeast and molds, which is the figure B. As you can see, after day 10 and control, the yeast and molds started to increase quite rapidly. Meanwhile, they stayed relatively stable in the coated samples. And the same with C, which is psychotrophic, and the pseudomonas, similar trends. When we check the pork loins, for example, we see a similar trend. So the coatings also inhibited the growth of the bacteria. We can see here even the inhibition of approximately 4 or even higher than 4 logs of colony-forming unit per gram in terms of TVC. But however, the shelf life was extended from approximately, I would say, 5, 6, 7 days for the control to approximately 14 to 15 days for the coated samples. And if we check the sensory were good. Here I indicate only the evaluation by the sensory panel. As you can see, all the sensory scores dropped during the storage. However, in terms of Atlantic mackerel, it was still on the acceptable limit by the sensory panel after 18 days. And in terms of pork loin, you can see that also the pattern was similar to the microbiological contamination. So after approximately 15 days, the samples were not acceptable for the sensory panel. Just not because I showed you only the graphs, but now to show you the pictures. This is how the control of the pork loin looked like after 14 days. You can see the brownish parts on the edges. This is actually the rotting of the pork loin. The smell was also not very good. Meanwhile, on the coated samples, we can see there's no changes like that whatsoever. Just to show you just for a comparison, On the left side, you have the control. On the right side, you have the coated sample. So the difference is not only in the sensory, not only in the microbiological way, you can see it plainly with just a visible naked eye. In terms of Atlantic mackerel, after 14 days, you can see on the control, there's those slight white dots, most probably Pseudomonas, which are growing there and different types of bacteria. Meanwhile, on the coated samples, we don't have any changes like that. And when you check the consumer, because one thing is to check how it works microbiologically, but the second of all, someone has to buy it. So the consumer has to say if he likes it or not. So first of all, we want to check what are the sensory properties of the product and what is the value for the consumer, because the sensory panelists sometimes said in some cases that they can smell the slight but distinct aroma of the oregano. And if you apply it on the Atlantic mackerel or different kind of meat or fish, and you mean about fresh meat or fresh fish, sometimes the oregano might not be good. Might be good or might not be good, depending on the consumer. So we wanted to check because the sensory panels are very trained to distinguish this. So we wanted to check on the different non-trained consumers how it works. So the research question was whether the consumer will notice that the coating is there and how it will affect the product's value. And in terms of, we use it for salmon fillet because we decided not to use it on the Atlantic mackerel because Atlantic mackerel might not be considered approachable, and for the consumer, we decided that salmon fillet is something that the consumers are more aware of. So both odor and its intensity were comparable in both groups. So actually the consumers were not able to detect which one is coated on or not coated. I should also state that the consumers did not know that some samples were coated or not. We didn't tell them nothing. We just said, do you smell any difference or do you see any kind of difference? Also, the consumers could not distinguish control from coated groups. So they did not see that there is any kind of difference in appearance. Also, the presence of coating significantly increased the overall quality score, the freshness score, and willingness to buy. So actually, the coatings were not visible for the consumers and improved the value of the consumers unaware of its existence. And this was tested on approximately 100 consumers. We did the second trial of the consumer analysis on different consumers on the pork loin. We had a slight different approach in here. We told them that one of the samples are actually coated, and we wanted to ask them which one are coated. And this was like in 3 repetitions, so they had a lot of different samples. And consumers were more convinced that actually the control sample had a coating on it than the samples with the coating. So they could not distinguish because we were afraid that because we put those 3 layers inside. So we were afraid that they can see, for example, the gloss, or there will be some kind of slight thickness or something like that. But the consumers, it starts ticking. It's like a bomb. It says that's 50 seconds. Okay, I will speed up. So we wanted to ask, but the consumers could not see that there's the coating is there. And the difference was statistically significant. However, the effect size was low. So nevertheless, it actually means that consumers would not see the coating if we sell them on the market. And analysis of the sample gloss measured using just about right scale in 1 to 7, where 4 was ideal, showed that the gloss of the treated samples was more close to ideal than the control samples. So actually, because there is a slight gloss related to the coatings, but it seems that the gloss was more approachable for the consumers than the control samples. So the conclusion is consumers did not notice the coating on the product, and it was okay. So what about I'm almost finished. So what about the application cost? Because one thing is how the results go, but now we want to try to apply it because this is the idea of the whole research. So we want to go from the lab scale to the industry scale, but for the industry scale, and mostly the problem is with many technologies like that, is the cost of the operation. So based on the lab scale, actually, on the prices that I got on the lab scale, I believe the prices will go down if we'll use the industry scale machines, because we'll use less of the coating, because I had a small machine, so I had only batches of there. So, but based on this lab scale experiment, for 1 kilogram of pork loin, we have to use approximately 100 meters of phytosan oregano essential oil and 15 meters of furfuran oregano essential oil, and less for Atlantic mackerel, because Atlantic mackerel is more thick. So actually, and this is on the surface, so it was less. So the ingredient cost in these terms is actually 10 euro cents or 3 euro cents per kilogram. So I believe this is actually not very very high price for the increase in approximately 7 to even 10 days of shelf life. So in the conclusions, coating significantly inhibited microbial growth. The coating significantly improved sensory scores of source samples. The oregano aroma could be noticed for some of the sensory panelists, but not by the consumers. And the difference in the appearance between treated and control samples was not distinguishable for the consumers, and the coating successfully prolonged the shelf life of the model and meat fish product. There are few problems which are associated with this kind of technology, so I just wanted to present you that we can show this, we can use those kind of edible coatings, and I believe they are even better than the films that some people are developing for the preservation of the food product. And I don't have time, so probably I have to finish. Okay, so about the problems, I will just say it later on. Thank you.
Thank you very much, Piotr. Actually, yes, you have covered almost all of your available time, but I would like to give the chance just for a short question, if there is any. I think Barbara.
Yeah, we actually did it. Then we baked it, but just for the sensory testing, no longer for any kind of shelf life or microbiology. So because now there are two approaches which can be done in terms of cooking it. We can either wash out the layer, because the layers are really, really thick. It's like few milliliters on the whole surface. If you wash it, it's completely not noticeable. When we baked it and tried it, we have not seen any kind of relevant differences. The differences were, but this is, I think this would have to be solved during the industrial stage. The differences were during the storage, actually, because we had more, I would say, watery liquid on the bottom of the tray. And that's the problem, but that would have to be solved with some kind of a sorbent or something that would take out. But during the baking and afterwards, no, there was no distinguishable aroma, no distinguishable taste and changes in the taste of the meat. At least for the sensory panel that we used.
Okay, thank you very much.
Thank you very much.
Okay, let's move to the last one before the coffee break. Anna Maria Mincuzzi from the Department of Soil Plant and Food Science from the University of Bari. She's going to talk about antifungal activity of the pullulan macroalgal extracts. Anna Maria, the floor is yours.
Thank you. My presentation concerns the antifungal activity of the pullulan seaweeds that were used to control the post-harvest diseases of pongnets. Although this is just a preliminary trial and an in vitro trial, algae are well known to be rich in active compounds such as phenols, terpenes, and tannins. And the polyphenols in particular are very important to control both the diseases of the pongranate and to improve the disease resistance of the host. Indeed, we decided to test their antifungal activity on phenols obtained by macroalgae. Generally, macroalgae that were used to obtain these active compounds are brown ones and red ones. These are used to produce biostimulants and fertilizer that generally are made of oceanic species, such as the Ascophyllum nodosum, a brown alga that you can find in the Atlantic Ocean. Ascophyllum nodosum is generally applied by spray application or root application and is able to improve the resistance to biotic stresses that could be caused by pests, fungi, or nematodes, but also to improve the resistance of abiotic stresses, such as weather or nitrogen disponibility. And in this case, we decide to, in the previous trial, we tested also the effectiveness of red algae, such as in this trial made on table grape and sweet cherry that were treated by a commercial product made of red algae. On the left, we can observe the water control, instead on the right, the chemical one. Our issues concerns the availability of seaweeds that were beached on the Apulian coast, as you can observe in this picture. Because we have a very spread and huge biomass that can be used, because generally is removed during the summer. In particular, we used 3 species, 2 red algae that were the Alopitis incurva and the Laurentiella marilze, and 1 green alga, the Codium vermillaria. Other trials concerning the brown alga are in progress. The first step that was requested to obtain a seaweed extract is the hydration, considering that the 90% of the biomass of an alga is made of water. Indeed, we wash it and we cleaned by the epiphytes the alga. Then we dehydrated at a low temperature to prevent the degradation of the polyphenolic compounds. Then we dipped the algae in liquid nitrogen to obtain a powder that was used to prepare the extract according to the Briganti et al. Protocol that was slightly modified. We used an ethanolic solvent that was acidified with the chloridic acid to improve the extraction of the polyphenols. And then we stored the extract at -80 Celsius degree to prevent degradation. We decided also to evaluate the amount of polyphenolic compounds that were within the extract according to the DeWanto protocol. So we had the cold extractions of the phenols and then a spectrophotometric assay. Results displayed that Laurentiella marilze and Alopitis incurva are the richest in polyphenolic compounds. Laurentiella had about 12 micrograms per milliliters of gallic acid equivalents. Instead, the Alopitis incurva reached the 56 micrograms per milliliters. It's important to observe that the green one, Codium vermillaria, had just 3 micrograms per milliliters of total polyphenols. We tested this algal extract against the post-harvest pathogens of the pomegranates that were Alternaria alternata, Coniella granati, Coletotricum acutatum, Aspergillus welwitschii, and Penicillium glabrum. These were tested in a microspectrophotometric assay according to the Brockert, in which we have a correlation between the dry weight of the conidial suspension and the absorbance. So in a PDB solution, we added the seaweed extract and the inoculum. Then we incubated the microwell dishes at 24 Celsius degree over 66 hours. Data displayed in these graphs are very significant because we can observe that Alternaria alternata growth and Coniella granati growth are inhibited by the application of this seaweed extract. And this is particularly important for Coniella granati because it's a non-specific pathogen able to hydrolyze the polyphenols. Coletotricum acutatum growth was reduced by the 30%, instant Aspergillus welwitschii by 50%. The effectiveness was observed for Penicillium glabrum. Indeed, we can conclude that the seaweeds are a very interesting matrix because are rich in polyphenols, and also it's very important to evaluate that the Codium vermillaria extract that was made by the green one is 1/4 lower than the extracts obtained by red seaweeds. Indeed, we can think that this is related to qualitative effectiveness of the polyphenols inside this extract. The effectiveness of all the extracts is comparable because we had a full inhibition in alternaria alternata and Cornelia granati, and their growth reduction in Coledotricum acutatum and Aspergillus welwitschia that were reduced by 30 and 50% respectively. Instead, no fungistatic or fungicide effectiveness was observed. It's just a preliminary trial, so we had to arrange further trial to assess the chemical characterization of this particular matrix and to decide if there's some dose effect or a minimum inhibitory concentration. So, thank you.
Thank you, Ana Maria.
Thank you.
So we have time for questions before coffee break.
Ana Maria, I would like to ask you, how did you apply these microalgal products? By immersion of the fluid in a solution or
No,
by spray?
It's a micro assay. So we had a micro well in which we had the PDB and we amended the extract and the inoculum.
Okay.
It's just an in vitro. But we
Okay.
think to continue this trial with the
Oh, so
in
these
vitro.
are just in vitro data.
Yeah.
Okay, thank you.
Thank you.
Any other question? We can just, okay, so let's just
Okay.
stop here. I think Gianfranco has some information.
Hi, all, just a technical announcement. No, I want to acknowledge all the speakers, excellent talks, and excellent timing. So an applause to the moderators that were, and to the speakers as well. Just a technical announcement, we can have the coffee break now, and before lunch, so the next coffee break, you are invited to stay here all together for a joint picture. Okay? Have a good coffee break.
Okay, so welcome again after the break and the poster session. So hopefully you enjoyed the coffee and also the poster. So we'll start now. The first presentation after the coffee break will be done by two speakers, if I'm right. Pablo Guarnaccia and Fabio Cresta from the Department of Agriculture, Food and Environment from the University of Catania, Italy. The title of the presentation is How Can Agroecology Contribute to the Reduction of Food Waste and Loss? So they will split into a short speech, okay, each one. So the floor is yours.
Thank you to the moderators, dear colleagues. I'm Fabio Cresta, and together with my colleagues Paolo Guarnaccia, we are agronomists. We work on plant productions. We will share this presentation on agroecology and food waste. I will deal about the first part on agroecology, and Paolo will speak about the second part on the effect on food loss and waste. We will make a very quick change. We don't want to waste time in a conference against waste. An estimation of the state of food security and nutrition 2023 by the FAO says that 735 million people are hungry, malnourished, and have difficulty acquiring food. But at the same time, in developed countries, even certain easily exportable food products are underused and go to waste, or sometimes are over-imported and over-consumed, like cereals or legumes. And also in developing countries, food is lost due to ineffective internal transportation and storage. It has been estimated that of all produced food on our planet, 1/3 is wasted. So what we may say is that hunger is not a problem of scarcity of food, but is more often caused by food loss and waste and inequality of distribution. Also, an estimation says that 1.3 billion tons per year of food never reach the table. And what else is wasted more than food? Is wasted a huge amount of water, dissipation of fuel and energy, land soil exploitation and deforestation and soil nutrient loss. Today there are 8 billion people on the planet, but in 2050 there will be nearly 10 billion people. So a huge problem is increasing, not stable. And what we should admit is that in my opinion, the paradigm of the current food system that has driven us until today has failed in some way. And to face this problem and to face this huge increase in population, we need a transformational change in all productive sectors that truly addresses the different dimensions of sustainability from economic, environmental, and social point of view. A possible solution could come from agroecology. Agroecology, compared to the conventional food system, could have a leg up. But what is agroecology? Agroecology is a polysemic concept with over 30 definitions adopted by government, academia, and civil society organizations. It applies, and despite, the word agroecology that seems to refer only to sustainable agriculture, it applies ecological concepts and principles to optimize interactions between plants, animals, humans, and environment, but also take into consideration social aspects needed to sustainable food systems. Agroecological approach simultaneously applies ecological and social concepts and principles both to the design and management of sustainable agriculture and food system. So agroecology represents a transdisciplinary field that includes the ecological, sociocultural, technological, economic, and political aspect, encompassing the entire production chain of the food system. In some way, it is like a multi-purpose tool. And the tangible challenges of agroecology, first of all, it relies on biodiversity. It promotes internal inputs such as nutrient cycling and locally adopted genotypes, rather than external ones like fertilizer, pesticide, concentrate, or feed. It is very sensitive to knowledge and innovation, attracting rural youth. It pushes for social equality, creating jobs and respecting gender equality. And also it promotes, last but not least, I would say, promotes local markets, direct consumer producer exchanges. The FAO drawn up 10 elements of agroecology. I can have not the time to describe in details, but the most important are the diversity, the biodiversity, the co-creation, the participatory approach, the synergies between people and community, such as the efficiency of produce more using less, recycling, reducing cost, the resilience of plants, but also the people, and human diet, the human and social value, as well as the culture and food tradition. And it may obviously need responsible governance and circular economy. So how can agroecology contribute to the reduction of food waste and loss? We think that it may have a very important contribution. I will let Paolo explain for you.
So, good morning, everybody. I will talk about how agroecology could determine sustainable food system in the world. There is a transition period. The first step that we are living today is the increasing of the efficiency of practices and resources and substituting external inputs. Then we are going towards transforming agricultural production system to be more resilient and sustainable. But one important step is to create new markets, new possibility for local farmers to provide food for local consumer. And then there is all the governance and political issues, legal issues that will help this transition. And to reach the sustainability in the food system, we need a different approach. One of these is the systemic thinking. The crisis we are living is not a particular crisis, it's the crisis of the system, so we need a systemic thinking approach. We need a different competence and skill And today we have an example about cost action, how different competence and skill are gathered together. We need a participative approach, and we need to act on the problem causes, not only on the effect. We need to give a look to the past about sustainable model for production and consumption, with the idea to design a new sustainable future. And then we need to act at locally. We have to create a new model in the territory, considering the difference between the territories. And then the last approach is the approach that WHO organization developed in 2015 about One Health, the health of the person and the health of the planet. In 2016, the Stockholm University Resilience Center developed this image that is called the wedding cake, the Sustainable Development Goals wedding cake. This shows us how food is linked with all the 17 objectives. And this gives us the importance when we want to make a change, how food could be the first thing that all people in the planet, all their life, 5 times a day, we eat food. So it's something that will help at transformation of all our societies. About sustainable diets, we see in the picture of Johnson et al. How sustainable diets are linked with well-being, health, biodiversity, environment, climate, equity. I mean, all the environmental, social, and economic aspects of sustainability. The diet is related with so many aspects like crop system, climate change, land use change, water consumption, pollution, landscape, biodiversity, food waste, renewable energies, local economy, jobs, culture, and health. And we have to consider all these relations. About the sustainable food system, they should be sustainable, inclusive, resilient, local. They should provide enough healthy food for 10 billion people in 2050, stimulate a change towards diets healthy for humans and for the planet, produce local, assorted, seasonal food, protect small producers and processors, avoid waste production, protect biodiversity and landscape, use renewable energy, reduce consumption of resources, low external input and greenhouse gas emissions, favor the local economy. We have to reevaluate the role of municipal wholesale market, local farmers market, and also the small retailers like bakeries, greengrocers, butchers, fishmongers. Generate stable and decent job, and the sustainable food system should be linked to local tradition and cultural identity. We have to reduce loss and waste, and this is in the agenda to 2030, the objective 12, especially 12.3, aim to halve food waste and to reduce food loss by 2030. So food loss and waste, consider all the aspects of the food supply chain, from the production to the harvest, to processing, packaging, transport, wholesale and retail, and our consumes in hotel, restaurant, catering, and so on. And also about the discard of the food in the landfill or so on. So it's something that is related with everybody every day. And this is a picture to see how no waste is one of the main principles of a sustainable food system, together with ecological, responsible, fair and accessible, local and healthy. Thank you very much.
Thank you for the presentation. So, questions from the audience? There.
Thank you for your presentation, which was very interesting. So what are you suggesting should happen at, say, EU policy level? Do policies need to be implemented to support the development of agroecology compared to more conventional food systems? How do you see the solution really taking root?
I think it's a very important question, because without creating new models, new markets for small farmers, it's impossible that the local small farmers, young farmers that are starting to do sustainable agriculture could sell their products to the supermarket. Impossible. They speaker was going through the municipal market. It was like a hub for local producer and for all the small retails in the cities. So we have created new kind of this platform. I think in the world, in the States, also in Northern Europe, they are running a food hub. I think the food hub is the good solution, a small platform, 200, 300 square meter, where local producer bring their products, and a local consumer, small restaurant, small hotel, and private families can go, and they come connected together. And about food value, 80% goes to the farmers, not like supermarket that only 10 or 15% goes to the farmers in this moment.
Any other question?
Thank you for your presentation. Very interesting. In developing countries, there's a problem of infrastructure, and it's not always easy to take the food or the produce to these food hubs or to this small town, and they rely on middlemen. How would you suggest we can mitigate the effect of middlemen that create problems with food loss and waste because of additional handling and so on?
I think in developing countries, they suffer as we are also suffering in Europe when a product that is produced with subsidies from the government arrives, big ships, 50,000 tons of wheat coming from Canada or Ukraine, arrive in the port, in the Mediterranean port or in Africa. They let the price of the local produce down. So the small farmers, they stop to grow anything because in the port arrives very cheap food. And this food doesn't belong to the tradition. It's not also, I think, healthy for it. No, in Africa they always ate sorghum, millets, and instead now they are arriving wheat, soybean. It's completely full, and so the small farmers they have to abandon and they start the migration also because of this. So I think the same thing, sustainable food system has to be applied all over the world. Any difference between developing countries or developed countries, that when we have to buy food in supermarket, also in Italy, 80% come from other countries, 3,000 kilometers from the producing area to our house. So. Food has not to be transported like other goods. I think food is not a goods, it's something very important. No, it's not something that the price of wheat in this moment is decided in Boston or Chicago auction and so on. It's very...
Sure. Yeah.
Totally. What can I say is that in developed countries is more a problem of waste of food, while in developing countries, more a problem of food loss. So what we may say, first of all, is to promote the producer, the food producer, or local food producer, to produce more and to give more contribution to food products, but also to the economy of this population. And this is something that the FAO is trying to do at the moment.
Okay, thank you very much.
Thank you very much.
And now let's close our session with the last presentation by Stella Todorova, that she comes from the University of Agriculture in Bulgaria. She will present, talk about the short food supply chains as drivers of sustainability in rural areas. Stella, the floor is yours.
This is not the one. Sorry.
But mouse, no, enter on.
This is yours, right? This is your presentation, right?
Yes, but how to touch or?
No, just here. Here, this.
Okay. And
You
we
want
have a short video.
I don't have it. She's asking for a short video.
Okay, to put
Yeah.
this one.
I don't know it's going to work.
Yeah, you put in one folder, one presentation in order to buy.
You already have it
One
here anyway.
presentation and 2 files for movie. Yes. For movie. But, would you help me when I finish presentation with this file?
Okay.
Sufina Farm must start Sufina Farm.
So let's start and then we can see if the file is going to work. But anyway, the time is running. So just try to start now and then we'll see.
Only here.
Yeah, just move on. Forward is this one. Just start this, this one. Okay.
Forward this one. Okay. Dear colleagues, let me start my presentation. My name is Stella Todorova, and I work as a professor in the Department of Agricultural Economics and Management at the University of Plovdiv, Bulgaria, Agricultural University. My presentation is about short food supply chains as drivers of sustainability in rural areas. According to the regional typology of urban and rural areas, half of the territory of European Union is covered mainly by rural areas, and approximately 20% of the population is concentrated in them. The role of rural areas as producers of food and other important tangible and intangible goods is well understood. The objectives of sustainable rural development is logically linked to the direct marketing systems. Food is lost or wasted throughout the supply chain from initial agricultural production down to final household consumption, according to Gustafsson et al. Agroecology and organic farming are highlighted as one of the sustainable agricultural practices that can help achieve the objective of the European Green Deal and related farm-to-fork strategies. local food system and short food supply chain. These are two concepts, two different concepts, but they share some similarities. The location of production and the length of food supply chain have been and continue to be of interest to many researchers, and they have been well studied. Most researchers define local food systems as those in which the production, processing, marketing, and consumption of food takes place in limited geographical area with a source radius of about 20 to 100 kilometers. A key feature of short food supply chain is that these are places where the number of intermediaries is kept to a minimum. There are different short food supply chain in terms of number of these intermediaries. The ideal case is direct contact between producer and the consumer, between farmers and consumers. The marketing of food through a short supply chain, which business is organized mainly by small and medium-sized organizations without a special marketing unit, is constantly growing, especially in Bulgaria. Short food supply chain can include a variety of food production methods, organic farming, agroforestry, permaculture, and etc. What are the impacts of local food system and short food supply chain? They are fair market structures and contracts for farmers that help support the uncertainty of their trade and reduce food waste and food loss to a greater extent, including on-farm loss. Reducing food loss at the different stages of the supply chain. Here we include awareness of the changing food standards and regulation, collaboration across the supply chain, formation of communities of practice, developing a resilient supply chain, lean and total quality management. Some of you know that total quality management, this is methods for monitoring the quality of products, processes, and services. Other impacts of this is short food supply chain in social. They support and facilitate the connection in interaction between farmers and consumers. Another effect is economic impacts, and there are records that local farming system and short supply chains have a higher multiplier effect on local economies than long chain. For example, maintaining local employment, especially in rural areas. Another effect, this is environmental effects, and usually the environmental benefits that are cited in the literature and we support are food miles and carbon footprint for a local food, positive impact on biodiversity, and reduce the use of agrochemicals for organic farms. The case study in my presentation is one of the organic farms in Bulgaria. The name of this farm is Sufina Farm, and location of this farm is located in one village. The name is Lesnovo, near the town Philippines, just to 20 kilometers from our capital, Sofia. The Sufina family farm has been existing since the beginning of 2009. About 60, 70 different products are growing, each of which during the respective season of the year. The farm products include nuts, hazelnuts, different type of tomatoes, cucumbers, pumpkins, carrots, potatoes, and as well as the non-standard kale and chard, which are extremely unusual for our region. The land size of the farm is about 28 hectares, and the farmland is cultivated by a farmer and his family. And this farming activities involve also Bulgarian Food Safety Agency and Bulgarian Bioproducts Association. And this farmer is member of the Bulgarian Bioproducts Association. The proximity of the farm to Sofia allows the products to be picked up a few hours before delivery and to be as fresh as possible when they reach customer. No couriers are used, but the products are always delivered in person less 24 hours. The farm has online shop, and it's possible to order some products online by phone or by computer, and to receive these products during 24 hours. Result and learn lessons from this case study: how food from a farm ends up on our tables. The stages include plant production, processing, food production, distribution. Chain includes aspects from processing, distribution, consumer purchase, and consumer use. Result and discussion. Many studies emphasize that trust building is a major component and important advantage of this local food system and short food supply chain. Our case shows that building consumer-producer relationship is essential and provides not only a unique experience, but also prevents food loss. We also discuss the market in terms of community, building a place and improving relationship around food and neighborhood activities. Number of local food system and short food supply chains seek to build communities and relationship around food production and consumption. Creation of learning alliances and communities have been proposed as a way to reduce food loss according to World Bank. For example, in our case, the participants in the SHIM gain increased knowledge of food and agricultural system, increased knowledge related to their daily routine, as well as the food consumed can lead to some changes in the behavior. Other result, many researchers have discussed the reduction of food mile associated and food mile associated with local food system and short food supply chain as an environmental benefit. The concept of food miles, first created in 1992 by Tim Lang, is based on comparison between food products in terms of carbon emission and transporting the goods from the producer to the retailer or consumer. There are many concepts, but usually they are related with distance between producers and consumers. Thank you very much for attention. I will show a small short video about this Sufina farm. I prepared this
Stella,
video.
I think that we have
Yes.
covered the time and we will be in delay for the next session. I'm sorry.
Pardon? You may start.
How long it will take?
Sufina farm. Would you help me to?
So you already consumed the time. So what we can do now is that we probably include the link in the program or in the web.
No, this is not from internet. I have two files.
Yeah, but we have already covered the time.
Only to find on my.
But maybe we can do this later, and then I mean.
This is short. This is only 1 minute. And
1 minute.
very short video about there is
Okay. Stella, thank you very much. I think that we don't have time for questions, but if there are some, you could ask during the break. We would like to thank all the presenters that participated with the presentations in this session. I would like to invite members of Working Group 1 tomorrow for some discussions according to the program. And now we will leave the floor and to Slaven and Louis for the second working group number 2. Thank you very much.
Sorry, a technical announcement. Several people the afternoon didn't send yet the presentation to Lucrezia. So please, they need to be uploaded onto the system and try to send Lucrezia as early as possible. Thank you.
Yeah.
Yeah.
Good morning, everybody. We are starting with the presentation of Work Group 2, Agro-Food Loss and Waste. And our first speaker is Louis Palot from Institut Valencia de Investigaciones Agrarias. Louis?
Thank you. Thank you, Slaven. Good morning to everybody. I would like, first of all, to thank Gianfranco and all the organizers for inviting me and to hold this very interesting cost symposium. Okay. All right, we talk about antifungal edible coatings for fruits. This is a research line that we started at the IBIA in Valencia many years ago, about more than 10, with my colleague here, Maria Bernardita Perez. She's a chemist and I'm a post-harvest pathologist, so we join our research lines to trying to develop coatings, edible coatings for fruits, but with some antifungal ingredients. Here just to see where we are located in Valencia. It's a very important citrus growing area, so most of our work has been devoted to citrus fruit, but we have all work with coatings for other fresh fruits like stone fruits or persimmon or pomegranates and some others also. Here the outline, introduction, natural antifungal edible coatings in general. This will be just a generic talk about this topic, not to show any specific research, just a kind of theoretical talk a little bit and how we work in our case to develop the coatings and to test them. Oops. Okay, so we saw this as an alternative post-harvest antifungal treatment. We all know here, especially the people who were yesterday with the Stop Med Waste project, the need for alternatives to synthetic chemical fungicides for the post-harvest treatments of fruit, for fruit preservation. And these kind of alternative treatments, they can be physical, biological, or low toxicity or low risk chemical treatments. In this case, we can talk about natural compounds, food additives, or disease resistance inducers. And in our case, about antifungal edible coatings, in which in a coating, firstly devoted for physiological reasons to keep the post-harvest life of the fruit in terms of regulation of gases and water. We try to add some of these other natural compounds or antifungal ingredients in this formulation to make an emulsion that can be later applied to the fruit. So in general, we can, the edible coatings, the two different functions, the physiological and the antifungal by adding these ingredients. So it would be an increase of functionality. And then the first idea, especially in the case of citrus, would be to replace the conventional post-harvest waxes that are used by the industry right now, which are mostly formulated with chemical fungicides, especially with imazalil in their formulation. So natural antifungal edible coatings in general, we can talk about natural and synthetic. Natural can be coatings or film-forming substances which has an inherent antifungal activity, for example, chitosan, or antimicrobial, even more general, or for example, some species of aloe gels, or synthetic. We focus more on the development of synthetic composite films on coatings, in which an hydrocolloid and lipid matrix, we talk about composite coatings for this because we got two main ingredients in the matrix, an hydrocolloid and a lipid, and then we can add many other different kinds of ingredients, as for example, Werner explained yesterday for our results of the Stop Med Waste project. So on chitosan, we have here today, this morning, a talk on chitosan for meat and fish. So it's a very known natural biopolymer with inherent antimicrobial activity. So many works have been devoted to the use of this substance. And what's important from their point of view now is that has been approved as a basic substance for the post-harvest treatment of citrus fruit. So it's one of the few alternatives that can be in the market right now. Aloe is also, especially in countries like Mexico or others, that pulp, which is obtained from the leaf pulp, is the capacity to form film, and also it has some antimicrobial activity that has been tested alone or in combination with other antifungal ingredients for against many post-harvest diseases. So in our case, the type of coating matrices, we said about that, hydrocolloids could be basically polysaccharides or proteins, the lipids, usually we talk about natural waxes or monodiglycerides or fatty acids, and the composite coatings means that we use these two kind of ingredients as the matrix of the coating. And then minor ingredients that are needed for stability and functionality of the coatings, emulsifiers, plastifiers, and also the specific carriers of antifungal ingredients through impregnation, encapsulation of different compounds. What can be these antifungal agents? They can be of many types, food additives and grass salts, natural compounds, essential oils, plant extracts, antifungal proteins and peptides, even metal-based nanoparticles, or even living microorganisms like yeast or bacteria that have been previously tested as biocontrol agents against post-harvest diseases. And then we can explore the incorporation of these biocontrol agents into coatings as a new way to apply this kind of post-harvest treatment. So the antifungal ingredients the incorporation can be amended, imbibbed, impregnation of solid particles, or as we have seen today also, it can be micro or nano encapsulation, one layer or multi-layer coatings. So there are many different possibilities. What about the evaluation of these antifungal coatings? We are talking about the methodology that we are using in our lab in the IBIA in Valencia. First, we are preparing the emulsions. For that, we incorporate the antifungal ingredients at previously selected concentrations to the hydrocolloid lipid composite emulsion. For example, here you can see a typical emulsion of a coating that we have been working with. Hydrocolloid HPMC, hydropropyl methyl cellulose. This works as a lipid, glycerol as a plasticizer, oleic acid as an emulsifier, then a wetting agent also. And we will be talking about emulsions, about 6-10% of solid content. These samples has to be homogenized. Usually we use ultra-turrex for that. And then also cooled and agitation. So usually, I didn't have time to explain, but usually we evaluate the antifungal activity against our target post-harvest diseases previously, sometimes in vivo, sometimes in vivo, sometimes in vitro, sometimes in vivo. But once we have selected that antifungal ingredient, we have first to look at the compatibility with that specific matrix that we use. And then once we have a stable coating that we believe can be promising, then we start to looking at in vivo disease controllability of this coating. Usually we look for curative activity. We discussed a little bit yesterday about what was more convenient to look for curative or preventive activity. In our case, because most of the infections of oranges and mandarins that are causing post-harvest decay in the packing house are from infections that are recent infections, usually during the harvest time, because Penicillium digitatum and Italicum are strict wound pathogens. So in this case, we are interested mostly in curative activity. For example, in this picture, you can see work we did with cherry oranges some time ago with a student from Brazil. And also in that case, we're also looking for curative activity because most of these infections are from latent pathogens that infect the fruit in the field. So you need a treatment after harvest to stop those already established infections. So we select the fruit. We used to work with artificial inoculation to be sure that we got enough decay in the controls to be sure to say what is the effect of our treatment, because if the control is not decayed, then you can have many problems of why it's not decayed. So is for my treatment or not? No? So you need high levels of disease in the controls. So we use artificial inoculation also. Then we have selected of antifungal ingredients and we have our emulsion. And then after, it depends on, this is another factor interesting, the time between inoculation and coating, but usually when we can say about 1 or usually 24 hours could be, this depends on each pathosystem, no? We have to mimic all what is happening in the epidemiology of that specific post-harvest disease. So two within 24 hours after fungal inoculation. There are also several ways to apply the coating, can be by immersion, by spray, different times. And after that, we incubate the fruit at conditions which are good for fungal development. So, for example, between 14 days at 20-25 Celsius. And after that, you can look directly to disease incidence, disease severity, disease population, and then calculate the disease reduction with respect to your control. And of course, not only antifungal activity is important, we said that is important, the double functionality of the coating. So we are very interested in the quality of the fruit, the coated fruit after cold storage. For that, we have to look at the, of course, initial quality. And then in every case, the storage conditions will depend on the commercial storage conditions more adequate for each type of fruit. So once we have the coated fruit, for example, citrus fruit is common to do 30 or even late Valencia oranges can go until maybe 60 days at 5 Celsius. That would be the more commercial time. Of course, after some shelf life to simulate the time that the commercialization period after cold storage. And then, of course, on coated and uncoated fruit, we have to check at all the quality parameters, which mostly most of you, I'm sure, that are familiar with the quality evaluation, post-harvest quality evaluation of fresh fruit. Even sensory also. We have a panel of consumers also in the department, so we can check at all the quality. So thanks to Stop Med Waste Project, the Prima for financing these last years, most of the work we've done with edible coatings, antifungal edible coatings, and of course to this new cost action that we are initiating, and that I'm confident that this meeting here in Ancona will be a very nice start point for a great collaboration working on that. So just to, if somebody is interested in us working with edible coatings, we are editing a special issue on that in the journal Coatings. So I left some leaflets there. If somebody is interested, it can be published fast in this journal. So thank you very much for your attention.
Thank you, Luis. We have time for 2 questions.
You have mentioned the potential application of iron oxides in your emulsions. Have you already tested them?
We did very small work on that because this is a little bit more polemic for food because these metallic oxides, it can have issues about safety. But you will see in the literature, there are some groups, many groups in fact, working of using these oxides as ingredients of some theoretically edible coatings, but the legislation is always, let's say, regulating these kind of things. So maybe today you have a law or whatever, it's especially in Europe, saying that you should not go more than some concentration, but then 6 months later, new evidence show that you have to go down to this concentration. So people working with that has to be very aware of the regulations at every day.
Okay, thank you.
Yes, there is one more question.
Hi. For the inoculations, when you were doing the coatings, did you notice that because you were doing a wound inoculation, if the pathogen also will grow if you had a wax? Because your control was uncoated, inoculated. And I wondered if there's an effect just by covering the disease and having less oxygen into the wound that may be limiting the growth. So that's one point. And then the second, have you tested any cross-contamination, contact-based, disease spread in these oranges?
About the second part, no cross-contamination, because if you got an infection by penicillium in an orange, you will be sure that nothing else will grow there. So especially in other pathosystems that can be a problem, for example, with Geotrichum, the cause of sour rot, which is a very weak pathogen. And then from an initial infection of Geotrichum, then you have large possibilities that it will get a secondary contamination by penicillium that overcome the growth of Geotrichum. So that can be an issue, but not in the case of penicillium. So penicillium is a very strong pathogen on citrus, Digitatum italicum, I'm talking about, the cause of green and blue molds. So if you artificially inoculate that fruit, you will be 100% sure that nothing else will grow there. And in the first question, sometimes it depends on the objective of your specific research in each time. If you want to see your antifungal coating as a whole treatment, you can compare with uncoated fruit with artificial inoculation. There's no problem for that. But sometimes you're interested to see the effect of the matrix, for example, in the case. So then you just need to add another treatment in your set of treatments in which you will use the matrix without the antifungal ingredient. And then you can see, will my matrix alone have any effect without the antifungal treatment? So you can do that in the in vivo disease development assay at the same time if you want to. Okay, so we will proceed with the next presentation, which will be by Alessandra Di Francesco from Udine University here in Italy. So Alessandra, please.
Thank you, Luis. Thank you very much to everybody and good morning. My study is about the efficacy of wild Basidium pullulans volatile compounds and their potential application methods to contrast the strawberry fungal pathogens. The study was conducted together with Dr. Sinem Toludoglu from the Niğde University in Turkey. So everybody knows that the main post-harvest control strategy are now focused on the alternative approaches, trying to avoid the use of fungicide treatment with the aim to reduce the food waste and have healthy foods without any kind of problem of residues or toxicity of the products that are used during the post-harvest phase. So in this study, we decide to work with the epiphytic yeast derived from different sources that we called wild sources because they are coming from local apple genotype, Nidelmasi, rose hip, hawthorn, wild pear, that are located in different zones of Turkey. So 375 strains were isolated and selected previously against another important pathogen of post-harvest that is Alternaria alternata. So as the population was so intensive, we decided to work just with 32 strains that show in the previous essay mycelial inhibition of the pathogen of 40% or exceeding the 40%. So the aim of this study was to use wild sources because we hypothesized that these wild sources can be correlated to new metabolic fluxes of biocontrol agents. So this evaluation of the strain diversity can provide a chance to increase our knowledge on the metabolic function of this microorganism. So the aim of the study were to explore the volatile compounds antagonistic potential of the yeast strain that were isolated from wild fruits against Botrytis cinerea and Colletotricum acutatum. The second aim to characterize these volatile compounds and evaluate the effectiveness of two different methods of application of volatile compounds. So this by the first molecular characterization of these preselected strains by using the genes ITS and elongation factor. The second one to test the effect of yeast volatile compounds by using these two methods by in vitro assays and using the solid phase microextraction to characterize the volatile compounds. Finally, the box application by the hydrogel spheres on strawberry. So as you can see, all the strains that were analyzed molecularly by showing the ITS gene, you can see no particular differences between the strains. So this phylogenetic altruism is not so important to consider, because just using the elongation factor gene, you can see different clustering of the population, where help us to consider the best strain that we analyzed previously that derived from different clusters and were respectively isolated from the 4 different sources. So local Turkish apple, hawthorn, rosehip, and wild pear. So these strains were used for our in vitro assays. The first, the classical one, was by using the double petri dish assay, where NIDA plates were inoculated with 100 microliter of each strain suspension, and then incubated for 2 days at 25 degrees. Then the plate was joined to another plate that had the PDA inoculated in the middle with a mycelial plug of both pathogens. So the plates were joined with parafilm and incubated for 5 and 7 days, respectively, for botrytis and choledotrichum. The second assay, the most original, was to put inside of an EDB medium, inoculated also with the yeast suspension, the sterilized spheres, hydrogel spheres. So as previously, these spheres were inoculated with the strains, incubated 2 days at 25 degrees. Then washed and prepared the double petri dish assay as before I explained for the NIDA medium. So as you can see down the results of these experiments, you can see that the hydrogel volatile compounds produced by the strains tested show a higher mycelial inhibition if they grow on the hydrogel spheres. And so, as you can see, we selected the 2 strains, C10 and M13, respectively for Botrytis cinerea and Choledotrichum acutatum, as a best strain to inhibit the growth of these 2 pathogens. So, as you can see in this slide, you can see the differences of the mycelial growth of Choledotrichum acutatum and the activity displayed in particular by the strain M13 that we selected to contrast the activity of this pathogen by the emission of volatile compounds on NIDA plates and by hydrogel spheres. So, we characterize these volatile compounds. So the preparation of the samples were inoculating NIDA medium with 100 microliter of each biocontrol agents, and then after 48 hours at 25 degrees, the plates were joined with an empty plate. The same was made with the hydrosphere gel that on EDB medium and grow for 2 days at 25 degrees, then were washed and joined with an empty plate. By using an SPMA fiber, we make a little hole between the 2 plates to exposing the fiber for 30 minutes. And after that, we extract the volatile compounds by using the gas chromatographic mass. So as you can see from the heat map, you can see that the main volatile compounds and the most intensive concentration were produced by the strain that were inoculated on hydrosphere gel. There was a big differences, as you can see, if we compare the strain M13 grown on hydrosphere gel and with the NIDA plates in the case of M13. And as you can see, also the main compounds that Arobasidium pullulans strains produce belonging to the alcohols class. So, deciding that the hydrosphere gel were the most active method to produce volatile compounds, Fragaria Ananassa cultivar Candonga from organic production were sprayed each one by 5 ml of Choledotrichum acutatum and Botrytis cinerea and put inside of sterile boxing containing the inoculated spheres and a sterile krill where the strawberry was put on. So the strawberry were put at 20 degrees for 5 and 7 days, respectively, for Botrytis and Colletotricum macutatum. And as you can see from the graphs in the slide, we detected a reduction of 46% of the incidence of the disease in the case of Botrytis cinerea for the strain C10 and for the strain M13 against Colletotricum macutatum by 84%. So we can conclude that if we think about the volatile compounds application methods, probably the polymeric spheres have a higher water binding capacity that can increase the surface where the strain can grow. So as displayed in 2017, Parafati et al., this sphere were and show a high stability over time of these microorganism cells viability. What is the novelty of this research? That this hydrosphere gel can improve, in our case, the antagonistic activity of our Arobasidium pullulans by the production of active volatile compounds, because we see a higher number of volatile compound produced with a higher concentration. So the main question to say is if can hydrogel spheres could be considered as a new tool for the application of volatile compounds during the post-harvest phase. Maybe try to find some answer to this question could be very difficult. However, if you think about the volatile compounds produced by the biocontrol agents, what we see that the main compounds were alcohols that were more active against Colletotricum macutatum with respect to Botrytis cinerea, and as we can see in our past studies with Arobasidium pullulans, this strain that belong from wild environments and wild sources seem to produce a less complex volatilome with respect to domesticated yeast. And so, however, this condition do not compromise the activity and the efficacy of these strains. So I'm finished, and thank you very much. And thank you very much to my group that is also online following us. Thank you.
Thank you very much, Alessandra. Is there any questions from the audience? Wait. Micro, because there is the online.
So can for grapes use the sodium metabisulfite sheets? The question can you use in similar way as you have like for grapes in your case? Because the problem with the material you have, it has water inside. Or I don't know.
Yeah. You mean in the field or during the post-harvest phase?
Post-harvest is to send in a box to the supermarket.
Yeah, why not? Could be the good strategy for this kind of application. The problem is that the real cost of these spheres and the sustainability of these spheres. The great things that I saw during our experiment said you can easily sterilize and put in autoclave these spheres, so you can reuse.
But whenever you send it to the market, it's not sterile anymore. Everything is, the question is the treatment is good enough to go to a second stage from the lab.
Yeah, I hope I can answer you if yes or no. I hope so. Probably this could be the next step that someone that practically work in the post-harvest phase could make some experiments about this. Why not? Thank you.
So
there are no other questions. We have time for another question.
Well, can have one. Do you see any potential to use the gel, for example, not as a sphere, but as an ingredient of a coating, for example, a gel that can be? Do you think it can be mixed with other?
With this kind of spheres, no, because inside there is acrylamide. but probably from Rhobasidium pullulans, we can try to find new interesting substances to use as edible coating. Yeah.
Thank you very much.
Thank you.
The next speaker is Giulia Pemolit from University of Torino, Italy.
Okay, good morning. My presentation is about the efficacy of antagonistic yeast, control brown rot of nectarines, and their effect on the fruit microbiome. So brown rot is one of the main diseases affecting peaches and nectarines worldwide, and it causes important production losses, especially in post-harvest. The causal agents are several species belonging to the genus Monilinia. In particular, Monilinia fructicola is the main species present in Italy. Disease management is currently carried out using preventive treatment with synthetic fungicide, but this is linked with several issues, such as the presence of the residues on the fruit, the environmental pollution, and then the development of fungicide-resistant strain of the pathogen. Moreover, as we said this morning, the Farm to Fork strategy adopted by the European Union is calling for a reduction of the 50% of the use of synthetic pesticides by 2030, and now it was then postponed to 2035, but there is a need to find alternative solutions to manage this type of diseases. A solution can be the biological control, in particular the application of yeast as biological control agent, because they have many characteristics that make them suitable to be tested as biological control agents, because they are adapted to the fruit microenvironment, they are tolerant to different conditions, such as low temperature and low oxygen that are typical of the storage room. They don't produce any type of mycotoxin or allergen, and they have simple nutritional requirements that make them able to colonize the fruit surface for a long time. So the aim of our work were first to evaluate the effectiveness of the treatment with antagonistic yeast on nectarine to control brown rot caused by Monilinia fructicola, and then to evaluate the effect of the treatment both on the fruit quality and on the fruit microbiome. So this was the experimental setup that we followed. First, we did some screening tests in vivo and in vitro to select the best performing microorganisms that were tested then in an efficacy test in vivo. Then we performed some quality analysis. We identified the most effective yeast that we applied in the efficacy test. And last, we performed the microbiome analysis. So the first was the screening test, in which we aimed to evaluate the efficacy of the treatment with 14 yeast strains to control brown rot. The yeast that we used were isolated from the carposphere of different fruits, and some of them were endophytes. So we set up the treatments and we added 3 control, a chemical control treated with fludeoxolil, an inoculated control, and an healthy control. Then we stored the fruit in the storage room, and after a period of storage and shelf life, we measured the diameter of rot. And we found that the average diameter of rot developed for all the tested yeast was significantly lower compared to that reported for the inoculated control. So we decided to select 3 strains for the further analysis. These 3 strains were then further tested in vitro to confirm their antagonistic activity. So dual culture assays were performed, and after 7 days at 25 degrees, we had a mycelial inhibition ranging from 39 to 48.5%. So the antagonistic activity of this strain was also confirmed in vitro. Then we performed an efficacy test in vivo in semi-commercial conditions, so using a higher number of fruits. Also in this case, as in the screening test, we added 3 different controls. We inoculated the fruit by dipping them in a conidial suspension of the pathogen, Monilinia fructicola. We performed the treatment with the biocontrol agents by dipping the fruit in the yeast cell suspension. And then we stored the fruit at 1 degree for 1 month in the storage room, and then we exposed them in shelf life for 4 days. So after 28 days of storage, we recorded the rot incidence of the fluids, and we found that rot incidence for all our treatments was significantly lower than for that reported for the inoculated control. And in particular, we had a strain, the MS, that had a rot incidence comparable to the chemical control, to the fluodeoxonil. Then the fluids were exposed in shelf life, and again we recorded the rot incidence, and we found that rot incidence for all treatment was still significantly lower compared to that of the inoculated control, but no one of our treatment was still comparable to fluodeoxonil in shelf life. Then we performed some quality analysis by measuring firmness, total soluble solids, and titratable acidity for all the treatment and the control. And we didn't find any statistically significant difference both at the end of the storage and at the end of shelf life. And so we concluded that all the tested treatment did not significantly affect the fluid quality. Then we would like to know who were our strains. So we performed a molecular identification of the 3 strains that we used in the efficacy test. And we found to have 3 different species, Amechnicovia fructicola, Anaerobasidium pullulans, and then Amechnicovia pulcherrima. Last, we performed the microbiome analysis to evaluate the effect of the treatment on the fluid fungal microbiome. So the microbiome was sampled during the efficacy test at 3 time points, at harvest, so before starting the trial, at the end of the storage, and at the end of shelf life, for all the treatments and all the control. Both the epiphytic and the endophytic microbiome were sampled. DNA was extracted from this microbiome by using optimized protocol, and then the ETS2 region was sequenced and the metabarcoding analysis was performed. So we were interested in different parameters. The first one was the alpha diversity, so the diversity within each sample. And by regarding the treatment, we found that both for the epiphytes and for the endophytes, the treatment with the strain of Mechnicovia had a lower richness compared to the control, in particular with the chemical control. Also, by regarding the sampling time, we found that the treatment with Mechnicovia had a lower richness compared to the richness of the fluid at harvest. The second parameter was the beta diversity, so the diversity among our different samples. We performed a multivariate statistical analysis that shows that all the 3 factors that we considered, so treatment, tissue, and sampling time, had a statistically significant effect on the microbiome composition. In particular, the treatment was the factor that had the highest impact on the total variance, followed by the tissue, and then by the sampling time. Last, we did a compositional analysis. This is the graph for the epiphytes, and we can see a development of the biocontrol agents, both at the end of the storage and at the end of shelf life. In particular, a significant development of Mechnicovia in the 2 theses treated with our strain of Mechnicovia. We can also see a development of monilinia, in particular in the inoculated control. So monilinia was inoculated, and this is a confirm of inoculation. And then the abundance of some other fungal genera was found to be modified by the treatment. In particular, we can notice a significant presence of Geotrichum in all the control at the end of shelf life. In particular, but Geotrichum is not present in our treatment, and also the presence of Penicillium in the healthy and chemical control. The situation for the endophytes is not so different. We can see also in this case a good development of Mechnicovia, in particular in the treatment with MS and AP47 strains that are the 2 Mechnicovia that we used. Also in this case, the development of monilinia and Penicillium. And then for the other genera, the presence of Geotrichum that is present in this case also at harvest, but a significant presence was recorded in the control at the end of shelf life, and then the presence of Cladosporium in the endophytes in almost all the theses. So to conclude, in our work, we found that treatment with some antagonistic yeast were effective in reducing brown rot caused by Monilinia fructicola on nectarine in storage. And in particular, we had a strain, the Mechichovia pulcherrima strain, that had an efficacy comparable to the chemical treatment at the end of the storage. By regarding the fruit quality, we didn't find any statistically significant effect on fruit quality parameters such as firmness, total soluble solid, and titratable acidity. And then the microbiome analysis led us to see that the yeast that we use for the treatment are able to occupy very, very well the ecological niche of other fungal genera, in particular of Monilinia that was inoculated, and also of other pathogenic genera such as Penicillium and Geotrichum that we found on the microbiome of the control, but not in the microbiome of our treatment. So to conclude, treatment with antagonistic yeast could represent a promising tool for reducing post-harvest lossing while maintaining at the same time the fruit quality. And a perspective of this work can be to find a strategy to apply this yeast in the field, so in pre-harvest, to reduce then the post-harvest decay caused by Monilinia. And thank you for your attention.
Thank you, Julia. We have time for some questions. George.
Thank you very much for your very nice presentation. I would like to ask you two things. The first, you mentioned that it would be nice to test this product in the field, applied that under pre-harvest conditions. And I would like to ask you if you have any data on that, if you have already attempted to evaluate the efficacy in the field, in the orchard. And the second question is something about the microbiome analysis. I would like to ask you whether the microbiome analysis was conducted on symptomatic or asymptomatic fruit. Okay, in the first assessment, just after harvest, all the fruit, I suppose, that were asymptomatic, but on the treated fruit that were stored for 1 month in the fridge or exposed to shelf life, did you analyze the microbiome on symptomatic fruit or not?
Okay, so for the first question, it will be a nice perspective of this work, but we didn't do until now any trial in the field. It's like we would like to test, of course, to see if our antagonistic yeast has the potential to control Monilinia also during a field application, and then to record the disease incidence in post-harvest. For the microbiome analysis, yes, we performed first sampling at harvest, so before starting the trial, and then we performed the microbiome. We had some boxes with fruit only for the microbiome part. So after storage and then after shelf life, we performed the sampling for all the fruits, infected and not infected, of the box.
Okay, thank you, Julia.
All right, next speaker is Selda Dailer from Yozgat Bozok University from Turkey.
Hello, everyone. Thank you for being here. My name is Selda, and I am a viticulturist at Yozgat Bozok University in Turkey. Today, I'd like to talk to you about the potential of smoke solutions from vineyard pruning waste to mitigate heavy metal toxicity in grapevine saplings. Grapevines are really important for the economy. They give us lots of useful products like grapes, must, and even things from their leaves and waste branches. The International Organization of Wine and Wine states that approximately 79 million tons of fresh grapes were obtained globally from around 7 million hectares of vineyards in 2022. Of the total volume available, 46% is allocated for pressing. 40% consists of table grape production and 7% of grape production for drying. The remaining 7% is losses. To get good product from vineyards regularly and sustainably, how you manage them matters. Things like pruning and training the grapevines are crucial for successful cultivation. Viticulture activities create a lot of waste and cause environmental problems. Authorities are now paying more attention to the problem of waste and its impact on environment. Leftover pruning waste in vineyards is a big issue for tillage and controlling pests and diseases. It's important to find eco-friendly solutions to cut down on waste, like recycling and reusing resurges. Globally, vineyard pruning waste amount to about 2.5 tons per hectare each year. In Turkey, vineyards produce about 2.3 million tons of pruning waste each year on average. Lately, there has been a growing interest in finding new uses for leftover materials from viticulture. This is driven by two main goals, reducing environmental impact and meeting the rising demand for sustainable materials, nutrients, and bioactive compounds for food and feed industries. Recycling plant waste back to where it came from helps nutrient cycling, increases productivity, and supports plant growth. Cadmium is a highly dangerous heavy metal that builds up in soil and water, leading to severe toxic effects on various living organisms. Factories and industries, excessive fertilizers and pesticides, wastewater and sludge lead to the contamination of soil and water with cadmium. The critical cadmium value for soils is 38 ppm. Above this limit, cadmium slows plant growth, weakens root, and reduces both yield and quality. Plant-derived smoke solutions, seen as a valuable biostimulant, are gaining attention as an eco-friendly option for agriculture. Plant-based smoke solutions contains various active compounds that can stimulate different plant species in a specific environment. The most important active components among these are kerikins and cyanohydrins. These compounds promote plant growth and development and provide protection against environmental stress. Functions of smoke solutions are known in various plants, but their impact on grapevine under heavy metal stress has not yet been explored. The study aims to assess how different concentrations of smoke solutions derived from vineyard pruning waste affect grapevine saplings exposed to cadmium stress. Saplings of narince in Turkish grape variety grafted on 1103 Paulson American grapevine rootstock were used as a plant material in the study. Rootstock and scion cuttings were taken during the resting period. Respectively, grafting, paraffinization, and acclimatization were applied. They were then transferred to the greenhouse. Vineyard pruning wastes were initially dried in an incubator and then separated into replicates. The apparatus used in the preparation of plant-derived smoke solution was this. A mixture of equal volumes of perlite and peat was used as a growing medium. Irrigation and fertilization were done regularly. The smoke solution were sprayed on the leaves while cadmium chloride was applied to the root zone. The experiment consisted of 5 treatments. Was completed after 3 months of growth from May to July. Morphological, physiological, and biochemical analyses followed by statistical assessment were conducted to understand the changes caused by smoke solution treatments in grapevine saplings under cadmium stress. Cadmium toxicity hindered plant growth, impacting shoot and root length and fresh and dry weight. It also decreased chlorophyll content, stomatal conductance, and leaf relative water content, while significantly increased membrane damage and lipid peroxidation. Under cadmium stress, smoke solution applications effectively increased plant growth, chlorophyll content, stomatal conductance, leaf relative water content, total phenolic content, and antioxidant enzyme activities, while significantly reduced membrane damage and lipid peroxidation. Especially the 0.5% smoke solution effectively elevated cadmium stress damage compared to other concentrations. In summary, this study found that plant-derived smoke solutions mitigate cadmium stress in grapevines, enhancing antioxidant defenses. The findings also support the idea that smoke solutions can serve as a cost-effective, quick, and eco-friendly biofertilizer, promoting vegetative growth for sustainable agriculture. Thank you for your attention.
Thank you very much, Zelda, for your presentation on this highly creative technology. So, are there any questions for Zelda? Seems it's so highly creative that nobody wants to talk about. Come on. Yeah.
Okay, thank you for your presentation. You've shown in the first or second slide that you have 2.5 million tons of grapevines. Grapevine. Did you consider using that biomass for producing pellets?
Üretim şeyinin dışında olan diyeceğiz. Söyle. Except the production.
You have many leftovers from.
Only leftover.
Did you consider or did you make some for using that biomass for production of pellets and for using that pellets for heating or something else? Or did you just consider it your research?
Maybe.
Okay.
Thank you.
Thank you so much. I would like to ask about what is the practical use of this kind of treatments? Can we use in the practical in the big grapevines with these treatments? What is your idea for in the vineyards? Can we use it in the practical uses?
Yes, yes.
Okay, thank you.
Thank you. Thank you.
Thanks.
So we are coming to the last presentation of this session. This will be from Kata Ludman-Michal from Hungarian Interprofessional Organization for Fruit and Vegetable. Kata, the floor is yours.
Thank you, Simon. Okay. Hello, everyone. I hope you are not so hungry. It's always a good thing to give a presentation before lunch. The title of my presentation, cold storage may elongate the fresh consumption period of sour cherries. Sour cherries play an important role in fruit production in Hungary. It has the second largest production volume in our country. It's mainly used by the food industry because this fruit has a really sour taste. But we have several Hungarian cultivars which excellent for fresh consumption because they have a harmonic sugar-acid ratio. Moreover, they are really healthy because they are rich in bioactive components too. We have only one problem with this fruit, the short shelf life. During storage, there are some physical changes like softness or color change, but the main problem, the decay which caused by fungal infection. Actually, the Hungarian sour cherry sector doesn't have any adequate post-harvest technology. For this reason, a really small proportion of sour cherry are marketed fresh. And during the sale, a huge percentage loss produced, thus increase the food loss and waste. So the aim of our work was to investigate the shelf life and the storage potential of Hungarian sour cherry cultivars. The experiment location was at Újfehértó, and we started the work from 2016 until 2019. And we worked the three predominant Hungarian cultivars, Érdi Bőtermű, Újfehértó Fürtös, and Petri. The same plant protection strategy was applied for all cultivars, and the samples were harvested with stem randomly from the whole tree, and we use only intact healthy sour cherries for the experiment. With this flow chart, I would like to show the main steps of our work. So after the harvest and the quality control, we made two groups of samples. With the first group, we measured the fruit firmness, and after carried out the shelf life test, which was 2 weeks long, and after calculate the disease severity index. The other part of the sample were stored for cold storage for 6 weeks, and we used two different storage methods, normal atmosphere and modified atmosphere packing. When we finished the cold storage, after we measured again the fruit firmness, did again the shelf life test, here it was only 7 days, and calculate the disease severity index, the weight loss, and the healthy fruits ratio. During the shelf life test, every second day we checking the quality of the sample and calculate the ratio of the healthy fruits and the disease severity index according to Mackin index. For this, we use an empirical 6-point scale. The cold storage was a 6-week period, and the temperature was around 0 degrees, 0 and between 2 degrees. And we used two different methods, normal atmosphere and modified atmosphere packing with using extant cherry plastic bags. When the cold storage finished, after we measured the samples and calculate the weight loss and the ratio of the healthy fruits. In the case of fruit firmness measurement, we use an Agrostat device, and the results were given in Durofer index, where the higher value indicated the higher fruit firmness, and we use 100 healthy fruits for each sample. For the data, those statistical analyses we used. And now I would like to show the main results of our work. This diagram shows the proportion of healthy fruits of the free sour cherry after harvest in 4 consecutive years. We saw that the cherries had different shelf life. The highest proportion of healthy fruits were determined in the case of Újfehértó Fürtös. It means that at the final day, 64% fruits were healthy. VIO, we saw that in the proportion of healthy fruits was significantly lower in the case of Érdi Bőtermű. The disease severity index was determined on the 6th day of the shelf life test. After harvest, there was significant differences between the cultivars. Erdibőtermű had the highest value. Following the cold storage, in any cases, the disease severity index increased significantly. And when we compare the disease severity of the cultivars following the cold storage, we saw that Erdibőtermű had the best or the smallest disease severity with MAP storage. The fruit firmness measurement was determined after harvest, and there was significant differences between the cultivars. Erdibőtermű was the softest fruits. Following the cold storage, the MAP significantly increased the fruit firmness for all cultivars. After we compare the two different storage methods, and in the case of modified atmosphere packing, significantly higher ratio of healthy fruits and lower weight loss were determined independently of the cultivar. When we analyzed separately the cultivar, we observed that significantly less healthy fruits remained in the case of Erdibőtermű, and there was no significant differences between the cultivar in the case of weight loss. Finally, we did again the shelf life test after the cold storage, and in every case, so in every cultivar, the shelf life was reduced significantly. The highest proportion of intact fruit was determined in the case of Erdibőtermű. With MAP storage, 51% fruits were healthy. In the case if the other two cultivars, the proportion of healthy fruits was under 20% in all cases. So, summarize the results. We can say that Újféltő Fürtös had the best shelf life after harvest. The applied MAP storage, extend cherry plastic bag, reduced the weight loss and the percentage of the decayed fruit, and preserved or increased the fruit firmness of Újféltő Fürtös and Petri cherries after cold storage. Following the 6 weeks cold storage, Erdibőtermű had significantly lower proportion of healthy fruits, and the shelf life of the studied sour cherries was shorter. The best storage shelf life values were detected in the case of MAP stored Erdibőtermű cultivar. Thank you for your attention.
So the floor is open for questions.
Thank you for your presentation. Just one question about the MAP application. Did you monitor the gas emission in your field? And if yes, what about the fermentative metabolites that usually produce it after long storage?
Thank you. Yes, thank you. In this research work, we didn't measure the gas inside the MAP. But now we started to measure because now we have a device for it. And yes, thank you.
Other questions? Yes.
Did you find chilling injury symptoms such as pitting in these cultivars, or you only measure firmness but you didn't see? I mean, cherries are susceptible to chilling injury during long-term cold storage. Did you find any symptoms or not?
This kind of symptoms I didn't see.
There are other questions? We have time for one more, maybe. No questions. So, Ata, for your very interesting.
Thank you.
So this was the last presentation of this session and from this work group. I thank all the presenters, and I thank also all you here, and I invite you also to join us tomorrow, the meeting of Group 2, who is interested in food loss and waste management.
And a big applause to the speakers and to the moderators of the session. And if you want, all together, come down and we can do a long line in front of the table to have a joint picture all together.
Ready to start? Okay, we have one more person just coming in. And I want to just say that I've organized a lot of conferences, but I've never thought of this brilliant idea of serving some bubbly and do a wine tasting before the second afternoon session. So you'll make sure everyone's in good spirit. So thank you very much for that. So let's please start with the first session. And we're very privileged to have with us Rosa Rollet from the FAO. And she's from the Senior Enterprise Development in the FAO and a leading figure in post-harvest science and technology, who's funded and supported a lot of post-harvest training and initiatives all over the world. So thank you, Rosa, for coming. We're very happy to have you here. Over to you.
Thank you very much, Lisa. It's a pleasure to be here. I also want to also say a big thank you to Gianfranco for inviting me to participate here today. My presentation is a bit of a departure from the science because it's all about development and in the context of food loss and waste. FAO, as you may know, is an international development agency and with a mandate to address to lead international efforts to defeat hunger, food insecurity, and malnutrition. And FAO is also mandated to raise levels of nutrition and reduce food loss and waste within the context of sustainable production and consumption practices. FAO's vision is a world free from hunger and malnutrition, where food and agriculture contribute to improving living standards of all, especially the poorest, in an economically, socially, and environmentally sustainable manner. And the organization is responsible for contributing to achieving all of the 17 Sustainable Development Goals, but in particular has a responsibility as a custodian agency for 21 SDG indicators. In this context, if you look at the bottom, you see SDG Goal 12 on sustainable, responsible consumption and production. And this is where food loss and waste is addressed. And FAO is responsible for the SDG 12.3 indicator, and I will come to that in the course of my presentation. As you may know, be aware, the goal 12, target 3, SDG target 12.3 is where the food loss and waste is enshrined in goal 12, target 3. And of course, within the context of that goal, there's a specific target for food waste reduction to reduce food waste by half. There is also specific mention of reducing food losses, but no specific indication of how much. Of course, this is a very tall order because reducing food losses is not a very easy task, but we know it's doable. And also in the context of reducing food losses, also including post-harvest losses. Now, how do we define food losses? Well, in 2019, as FAO set about going into establishing a food loss index, it was necessary to identify and define
define boundaries within the supply chain as to how and where we would be looking at measuring food losses. So in terms of a definition, food loss is defined as a decrease in the quantity or quality of food resulting from decisions and actions by food suppliers in the chain. And that does not include retail, food service providers, and consumers. This is a major domain that FAO addresses. On the other side, food waste is a decrease in the quantity or quality of food resulting from decisions and actions by retailers, food services, and consumers. And this area of food waste is addressed by UN Environment, who deals with specifically with food waste and all of the environmental issues associated with it. But and so the SDG includes two different indicators, and I'll come to that more specifically in my presentation. According to FAO's estimates, based on the boundaries and that we I've just highlighted, an estimated 13.2% of food valued at about $400 billion is lost on an annual basis between harvest and the retail market. And according to UN Environment's data, and that comes from 2020, I know they will come up with more recent estimates shortly. 17% of food produced is wasted in households, food services, and in retail. A majority of that food, however, is wasted in the households of consumers. And then food loss and waste also contribute to the generation of greenhouse gases, accounting for 8 to 10% of global greenhouse gas emissions. Now, food loss and waste has a number of implications for a number of different, let's say, development targets. And this is why it is so important that we look at it from a number of different angles in terms of the impacts that we can have if we begin to really work hard in terms of reducing food losses and waste. We see that from a food security context today, across the globe, we have somewhere around up to estimated 735 million people in 2022 were undernourished. 3.1 billion people lack access to healthy diets. Food loss and waste also, if we produce that food, it accounts for somewhere for large quantities of water. It has a high water footprint. It has a high land footprint. And if you look at the graph on the slide here, if food loss and waste were a country, it would be the third highest emitter of greenhouse gases globally. Excuse me. And we talked also about the costs. So these are some of the really important aspects and issues that we can impact if we do well in terms of how the approaches that we apply to reducing food loss and waste, we can make a difference in terms and generate sustainable impacts, economic, social, and environmental beneficial impacts. Now, in 2021, the Food System Summit, at the Food System Summit, 131 countries presented the impact pathways, and FAO analyzed the results of these, the impact pathways, did a very sophisticated use of the data lab to actually analyze what is happening in the context of the country priorities. And there were a number of 71% of the countries who presented their impact pathways prioritize food loss and waste as an urgent issue to be addressed. And within that context, analysis shows that the highest priority accorded in terms of the actions that are required are, number one, awareness raising,
Secondly, to address the issue of waste management, recycling, upcycling, and use as feed. The third priority was to look at the whole food supply chain towards education and capacity development, fundamentally important for any change we want to see, as well as data. Now, in terms of the context of FAO's actual core work on food loss and waste, it is a priority, a program priority area of the work that is done in the work that we do in development context with countries. And so much of what is being done in that context maps very well to the core functions of FAO, which include, and I'll just go very briefly through these because I'll then provide specific examples of what is being done in that context. So a lot of work to address the issue of data and information. Data is vitally important in terms of helping countries to actually develop policies, but also in terms of tracking what is happening, in terms of reducing the food, in terms of the food loss index. Then we work also on standard setting, normative and standard setting instruments, facilitating dialogue at different levels, the global country levels on different specific issues that relate to food loss and waste and to a number of other developmental issues, supporting institutions to prepare, implement, monitor, and evaluate policies, programs, and to leverage instruments. Also facilitating partnerships and coalitions for more efficient, inclusive, resilient, and sustainable food systems, and to advise and support activities that assemble, disseminate, and improve the uptake of knowledge, technologies, and good practice, as well as advocacy and communication for reducing food waste at the global, national, and regional levels. Okay, so in terms of measurement, FAO, as I mentioned, is responsible for the food loss index, and this is SDG 12.3.1A is categorized as 3.1A. And these, the estimates I mentioned of 13.2%, this is where this originates. Currently, the food balance sheets of countries are utilized to actually make those estimates. And this is very useful because what this means is that because it's a standardized procedure, you can actually use that information and compare across different countries and across different regions. And many countries now have started looking at, and FAO is also looking at what is happening in the context of field level losses as well, because what we're seeing also is that you have, particularly now as you see increasing temperatures and weather variations, now the losses at the field level now are increasing significantly. So these are some of the things that are now happening. So we may see some changes in the future as we have more information pertinent to in the food loss index, as we do more work with the harvest area of work, which is now being done. Because what we have to do is to compare what is happening on the field with those food balance sheet estimates. And then UN Environment is working on the food waste index. However, in the future, those two will be combined. So that when I talked about the 13% that is lost, we can look at it from in terms of the types of crops. And here you see that the majority of the losses take place in the most perishable items, fruits and vegetables, as well as in meat and animal products. And on the right-hand side, my right hand, maybe left, I'm not sure, you will see the levels of losses estimated for different regions of the globe. Okay, in terms of the normative and standard setting instruments, FAO has produced a number of these types of instruments. In particular, what would be of interest here is a code of conduct, a voluntary code of conduct that was endorsed by one of the highest level governing bodies at FAO. It was actually requested by the countries, and now many countries are actually using that code of code, the standardized version of this document, and they are adapting it so that it can be utilized within their context to develop specific actions and to also to begin to design and implement policies, and also to inform some of the programs that they are going to be working on to address the issue of food loss and waste. There's also one of the documents, the first, I think you can't see very well, that talks about the food loss index. And another one which is very pertinent is the EXACT tool, which is a tool that is used, an online tool that will help to analyze what is happening in terms of greenhouse gas emissions. All of these, later on, I'll show you where you can actually access the specific materials that I've been talking about. Then in terms of helping countries to develop evidence-based policies and strategies, the way in which we work is working with countries at the field level. The areas of work, of course, we prioritize first of all and foremost the prevention of food losses, and then reduction, rescue, and redistribution, followed by recycling and upcycling. Upcycling, of course, is priority over recycling, because upcycling can move that what would normally be likely to go into waste. You move it up back into, for the most part, food options. These are what we intend to do and try to do, is to make sure that we can maximize the amount of food that is produced and eaten and can be consumed in light of the issues of food security and nutrition. So whatever we can do in the first two rows, this is really the highest priority that we will continue to try to do, and encourage the application of the circular economy to keep that food in the supply chain and for use as food in food consumption. This is just an example of, the priorities. Priority 1, prevent and reduce. 2nd, recover, reuse, and then transform. Of course, upcycling would be our first priority, to come up with food products, and then look at other options. This is just a very quick view of some of the work that we do to help the countries to build an evidence base. This is where we recently completed a project in 3 low-income food deficit countries in Africa, where we look at across the entire chain and look at where the problems lie, work with the different stakeholders to come up with solutions that are adoptable by them and in a sustainable manner. And in this context, we were working with grains. We'll introduce 2 different storage options for the grains, and also to improve the harvesting, because there's often that problem of the late harvest that results in a lot of infestations and pests, and you have problems at the harvest phase. We improved on the drying facilities, because a lot of the drying takes place on the ground. And so with all the different improvements at all various stages of the supply chain and the introduction of improved storage, the PICS bags, which are hermetics, they're still very, very good, very efficient, but we still have to consider in the context of sustainability that you produce a lot of trash at the end of the day when those bags, and they can be costly, very effective, but these are some of the considerations we have. And so the grain silos are some of those that we would encourage and promote, because at a household level, they make a lot of sense. The ones of the PICS bags, they have the benefits in that they can be easily moved, transported. So, you have to look at some of the trade-offs, what makes sense, where, and what to use and adopt. And then here's another example where we've done work on the policy recommendations for governments. This was a project that I worked on, and we also implemented in South Asian countries where you have a lot of losses, high levels of losses of fresh produce in the supply chain, mainly due to packaging. So again, these are some of what we did was to introduce improvements that generate sustainable benefit, because in so doing, we reduced the amount of plastics in the environment, we improved the quality, we reduced the losses. In so doing, the consumer benefits because they get a better quality crop, better quality, they throw away less. And so there are a number of different benefits that are derived by just making those simple adjustments that the countries were willing to take up. And you can see some of the impacts. For tomatoes, we consistently found that we could reduce the losses by anywhere between 80 and 100%. All of the information that I have shared with you, the resources that are available are now consolidated in a technical platform on the measurement of food loss and waste. It's an online platform that includes a community of practice, and it is designed to provide opportunities to access information, not only information, but also a number of training programs, provides an opportunity for collective learning, also for awareness raising as well, and I'll come to that very quickly, and facilitating networking. I talked about partnerships. This is one example where you bring together a number of different like-minded agencies and partners working to support the operations of the platform. And also in the context of leveraging what is all the partnering and working together to form coalitions. There was this coalition, the Food is Never Waste coalition, that was established at the Food System Summit in 2021. It continues to work in an informal manner, but working together. There's a specific page on the platform that addresses this. And finally, in terms of awareness raising, which is a number one priority for countries across the globe, in 2019, the UN General Assembly approved the observance of an International Day of Awareness of Food Loss and Waste, which is now has been observed since 2020 on the 29th of September every year. So there's a lot that happens around that. There's a specific page on the technical platform that gives a lot of information, documents on what is happening, the theme for that day, and how you can organize events, how to participate in observing the day. So this concludes my presentation. I thank you for your attention.
Thank you very much, Rosa. That was really, as always, a wonderful presentation, a lot of information. I know a lot of us will go back and go and unpack the information that you've shared with us. We are a bit tight on time, but I will allow one quick question. If there's a burning question. None. And then I think with that, I think your food loss index to me was very exciting, and I think expanding that and building a lot of the information that's generated at this platform into that will be wonderful. So thank you very much, Rosa, and thank you for supporting the Post-Harvest Initiative always, and for being a good friend of us. So thank you very much for your talk. And with that, I'm going over to our next speaker, Natalie, my chair. I'm going to introduce her. And for many of you, you know her well. She's a senior lecturer in food science and technology at Cranfield University, and of course that's where all the planes come from in the UK. And and I know your dean, your vice rector, well. He's a very good post-harvest pathologist and a friend of us. So with that, we're looking forward to your presentation.
Thank you, Lisa. And good afternoon, everyone. I know it's after lunch, so I hope no one falls asleep during this session. I would like today to talk about food loss quantification as part of this working group 3, but not only about the importance of quantification, but also the drivers, the reasons behind that loss, and how we can use that information to mitigate food loss and waste, and also generate actions that globally can sort this out. And try to put a little bit in context of what the situation is at the moment, trying to follow a little bit with what Rosa has mentioned from FAO, which I think it was really well summarized and kind of, oh, I've lost my presentation. It's all right. Yeah, can you see it now? No. Well, is it open? Oh, there you go. And then look at that food and loss waste drivers, those reasons. And then I'm going to try to explain one case study we've done in the UK that might serve as a method to follow this idea into other countries so we can get the information that we need. As we know, at the moment, we have a growing population. It's been mentioned this morning many times. We have increasing temperatures and other severe weather events. And that's also impacting on energy distribution, affordability of the food we have, and also the accessibility to that food. We have limited resources, limited food production opportunities, and even though that's happening, we are losing a lot of food. That's been mentioned in many COPs. It's been highlighted very severely in COP28. I don't know if you had the opportunity to follow it. And we are not only facing these type of issues, but also the supply chain disruptions, as we have seen with recent geopolitical situations, and for example, the COVID-19 pandemic that I don't want to be a catastrophist, but we will be facing similar or situations in the future. And all that is putting a lot of pressure in our system. All that is jeopardizing the sustainability, resilience, and accessibility of the food that we have. The estimations, according to FAO, of the growing population will imply that we have to increase the production of food in a 60% from the values we had in 2005 until 2050. And that only means that we are going to keep on putting a burden onto our food production system, increasing the degradation of our soils, the nutrition of our soils, reduction of the water availability, but also the quality of it, and increasing the greenhouse gas emissions. And that's why I'm here to just remind everyone that food saved is as good as food produced. So it's not only about increasing the food that we have, but also trying to preserve the quality and nutrition of the food that is already produced. Something when we talk about food loss and waste that, from my perspective, is sometimes overlooked is the nutritional losses that we have in our supply chains. So that's something that is more difficult to quantify, requires more technical resources, but that's something that we are at the moment looking at to be able to, through post-harvest technology, try to maintain that nutritional and organoleptic quality. So as I was saying, our global supply chains are facing huge amounts of pressure, and we still have or feel we have the luxury to have those food loss very high rates that Rosa was mentioning too right before me. But it's difficult. I'm not gonna say that it's not a challenge to try to quantify and understand different supply chains. Food chains are complex. They are very different from one country to the other. We need to maintain the food at different conditions during harvest, production, distribution. That's not only always possible, unfortunately. And we are juggling static and also mobile elements who need to be assembled all together in a perfectly find seamlessly for it to work properly, but that's not always the case. And I would like today to focus on fruit and vegetables. I'm not saying that the rest of food categories are not important, but as we know, fruit and vegetables maintain their metabolic activity after they have been harvested, and therefore their perishability is much higher. Also, when we talk about fresh produce, even though we have access to certain technology like freezing, like drying, and things like that, if we do that, we are changing actually the product. So it's not the same eating a fresh apple than having a frozen apple. So even though it might give us some of the nutrients, the consumer still wants to have fresh produce. So as a result of this challenging, the issue we have at the moment is that we have a lack of quantification detail in terms of food loss and waste that happens mostly everywhere. And when we have that data, sometimes, most of the time, is very focused on a specific part of the supply chain, on a specific supply chain in a country, and it's quite fragmented. So we don't have the full information from farm to fork that would be so useful to better understand how we can mitigate this food loss and waste. So in order to address this deficiency, I'm working with a multidisciplinary team in different universities in a project called Zero Emission Cold Chain, where we actually not look at only at food loss and waste, but also the environmental impacts that are linked to this, and how we can develop refrigeration technology and energy technology that can be sustainable, away from fossil fuels, but that still is able to meet the requirements of food. At Cranfield University, we are looking more at the biological drivers of food loss and waste, because at the end of the day, we are focused on post-harvest biology and technology, and this work has been done in collaboration with my colleagues, Ewan Gage and Professor Leon Terry. At the end of the day, this project this part of the project would wanted to know is how much are we wasting using the UK as case study, where that's happening, and why. And first of all, what we did is defining what food loss and food waste is following FAO definitions, so we are all on the same page. Being food loss more focused on the initial parts of the supply chain until it reaches retail, and food waste more looking at retail and consumer levels. So in high-income countries, in the industrialized countries, we see more food waste than food loss. In countries with low income, with lack of refrigeration, lack of cold chain, and lack of education to be able to understand what to do, we'll see more of food loss, but both are an issue, a challenge. In this study, the first thing we did was establishing the boundaries of our study. So it goes from farm gate until retail. We haven't included the consumer use in this study because the drivers behind it are more around awareness, are more around behavior of the consumer. And because of the technical nature of this project, we left out the behavior. I'm not saying that's not important, it's just the boundaries of this study. We did include imports only from the moment of storage, given that the UK relies around 73% of imports when it comes to fresh produce. We selected a few model crops based on the volume and consumer demand in the UK, and those are potato, apple, carrot, tomato, onion, cucumber, lettuce, sweet pepper, strawberry, pear, then broccoli and cauliflower, cabbage, and raspberry. Most of them are able to be, we are able to grow them in the UK, but we also rely on on imports to be able to meet all the consumer demand. And then the next step, what we did was a systematic literature review. So we wanted to know what was out there in the past 13, 14 years, and we focused on conventional food systems. Organic systems, we left them out too, because they have different challenges. And we separated the stages into harvest losses, storage, handling, and then retail. But what we found, as we expected, is lots of gaps in the literature. So the next step was to go and do a stakeholder survey. The idea was to obtain primary data, not only based in our study on literature and estimation, but also real data. Also to understand what the stakeholder saw, how they saw the food loss and waste and the limitations in the UK supply chain, and also to validate the data that we had on that literature. For that, we talked to the main growers in the UK, and we managed to cover 90% of the production in the UK. Our results showed that at the moment, 35%, a little bit more of 35% of food loss and waste is generated in the UK from UK grown products. And the main contributors were apple, onion, carrot, and potato. And that's because of the quality parameters, the requirements that the supermarkets ask for. Those standards are quite high for what we get and the lack of control we have during pre-harvest. We saw not a lot of waste in certain cases, and that's because the UK supply chain is quite short, so we have a little bit of wiggle room, but that won't be the case in other countries. So that's something we have to take into account. And the problems that we saw during retail were mostly dominated by consumer demands, a mismatch. So here you see in red the survey results against the black, that is the literature review. As you see, generally they match, mostly during retail, that they are exactly the same. But there is still more and a greater need to communicate with industry, a greater need to keep on collaborating, and a greater need for that primary data. So basically, we saw that quality and market behavior were the two key drivers. There was some loss in edible parts that there was no market for, maybe with new products, new ideas, we can get some market for those. And the reason for quality, that we had low quality and no match with the standard, was weather, pests and diseases, and lack of labor. That's a specific issue with the UK in this case. And market behavior of oversupply under demand, that's that mismatch with them. At the end, we need to think about if the specification we have at the moment are actually helping reducing food loss and waste or not. So that's the general concern that our stakeholders stated, was that they think food loss and waste is going to keep on increasing unless we change the requirements. Another key issue was pest and disease, given also the ban in certain products that had been used in generally, but we need to change. And we found a lot of differences between sectors. At the end of the day, what we found here is that the origins of food loss and waste, in the origins of food loss and waste, we have as a key driver the physiological reason. There's not that much research around post-harvest behavior when it comes to understanding the why behind those mechanisms, but that's changing, which is really good. And we definitely need a collaborative approach. And from here, I would like to call for collaboration in this cost action. Working Group 3, just contact us, and we can work as much as we can to get as much data as we can to be able to make decisions that are evidence-based. So for any question, just you can send me an email. And thank you very much.
Thank you very much. Okay. Thank you very much to my chair. And once again, we're a little bit behind schedule now. And is there a quick question? Otherwise, I'm going to, yes, a quick one and a quick answer.
Years from 2010 to 2023. So normally, to better understand, maybe you have to start from earlier point of time. And did you consider to make some subgroup or moderator analysis, meta-analysis, to understand which legume fruits are more lost or have the highest lost percentage for the intervention?
Okay, thank you for your question. For your first question, just very quickly, in the UK, technology has changed a lot in the past 10, 15 years. Going behind, like before 10 years time, it wouldn't make much sense because the technology is different and the data there will be completely different, and we wouldn't be able to benchmark with current technology and current practices. So at the moment we have people who are harvesting through remote sensing, for example. So trying to match that with practices that were like 15 years old would lead to put into the mix variability and outliers that are not real. We did do a meta-analysis to be able to understand which products to select. So that was prior to the study. But thank you very much. And I'm going now to introduce my co-chair. This is like musical chairs today. So I would like to introduce to Lisa Cortet, who is a professor in the University of Pretoria and who is going to be talking about food loss and waste in the context of South Africa. So over to you, Lisa.
Thank you very much. And I think you also, Prof. Leon Terry's photo on your slides, a good friend of us in the post-harvest world. So it was nice to see him. And with that, I have several hats that I wear actually. So I'm going to steal a few seconds of this presentation just to tell you about my latest hat. And I do drop a lot of balls, my apology, but I'm also now the president of the African Academy of Sciences. And it's really an amazing academy because we really represent the continent, and we have 6 vice presidents covering the whole region. And it's really about transforming lives through science and recognizing excellence in the continent, creating think tanks, dealing with government. We are part of the African Union. And very importantly, we have fellows that really we want to build in the next generation. And we also have extensive EU funding for big centers of excellence, which we will develop in the future. I just want to say a few words about the continent. And I don't know how many people know, but it is the biggest continent in the world. All these maps are totally distorted. You can actually fit Russia, China, America, and Europe into the continent, and you can add India and it'll fit perfectly. Having said that, 1/4 of the people in that continent go hungry every night. And we say 1/5 of the people in that continent is malnourished. And that's the most in the world. So things has to change. And what's interesting, if you look at the Global Food Security Index, South Africa is the only country in the continent that's food secure. But that's only at a national level. At an at-home level, we are in fact food insecure, which tells you a big story. South Africa is a prominent exporter of fresh produce. We are dominating in several fruit crops, citrus, avocado, mango, table grapes. And yet the questions about inequality is very prominent in our country. So you can imagine transformation for us in the food system is a priority. I also think it's important to look at the losses in the African continent, and they say 52% of whatever is produced in that continent gets lost and wasted. And a lot of it has to do with transportation, refrigeration. We don't have the supportive infrastructure to move the produce that's produced in our continent. But imagine we can. There's potential for the African continent to feed the world. And that's a WTO statement that said Africa can become a net exporter in the next 20 years if we can get our infrastructure sorted. And of course, climate change will intensify our challenges. So I want to make a statement, and I want to say, if Africa can stop wasting food and reduce our losses, we can feed the world. So let's go over to my research group. We have traditionally focused on fresh produce, quality, primarily post-harvest, but also post-harvest pathogens, starting pre-harvestly, looking at control, biological control. And then after that, we started looking at food safety in the fresh produce chain, because ultimately it affects the product that we put out on the market. And if we're not compliant, we will not put that product on the market. So the facts of South Africa's food waste and losses, and this is very important because this is a recent study from the CSIR, is that we waste about 10 million tons of fruit per annum, 45% of the available food. Now, keep in mind the statement I made about food security. and food insecurity in our country. So when they analyzed that, it was more or less 34% of the locally produced food. But remember, South Africa is primarily an exporter. So that waste percentage is actually skewed. It's actually 45% if you look at what's available in the country. And analyzing these chains, there's a lot of critique around the inadequate system that we have in place. And I hope to just unpack some of this for you. If we look at where the waste happens in South Africa, the bulk, 68% is on farm. And I think as a plant pathologist and an agriculturist, I find that strange because we have very effective integrated disease control management systems. And South Africa is one of the countries with the lowest percentage of non-compliance in terms of pesticide residues exceeding the maximum residue limits. So we do export well. We do extremely well on the export market. Yet the statistics does not reflect what's happening, which tells me that we are looking at the commercial sector. And what I want to reflect in this talk is the whole massive informal sector that is just growing in South Africa with growing numbers of small-scale farmers and food that's then be retailed in the informal sector with no regulation, no control. And the question is that distorting these figures? Of course, 49% is lost at processing and packing, and then 18% at the point of consumption. And if you look at the commodities, then fruit and vegetables would be 19%. So let me just show you a few interesting examples. And this was quite a few years ago. One of our PhD students worked on table grapes, and she actually tried to find out, well, why do we have this high waste and losses? And we took table grapes. It's a major export commodity. Most of these farmers are Global Gap certified. These farms are super efficient. They are actually fantastic. And we looked at, and she focused specifically on Botrytis cinerea, and we tried to map and understand what is happening from flowering all the way to harvest. And remember, we've got a summer and a winter rainfall region, whether it's in the northern provinces with a summer rainfall or in the Cape with winter rainfall. And both provinces do produce a lot of table grapes for export. So we saw that botrytis is more prominent during flowering stage compared to the other provinces. And that shifted our guidance for the industry in terms of disease control. So I think her study was significant. We had another student that then focused on the post-harvest part of the picture to try and figure out what is the major problems for us in the post-harvest environment. And the photograph that you can see is one of our fresh produce markets. Now, just to tell you, South Africa has most probably one of the best regulated and controlled fresh produce market systems in the world. We've got 16 major fresh produce markets. It's highly regulated with inspectors. Yet, due to a management collapse in the system, we now have incredible waste and losses. And it's primarily because of poor hygiene. And to prove that, we actually monitored the air quality in these major markets, and we looked at the volumes of fruit that's lost, and specifically focusing on penicillium because that was our major loss, and which penicillium. And these were just some of the air samples that we had from the major markets that we collected. And I don't know if you have your favorite pathogen. I think everyone in the world should have their favorite pathogen. Well, mine's penicillium. I just think it's such a cool pathogen. Nevertheless, so this specific student looked at the different penicillium species, both in the air and on the fruit, and we tried to link that with the waste and losses that we found. Now, what was interesting about this study, at the same time, we included an agricultural economist, and she did the whole project with us. So she worked with us on the farms and tried to understand volumes produced, practices, and agreements, contractual agreements, rejections. What kind of losses do we have? And what was interesting, if you look at the blue bars, the average percentage losses was the biggest in the export products. Keep in mind, this is an industry that primarily exports. And what was interesting that at the local market, the losses were almost nothing. But what was interesting, and one should remember this, is that we're having incredibly strict export requirements. So we have inspectors at each backhouse and at our export market and at the import market. So the farmer's loss for any export product is huge because of the strict requirements. And the question is, well, who's paying for these losses? And particularly if you look over time, and you'll see the blue bar is the losses at the retail point, which means once the product is received by the retailer overseas, they have another layer of inspection. By this time, that fruit is physiologically old. It's already giving in, and you cannot expect the same quality. And with stringent requirements, we have seen over time the increase of losses at the export market. Keep in mind the contract, the farmer is still the owner of that product until the retailer takes hold of it. So for us, it's huge, huge losses. And if we unpack the reason for losses, then the blue bar is physiologically, and what we can see that pathology is the botrytis and penicillium is the yellow bar. And you can see a significant reduction in that from 2011. And we hope to lay claim to that because I think of our research and advice. Now, I don't have a lot of time, but a very important story. And this is explaining to you the formal versus the informal sector. So here we have this highly regulated export farmers, all Global Gap certified, perfect. And then you have these supply chains for the informal sector. We had a question, is food moving through these informal sectors less safe than food going through the formal sector? And I can just tell you in short, the outcome of that study after 10 years is, no, it's not necessarily less safe. And the reason why those supply chains are so short, the food moves through it so quickly. They literally pick it and go and sell it, and people who buy it, buy enough, and they consume everything. The concern there was then what happens with your practices. So we started unpacking these practices. This is at some of the retail ends. You can see we call that a shopping trolley. It's full of fresh produce that's then pushed through informal settlements where people will buy it. And the problem is there's no potable water. So they wash the spinach in water that is contaminated. So the next theory was, well, perhaps we do have a lot of foodborne pathogens on these crops. And then we started looking at antimicrobial resistance. And what was very interesting was that you can see 43% of those were actually multi-drug resistant E. coli that were found in the informal sector. We then started comparing the formal and informal sector in terms of these antimicrobial resistance. It was very interesting with Eric PCR. We could do source tracking and we could trace it back to our water, which comes from highly polluted rivers. And I don't know if you know, recently we've had a cholera outbreak, which in a way reflects the poor management of our water systems. And this is typical informal producers, small-scale farmers, and they move their produce to these informal markets with a local vehicle. And you could see 92% of the microbes from that, the salmonellas particularly, were actually multi-drug resistant. So this is definitely a new wave and concern coming. And lastly, a very important example for us is two of our PhD students, Edwin and Grace, and they're not here, but they prepared these for me. And they were looking at informal small-scale farmers, green pepper and tomato production, to find out where they lose their product, what is their level of understanding of quality and using practices that is fit for purpose, and how can we upscale these farmers to get a better price for their product. So in this case, with Edwin, he found that 13 to 25% of the produce that was actually collected from these people would decay. It was primarily Alternaria. And he also looked at the reason for these losses, and a lot of it deformities, moisture loss, and then diseases. So my time is up. And very interesting, we tried to link that with microbiome studies to try and figure out decayed fruit and healthy fruit. And what's interesting is the most abundant genus, Cladosporium, Alternaria, and Fusarium. So we could link the Alternaria with the Alternaria post-harvest that we are picking up on the fruit. And then Grace was doing similar studies on tomatoes and finding a very similar pattern. So I think the information generated by these two brilliant young students will tell us more about more effective mechanisms and best practices that we can then share with the small scale farmers. I just want to close my talk. My time is up, but I just want to say something and go and look at this. The latest release from FDA, 700% product recall increase. Now that's significant. So we are wasting a lot of food in the chain due to product recall. And this is a very interesting article. And I want to also mention, let's not forget what's wasted on the plate. 41% is one of these global groups. It's monitoring it. And I don't know how many of you finished all your food on your plate today, but keep that in mind, practice what we preach. And then finally, excitingly, some network analysis, trying to figure out what's main reasons, where's the main losses. If we look at the literature at a farm level, storage, supply chain, and most of the research is focusing on diseases, then physiological, and then temperature. So finally, I know my last word, we mapped the timeline of patents, and what's significant is we used AI. What's significant was we could see this incredible increase in post-harvest patents that's registered. The best part of this story was, and we were analyzing to see what are they patenting. You can see the reddish bar, that's antimicrobial agents, your biocontrol products. And then thirdly, your refrigeration systems, and so it goes down. But the countries who's registering patents at the moment, the most is China. And it's the Chinese universities, which is a very interesting point. So thank you very much. From South Africa, we have tremendous load shedding. We dump a lot of fruit purely because our fridges are off 4 hours a day, sometimes twice a day. And this is just some of the statistics we have from most recent information. Thank you.
Thank you very much, Lisa. Really, really good presentation, really interesting, and put in context how other countries' continents are working. We might have time for a quick one question, if anyone has, but I'm sure if not, there's plenty of time to discuss with Lisa all these points. Thank you. So our next speaker is Andrea Segre. He's going to join online. He's a professor of the University of Bologna and the scientific director of Waste Watchers International. And he's here to talk about this tool and how we can better understand the waste worldwide thanks to their observatory. Also been told that there might be some technical issues, so bear with us.
Thank you very much. I hope you can hear me. I am in a very precarious condition. Actually, I'm in Rome and I presented here a project related to our observatory. I asked to Gianfranco, I thank him for inviting us in this important event and conference, or my colleague Luca Falasconi, whether they can share our presentation. If not, you will listen to me without having our PowerPoint presentation, and I will send you later, because this internet connection through Chrome, it does not allow me to share the presentation. Anyhow, I'll go because you know that we have a schedule, and I'll be strict to the minutes you gave me. We just a short presentation of our group, which is based in the University of Bologna. Thank you very much. I don't know whether Gianfranco or Luca. Okay, fantastic. Move to the next one. We started to investigate this field now called food waste and loss back some 25 years ago with an academic spin-off, Last Minute Market, that actually is still alive, and I would say also kicking, starting with the observation that we have unsold products within the retail system, basically, and we wanted to find an innovation, a solution to recover through donation these unsold good products to benefit charities, while in some 25 years ago in Italy, at least, the issue of food poverty was much less important as is nowadays in our country, and I would say elsewhere in the world. Then, after some 10 years of research and application, this academic spin-off is a social enterprise, and we implement project in Italy in particular, while our department is engaged in a number of EU and world academic projects. So we have this important opportunity to merge research with application from the field. When we started back in 2003, our recovery program to benefit charities, we had from the field, from charities, a number of technical, scientific, logistical, administrative issues that we try to solve through our academic research and then implement in the field, in the ground. So I'll keep this story very short. After some 10 years, we realized, so back in 2010, that along the food chain from farms to restaurants to households, most of the waste was concentrated actually in the household, in our household. At the time, we estimate more than 50%, so making 100 all the filière, all the chain, and now is a little bit more as of our estimates. So we try to focus our research and then our application on consumers, on behaviors. Also because in our household, all what is left is must be actually destroyed with a cost, and a cost which is, sorry, I am in an office and the owner of the office just entered. I said, when you are at household level, all what is left is garbage, and you have to destroy with a cost, an ecological cost and also an economic cost. So we tried to understand whether we have the possibility to implement a tool to investigate behaviors, our behaviors, and try to find solution to prevent, this is the key word, prevention, food waste. And by the way, we were in the frame of the hierarchy of waste in Europe, where at the first point you have prevention. The best waste is the waste that you prevent, so is no waste. So let's move to the third slide. We set up an observatory. Go on, please, Gianfranco. An observatory that we pilot in 2013 on behaviors. What does it mean on behaviors? We panel a sample of consumers, so an investigation with a methodology that I will explain you in a few seconds, having the opportunity, you remember perhaps 2015 Expo Milano, dedicated to food, and so open this sector of investigation, of measuring in at the worldwide level. And so the first works, the first pilots of this observatory, Waste Watcher, started some 10 years ago. And then through the years, we publish, and we, when Rosarolle mentioned earlier the International Day on food waste, on awareness on food waste and loss, we decided, also with the help of the FAO, to enlarge our observatory and then move to an international level. I have not here some data, but we can provide you with the results of our studies that we present in two waves, usually each year. One is the National Day, which is on food waste prevention, the next 5th of February. So in a few days, we will have the new data, and the 29th of September, our international presentation in the frame of the celebration of International Day. Move forward, because I'll show you the methodology. Behaviors, yes, lifestyles. Then objective is to understand why we waste, and final tool is to find policy options to benefit governments, institutions that can help to reduce food waste at the household level. We will see that we are going also outside our household, and try to implement measures that has this first objective, prevention, and also reduction, because we know the general figures at the worldwide level, and we see that this is an issue not only from an economical point of view, but also in terms of environment, and also production of pollution, and an effect important on global warming. So the methodology is very solid. We publish a lot from a scientific point of view, because we work, as I told you earlier, on representative sample. So is a panel of consumers by region, work, gender, and the like. With this CAWI methodology, is the computer-assisted web interviews, with a scientific approach which is very solid, motivation not to waste, abilities supporting food waste reduction, and last one, evaluation of intervention that tools that can have a reduction or a prevention effect that we call opportunities. And this is actually the part that should be focused and implemented by governments. Move to the next one, please. So in this framework, we can also make a sort of specific focus. Our first aim is to give a sort of gentle push to consumers to have a better understanding of their behavior. Nobody in 25 years of our work told us, I waste because I want to waste. It's a matter of behavior, of knowledge, as we said, and so we try to understand what is in the background of this behavior. And we can, with this methodology, not only go within our household, but also outside. I'm here in Rome because we just launched the project to estimate waste in restaurants. Somebody presented a law on application of what we call in the Anglo-Saxon word doggy bag, which is a normal actually behavior in a good part of the world. In Italy is not in our tradition. Along the years, I presented a number of projects with different names, because, as you know, doggy bag sounds a little bit just leftovers to your dog, and it actually is something that you paid and you can eat. But we don't have data, although if you see some EU projections and percentages, apparently we have. Italy is not correct, the figure. And so in order to implement this tool, we set up a project that we just launched through Waste Watcher, to have a focus, a sort of vertical approach, in order to have a qualitative and quantitative estimate of how much is and for which reason the food waste in this part of the food chain that exclude actually canteens and the rest. Move forward. So our analytical insights at this time are used, at least at the national level, to push not only consumers, that is our first aim, but also policymakers. And I can tell you that among the other influences, we sort of push the Italian Parliament back in 2016 to adopt a law related to food waste in particular, and to recover food products for charities, but not only food products, but also pharmaceuticals, for example, and other items that could benefit actually people in need. This is an example, but I can actually give you other examples. So it's the way to have a solid information base in terms of data, in order then to choose the right tool, the right measure, the right law to get the aim. The last product of our work, Move Forward, although it's not in this presentation, but from this observatory, we derived an application called Sprecometro in Italian. In English is waste meter, because, you know, we talk about Agenda ONU 2030 on sustainable development. The target 12.3, 1, 2, 3, which is that we know that we should reduce by 50% per capita waste and losses. Well, if you don't have a metric, an engagement, and nobody tell you how to do it, it's very difficult, and actually results are not really in line with this target. But with an application that is dedicated with a sound methodology to measure your household food waste. By the way, you can work by yourself and in groups, schools, associations, companies, municipalities. So it's very well known now in Italy. Well, you have a number of contents of, let's say, advices, gentle advices, how to read the expiry data on a label, for example, how to cook a leftover and the like, that can measure your impact in terms of reduction, having a perception. Actually, I didn't say this is in terms of methodology, self-perception. So it's an engagement that is straight out of your experience. Well, you can measure. So you have, for example, a leftover today, I hope not, to buy 100 grams of cheese, and you have straight money and the environmental impact, and then an advice how to conserve, for example, in your fridge, the cheese. Okay, we are able to give you some data and also some information on this application, because ideally would be something that could be applied everywhere, because our aim always is food waste prevention, if possible, to zero. In fact, I conclude our campaign is called Zero Waste Campaign. Is possible, and perhaps our research can help to reach this important objective. Thank you very much for listening.
Yeah. This point. Yes. Maybe we can wait for the microphone so Andrea can hear you. Andrea, we are bringing her a microphone. Just let us know if you can hear her properly.
It's indeed very interesting to know about the food waste and food loss from the restaurants, but how to collect the data? Are restaurants owners willing to provide it? I mean, how to tangibly realize it? It's very interesting how they do it.
In terms of methodology, is the same. We have focus group.
You're on mute, but we cannot hear you. I don't know. You can hear me.
I can hear you.
Yeah, I can hear you now. Perfect.
Sorry. In methodologically speaking, we use Waste Watcher and focus group, but they are very much willing. I don't know whether you see on the back, I am in the seat now of FIPE, is the Italian Federation of Public Exercises, and they are willing. And by the way, they presented a new fantastic bag called Rimpiattino, difficult to translate into English, but is something very much far from doggy bag. So they are willing. Ourselves as consumers, out of our data, we are 30% of the Italian population is embarrassed. They don't want to ask. It's better to leave because it is a sign of opulence. Okay, so which is something unbelievable nowadays. So really is more a work on the other side, because restaurants, they are not happy if you leading something in your dish, because could be that is not good. So we are in their federation because they are willing to do, and perhaps we can show you in some 6 months the results of this study.
Victoria Wojciechowska, I hope I pronounced it correctly. And she's Associate Professor of Economics, so that's cool. That'll be very nice to read. Polytech National University from Ukraine.
Yes.
Nice to have you.
Yeah, thank you very much. Thank you for invitation. I'm very happy and proud to be among the distinguished speakers. Not so fast. Yes, so actually to give a little bit of the context and then to connect with another presenters, that indeed the ambitious goal of the Agenda 2030 shows us the more interlinkages between SDGs, and indeed the waste management contributes to, waste management contributes to most of the goals, like no hunger, the inequalities, education, and sustainable and responsible consumption and production. To achieve this ambitious agenda by 2030, we would need to act actually now, since it has been estimated by the United Nations this year that at the current state of events, we would need like over 200 years to be on track for the ambitious goals of the agenda. And of course, since now we are in Europe, we are more or less as Ukrainian, I assume I'm European too. So we are privileged, and we have one of the best policies and one of the best evidence-based policy making in the world. However, Europe is very unequal on achieving these ambitious goals and targets. And some countries have very different rates. We will see you can go on other slides, or it's me who. Yes, so more or less to have a global perspective, that evidence-based policy making and having kind of a plan to achieve the ambitious goal 12. We see here like countries that was more or less policies on track from the United Nations statistical divisions, and countries that do not have policies, do not have a plan. And to have a plan and to have it quantified and measured is very, very important in order to achieve the ambitious goal 12 and consequently other goals. So coming back to the topic of my presentation, I'm going to speak. There will be some technical details, but I will try to minimize them. So we are going to speak about the tool that is called using the fuzzy inference methods as a part of composed indicators to see the scenarios of development in Ukraine if in the context of EU Sustainable Development Goals and the circular economy scenario. So we see here a tree that basically on the right side there are indicators. I'm not going to name them because most of them you know, and so actually you can see it in a structural way. So it's kind of 3-level. So basically we have input indicators, we have then intermediate indicators, and we arrive at the composed indicator of waste management. And of course, here we have a global indicator framework structure and you see like 17 goals and how the regional organizations and regional national statistical system is working together with civil society organization, private sector, and academia, or to process the data for better policies and better solutions. And of course, why composed indicators in social research is actually relevant, because more often than not we have a lot of data which comes from different backgrounds and can be qualitative, quantitative, and it is important sometimes it is important to reduce the dimension and not to lose the important information from this data, and that indeed to make a proper aggregation and proper normalization and arrive at the indicator that can be used for prediction and can be used for benchmarking and can be actually of use. My time is not ticking, it's normal, yeah, because it's unlimited. Okay, so the features, actually why it is important to quantify this data that we receive from national statistical offices, but not only. So the structure complexity, some presentations before, I believe from working group 1, we had this holistic view from the complexity of the circular economy and let's say seeing the whole picture in a holistic way and why it is important. Then we have the operationalization of sustainability, meaning that sometimes with partial information we have to make an important solutions and still make use of it, and not always is possible to have all the indicators that would be black and white. Then we have, of course, social learning, and we have a challenge to interpret information and the influence challenge. Then we have to demonstrate accountability and benchmarking. Yes, therefore our output would be useful for researchers and the policymakers. And then, of course, the data gaps challenge and the information structure challenge. We propose one of the approaches here can be with the use of fuzzy logic tools, and the example of Mamdani. Mamdani method has been invented in 1975, and it is of course an important method in the area of social research, as it is, first of all, it is intuitive. It's very well suited for human input, and it's excellent actually for policy making and benchmarking. So here is a very brief what Mamdani method is, but I believe we are short on time. So the building of composed indicators actually has several steps. First of all, we have to have an input indicators for the study that I will show you during next slides. We use the data from Eurostat, and for Ukrainian statistics, we use both the Ukrastat, the Ukrainian statistic official information. The baseline year, was 2020. So indeed, the elementary indicator selection is very important. As well, what is important is that the indicators are unidimensional, meaning that we have a sample that has a comparable data. Yes, in other words, it is not always statistically correct to compare the data from, excuse me.
Starting to close up. Yeah.
You wanted to say something, no?
Yeah, you have to wrap up for time.
Oh, okay, the time. Sorry. Yes, so actually, like to present the results for the European countries, UK, because the base year is 2020, and then the Ukraine. So actually, that the 5 clusters were the clustering by means of fuzzy k-means using MATLAB was performed, and we can here see the countries with similar waste loss, and maybe here is better picture the ranking of countries by the level of development of the circular economy. And we have here the waste production, the level of investment, which is very important, and integrated assessment by the section waste management, and the level of circular economy patenting. So patents were also in a previous presentation, and it is an important part along with investment. Indeed, to see how so on the left side, we have the input indicators, how the simulation model for calculating the dynamics for the entire system looks like for the waste management, and the scenarios, which is actually my favorite part, and I would say that the most interesting, of course, to tell the context, as you know, that Ukraine is in the state of war, and then actually after pandemic, Europe in general is in very vulnerable situation. Therefore, there are some factors that actually there are factors that we cannot neglect, but at the same time, it is important, however, to make projections. So the projections here are Ukraine, if giving a little bit the context, as you know, that some countries in European Union are doing very well in waste management area. Some countries have over 50% of waste management. That is actually, I think, Italy, yes, Italy, Belgium, Netherlands, and Netherlands, of course, the Nordic countries, but some countries are not really doing very well, such as, or maybe some countries don't have very well the data collection. in place. For example, Malta, I had for some indicators, they had simply 0, which is impossible. That Romania as well, surprisingly, Estonia and Bulgaria. So Ukraine is, to make it more simple, somewhere in the middle. According to official statistics, the waste processing is around is close to 15%, but in the reality, it's very difficult sometimes to measure and to see that actually what and how things go. So indeed, yes, so the perspective is by 2025, including the pandemic and the war, Ukraine is more or less going to, the waste recycling rate is going to drop. But probably if the war is over, then the things are going to be better in 2030. But yes, but indeed it would depend on many other factors. And the conclusions. The conclusions, of course, it is important. First of all, it is important to benchmark in order to make proper evidence-based solution. It is important to have clusters of countries and with similar data and similar policies. It is important to calculate for each of them. For each of the block, the set of composed indicators. And of course, then we can, having in mind the base year, we can track and we can see how the country is doing on the circular economy path, and therefore contributing, yes, to the goal 12. As well, for the Ukrainian context, not coming back to previous slides, but I would like to tell that it is important to identify critical points and so-called growth points. Therefore, it is important to make sustainable investment in the sector of circular economy. And there were reports from the United Nations Development Program, for example, that Ukraine can build on the momentum and actually rebuild in a more sustainable way and have a better future. Well, hopefully so. And yeah, so I believe, yeah, thank you for attention. I believe I managed.
Thank you very much.
Thank you.
Thank you very much, Victoria, and I'm sure we can catch up during the coffee break. Our last speaker is Miguel Elias, who is professor in the Mediterranean Institute for Agriculture, Environment and Development in Portugal, and he's going to be looking at a quantification of food waste, in this case, Portugal meat plants. So over to you, Miguel, and thank you.
Thank you very much. Good afternoon. And we will we'll have a very short talk concerning food waste quantification in Portugal, Portuguese meat plants. This work was prepared by me and by my colleague Ana Cristina Santos. The results presented in this yes. The results thank you. The results presented in this study are a part of a larger study focused on 4 agri-food sector in the Portuguese agri-food industry: meat products, fruit and vegetable, bakery industry, and dairy products. We will present only a few results of meat products. The aim of the big study was to obtain a diagnostic of the agri-food sector in Portugal, a tentative to obtain a diagnostic. For this study, we used face-to-face surveys, and we inquired 11 companies, Portuguese companies. The activities of those companies are mainly meat products, transformed, processed meat products. 6 companies, 3 companies produce raw meat products and processed meat products. 1 enterprises raw meat products only, and a slaughterhouse was also inquired. The results concerns to the situation in 2020. The objectives of the survey was quantify waste. It's quantify, but using surveys, not measuring directly. Identify the destinations of the waste, record procedures to reducing and to recovering waste, and the other part wasn't shown in this presentation. 11 companies, 11 enterprises. The small one has 4 employers, and the total sales in 2020 was 156 million. 100,000, forgive me, thousand of euros, and the biggest of those more than 23 millions of euros. With this one, with 250 employers. We can see in this slide the amounts of the production, the identification and amounts when possible, when related during and destination of the waste during production, and the same during marketing. And we can see that there are several enterprises with no records. The biggest part, a significant part, are no records. The destination of the waste, it's several origin, casing damage, losses during cutting and slicing, bones, slaughter products, and during marketing, the main loss was due to the loss of packaging vacuum. It's the main cause of losses. And during production, we will see another slide, but the scenario is it's the same. During the manufacturing, the principal cause or reason of loss is slicing. When they slice products, the amount of waste is it's very big. And during marketing, the losses of the packaging vacuum is the main reason for the losses. The destinations are or mainly animal feed, or donations, or destruction, mainly destruction. One exception is the enterprise number 11 that reuse the biggest part of the waste. It's and the other significant information is the total amount of the waste produced during production by company number 5. It's slaughterhousing, and that company, or this company, produces a big amount of slaughter products, mainly bones and other slaughter products. Well, we can remark that the destination of the losses are animal feed and the destruction mainly. We could say that the principal destination of the or reason of the waste in during production, it's the resulting from the slicing operation, and during marketing, the loss of packaging. We can remark that the enterprises, the companies that have systems of certification are more representative in the records, and the losses represent between 0.05 and 20, almost 21%. I don't believe very much in these results, because they are obtained in face-to-face surveys. That's why we are preparing a project selecting some of those companies in order to measure according to the regulatory rules, the quantification of those losses. And an important data is that there are very few data. For reducing food waste, some companies, the bigger part of the inquired companies, made a better production management. They intend to be in accordance with between the demand and the production, and this is several times the big problem and result in devolutions and in losses. Two companies are invested in packaging equipments, because to improve the quality of the packaging, and other two enterprises are invested in technological improvements in order to reduce the losses. To valorization, and this is not the goal of this group, but just to say that one enterprise sell his bones, the tons of bones, to the market Chinese to produce soups, and there are a big interest in reuse for those enterprises, a lot of them, to reuse the waste resulting from the slicing process. This slicing process occurs mainly in sausages, very expensive, and dry cured ham. This kind of waste is from the sensorial point of view rich, from the nutritional point of view rich, and it's one of the goals of the project that I told you, to obtain new products using the waste produced from mainly meat plants and vegetable plants. and we have some ideas. In a few years, we will present the results of those researches. And thank you very much for your time. Thank you.
Yep. Any questions? I think we've done well. We've just run over time. Perfect timing wise. Thank you. And with that, I'm going to close this session. We ate away a little bit of your tea time, but I'm sure you can have a quick coffee and return. Over to Gianfranco.
Now, again, thanks to all the speakers for the valuable information that's shared with all of us. That will be very valuable for the continuation of this network along this time and maybe beyond. And thanks to moderators for this very good session. So thanks to all of you. Some very short announcement. One is that some of the speakers of tomorrow still didn't send the presentation to Lucrezia, and she is complaining about this. So try to do it as early as possible. The second is that this meeting was run in cooperation with CHU Association, the Italian Association for Plant Protection, and the Italian Phytobiological Society. And we have three sponsors. There is the Ministry of Foreign Affairs, Frontiers in Horticulture, that is a new journal that launched the special issue that we are coordinated with Luis Palou and with Barbara Blanco. And so the first two that arrive have the complete waiver of fee. So you are welcome to send, if you want, your important research results to this new journal. And last but not least, we were with Victoria, and the day after tomorrow, it will be presented in the university, in the Faculty of Economy, a book written by a writer from Ancona, that is Pierfrancesco Curzi, and the name of this book is Checkpoint Kiev. We as university go toward the peace, and this book has an important part dedicated to Victoria, so you are welcome to talk with her. And if you are still here around on Friday at 12:30, you can follow this kind of presentation. So outside on the left panel, you can have some further information if you want. A good coffee break to all of you. And in 15 minutes, we can be back for the last session on valorization.
We are ready for the last session of the day. It's a special session because it will last for 2 days on the road. This is only the first part. And that's because we have a lot of speakers for this working group. This is the working group number 4. And the focus is on the valorization of the agro-food waste. So we all wish to reach the zero waste, of course, but with the waste that so far we have, we will need to figure out the best way to valorize it. So here we are for this reason. And I'm starting with the introduction of the first speaker of the session of today. Is Dr. Milliken from the University of Greenwich in United Kingdom. And her speech will be about the valorization of agri-food waste for a circular bioeconomy, highlighting policy incoherence through nexus thinking. So please, the floor is yours.
Thank you very much. It's very nice to be here. I just put on this first slide, you can see the little logo for NexusNet. That's another cost action that might be of interest to you. That looks at the nexus between food, water, and energy, which is part of what I'm going to be talking about later. So I'm just about to take over leadership of the working group on policy. So again, if any of you are interested in policy in relation to food waste, you might want to get in touch to see how you can get involved in that cost action. So the premise for my talk really is that if we accept that the agri-food system will always result in some waste, in other words, byproducts and residues, better word than waste, how can they best be valorized? So how can they be made more useful or more valuable? And to me, that always raises a question about valuable for whom? Are we talking about valuable for society? Are we talking about economic value for somebody? I think it's an important point, which we'll come back to. So I'm just going to, in this brief talk, I'll be looking at sort of three key points that I want to get across. One is the concept of nexus thinking. So not like a siloed approach to the agri-food system, but looking at other systems that are intricately entwined and cannot be divorced, and with which there are feedbacks. The European Green Deal targets and ambitions, and the food surplus and waste hierarchy. And of course, valorization of food waste is central to the EU's bioeconomy strategy, which it aims to adopt or to address the three pillars of sustainability, so environmental, economic, and societal. So in terms of valorization of agri-food waste, we're talking about improved exploitation of organic residues and byproducts, so taking outputs from one value chain and using them as inputs to another. In other words, waste becoming a resource. So looking at nexus thinking, we've seen this so-called wedding cake diagram already today. And it was a publication in 2016 that showed how food really connects all of the SDGs and shows how economies and societies need to be embedded, seen as embedded as parts of the biosphere. So there's a nexus between all of these sustainable development goals. This connection between them obviously means that in order to achieve some, you cannot achieve others. I think we've all woken up to that fact by now. Looking at a smaller nexus, okay, the water, food, energy nexus. So you need water and energy to produce food, energy to produce food, and water, and so on and so forth. The nexus, the link between them is inextricable. So you do something to one sector and it has an influence on another. So everything's connected. So when we're looking at the valorization of food waste, then we need to understand the impact of food waste on food and energy and water and other systems like climate. And some of these things have been alluded to already. Looking at the European Green Deal, there are various targets: reduction of greenhouse gas emissions, increase in renewable energy, in the farm to fork strategy, increase in farmland under organic farming, decrease in the use of fertilizer. You know, how can these various different targets be reconciled in a system, in a nexus where impact in one sector influences another? The question of agri-waste is kind of vaguely treated in the farm to fork strategy. There's the recommendation that farmers should develop production of renewable energy from agricultural waste and residues, and even perhaps to source waste and residues from other sectors, such as the food and beverage industry. There's the suggestion that organic waste could be recycled into renewable fertilizers and can be used to develop alternative feed materials, for example, using insects as bioconverters, using byproducts from the bioeconomy. In terms of water, the EU doesn't really have very strong targets about water use and water conservation. Just in relation to the SDGs, there's kind of a vague commitment, no specific targets to contribute to the reduction of water use and ensure that the cap contributes to efficient management of water. So let's look at the food surplus and waste hierarchy, which I'm sure you're familiar with. It's hierarchical from the top to the bottom in the order of preference of how waste should be dealt with. So reduction, redistribution of edible food to people. Below that, the use for animal feed, and then going further down, anaerobic digestion for the production of energy and digestate. Below that, compost, incineration, and as a very last resort, landfill. So this is what I want to be looking at in relation to how these policies can fit into this. This is a very interesting study published last year by Postharvest et al., where they carried out systems dynamic modeling to look actually at the environmental impact of different waste management options in different sectors of the agri-food system. And this was a case study done in Bristol, which is a city in the UK. And what they found is that different food waste management strategies have very different dynamic impacts on energy, water, and carbon footprints in different sectors of the food system. So you can see on the left, in an energy-oriented food waste management system, incineration actually comes in at second position in primary production and in third position in the other sectors, which does not conform with the conventional food waste hierarchy. Water-oriented food waste management, in terms of environmental impacts, the best options are reduction and redistribution, and also to a certain extent in carbon footprints. So just because a certain waste management option is higher up in the food waste hierarchy doesn't mean that it is necessarily the most sustainable way to treat that that waste in terms of impact on other sectors. So yeah, you can see from this diagram that Postharvest et al. Found that reduction and redistribution are always the two, mainly the two preferable options. But then moving on to the third option, the next one is more ambiguous and leads to trade-offs between carbon and energy footprint. And then more recently, there's been a suggested revision of this food surplus and waste hierarchy, which is upcycling. So this would be more preferable than redirecting organic residues to animal feed. And upcycling has become very trendy. So a classic example is spent grains from the brewing industry. So a lot of barley grains are left over from making beer. And now you can find many companies setting up, selling this upcycled grain, primarily made into flour, which tends to be at least twice as expensive, if not more, than conventional flour, but also very much made into snacks. Is this what we should be doing? Is it better to be making snacks for humans rather than making it into animal feed? I think that's rather questionable. Something that we need to think of. So just going back to this hierarchy, how then do we reconcile the various targets in the European Green Deal with the hierarchy? There's the increase in renewable energy, the aspiration, which has been the target's been increased even more since the Ukraine war. But if anaerobic digestion comes so far down the hierarchy, how do we meet those targets? We know it's no longer sustainable to, grow biomass in fields which should be used for making food. Animal feed, if we're redirecting things, into upcycled snacks and so on, so where are we going to get our animal feed from? We're going to import it. So what's the environmental footprint of that? The ambition to increase by 25% the amount of organic farmland in Europe. How are you going to do that if you're redirecting, constantly redirecting organic resources up the pyramid? So essentially, what I'm doing is I'm questioning the validity of this. I'm hoping that tomorrow in Working Group 4 we can, have some quite good discussions about, first of all, about terminology when we talk about, waste, byproducts, or streams. What do they mean? And yeah, to see if there are better strategies for valorizing this waste. Am I out of time? I heard tick, tick, tick here. 1 minute. Okay, well, I'll skip that slide, maybe save it for the working group tomorrow. And thank you very much.
Thank you, Sarah. So we can have one question if someone wants to ask. Okay, then thank you again, Dr. Milliken, and let's go on.
Okay, for the next speaker, Dr. Ivo Safarik from Department of Nanobiotechnology from Czech Republic will present for us magnetically responsive waste biomaterials for environmental technology application. The floor is yours.
Good afternoon. Thank you very much for the introduction. I think for many of you, it's quite interesting to see the word magnetic in the title. And you may ask why magnetic materials are so interesting and important for me. So it's a simple answer. Magnetic materials belong to the group of smart materials which have very interesting properties. For example, these magnetic materials can be selectively separated from the studied system. They can be targeted to the appropriate plate using magnetic field. They can be kept on the place using specific arrangement of magnets. They can produce heat when present in the alternating magnetic field. It's important for cancer therapy in medicine. They can be used as a contrast agent for magnetic resonance imaging. Some organisms use magnetic particles, nanoparticles, for navigation in geomagnetic field. There is a group of magnetorheological fluids which can change viscosity when changing magnetic field. And sometimes magnetic metals can harden some biological structures, as exemplified by the teeth of chitons. And what's very interesting property of iron oxide magnetic particles, in 2007, it was observed that magnetite nanoparticles exhibit enzyme-like property, peroxidase-like property, as they became the first nanozyme. Okay, so magnetic materials can be used from molecular biology up to environmental technology, via biotechnology, biochemistry, medicine, bioanalytical chemistry, and so on. Basically, the same procedure can be used for manipulation of microliters as well as millions of liters of liquids. They can be used for work in suspension systems, and in addition to more advanced separation techniques, non-separation techniques are also very important. Okay, let's go to magnetic modification of food waste. It should be mentioned that food waste belongs to the broader group of general biomaterials. In fact, these biomaterials are mainly organic ones, so based on different types of biopolymers. But we have also some inorganic biomaterials like eggshells or oyster shells. In most cases, we are working with particulate materials, but sometimes also high aspect ratio, it means fiber-like materials are of importance. Okay, when we want to prepare magnetic derivative of originally diamagnetic materials, or we can say non-magnetic, but the physics don't like it too much, yeah, so they are magnetic materials. We can use different types of magnetic modifiers. In my lab, we mainly use magnetic iron oxides like magnetite, magnemite, or mixed iron oxides, sometimes different types of ferrites. What's important to say is that the magnetic particles are in general biocompatible, so we can also modify, let's say, microbial cells. Okay, during our work, we have developed many different types of magnetic modification of many different materials. One of the first procedure we developed is employing magnetic fluids or ferrofluids. So here you can see magnetic iron oxide nanoparticles present on the surface of sawdust left top, or right top, it's is a cells of further yeast, Glucuronidase, also magnetically modified disease nanoparticles. Another procedure, it's a mechanochemical procedure where we have used the system for modification of some special type of materials. On the top, you can see native and magnetically modified starch grains, for example. Quite often we are using microwave-assisted procedures. The first procedure we developed, direct microwave-assisted procedure, is based just on mixing of ferrous sulfate, the material to be modified, in this case, for example, sawdust, increasing the pH and putting it into microwave. And after 10 minutes, you can see we have a magnetic derivative of sawdust. It's a very nice procedure, not generally applicable for modification of sensitive biomaterials. For this case, we developed so-called indirect microwave-assisted procedure, where in the first step, we prepare magnetic iron oxide nano and microparticles just by microwave irradiation of ferrous sulfate at high pH. And the formed iron oxide particles can easily interact with different types of biomaterials. So at the bottom, you can see in the left test tube, a suspension of Baker's yeast, which is going to be mixed with iron oxide particles. And after some incubation, you will see that we have prepared magnetic derivative of yeast cells. For some really sensitive materials, we have developed so-called sub-zero temperature modification. So it's a procedure for modification, let's say, of native enzyme, immobilized enzyme, or some really sensitive biomolecules. And it's based on the standing of the modified material in the freezer, and the water is going away from the ice during the period of time. So we can prepare magnetically modified materials using different procedures. And now, what can be the applications of these materials? In fact, in our lab, we have magnetically modified many biomaterials, in addition to some inorganic materials and synthetic polymer materials. So here you can see some lists of the typical materials we used. So really some, I don't like the waste, food waste. I usually use term secondary materials. Spent grain was already mentioned in previous talk. We also work with other type of biomaterials, including chitin, chitosan, bacterial cellulose, and so on. But we can also modify microbial and microalgae cells, like Baker's yeast, fodder yeast, chlorella, and so on. We are working with microalgae, and we are also working with seagrass. What's important to mention, many of these materials can be also converted to biochars, and biochars are currently very intensively studied materials. There are more than 10,000 papers where the word biochar is just in title or in keywords or in abstract, very intensively studied materials. And one of the most important applications of the modified materials, biomaterials, is their use as biosorbents for the removal of different types of pollutants, xenobiotics. We mainly played with organic dyes because dyes are very important pollutants. Yeah, they are produced in hundreds of tons per year or over the world. The problem is that organic dyes are usually toxic. They are very difficult to biodegrade. And if we want to remove them, so the absorption process may be one of the best possibilities. And for where in large scale, the absorbent should be cost effective. So biosorbents prepared from waste materials may be one of the solutions. So we have studied many of these materials. What's important, I've mentioned that magnetic iron oxides behave like nanozyme or enzymatic material. And we have found during the last years that combination of standard adsorption and enzymatic degradation of dyes may be better procedure for more rapid and more complex degradation and removal of the dyes from the systems. In addition of organic dyes, we have played with bisphenol A, heavy metal ions removal, or radionuclides removal using different magnetically modified biosorbents. Similar materials can be also used for the separation of important biological compounds like lectins or enzymes. You can see that we work with mostly with hydrolysis. And sometimes these procedures are really very efficient. In the case of chitinase from Euphorbia, my colleague worked for some time in Catania, in Sardinia, as a department of plant biochemistry, and they wanted to isolate the chitinase from latex of some local shrubs. They spent 8 months without success. I have suggested to try magnetic chitin for the separation, and with this material they obtained, or we obtained, the pure chitinase in 2 days. Yeah, so very efficient procedure. Magnetically modified biomaterials can be used as carrier for the immobilization of enzymes or cells. So an example, for example, magnetic spent grain. There is a magnetic chitosan. We have also played with bacterial cellulose. In all cases, we could immobilize successfully these bioactive compounds or cells. And there is also other interesting application. You know that biofilm are causing problems, so for example, during filtration of large volumes of water. And in fact, biofilm is formed when concentration of signal molecules of the target bacteria exceeds some limits. So one of the possibilities how to reduce the biofilm formation is a decrease of concentration of signal molecules. So some groups are interested in degradation of these signal molecules. We are using other approach, adsorption of signal molecules on some low-cost biosorbents. And we have shown that magnetically modified spent grain could quite efficiently remove these signal molecules from Pseudomonas, and so biofilm formation was substantially decreased. Magnetically modified materials can exhibit catalytic properties. So we have played with modified Baker's yeast, which can exhibit hydrogen peroxide removal or saccharose hydrolysis. What's also interesting, during our study of biochars, we have found that also biochars exhibit peroxidase-like activity. So they behave like enzymatic material. And again, this activity can be used for some interesting applications, both native and magnetically modified biochars, of course. In fact, several times, 2 important terms were mentioned, circular economy and biorefinery. In all these cases, magnetic materials can be used efficiently. In the case of circular economy, we are trying to employ food or secondary materials from the food industry into valuable products like biosorbents. In the case of biorefinery, magnetically responsive materials like catalyst immobilized on magnetic carriers, magnetic absorbents for the separation of radioactive compounds, or magnetic solid phase extraction for the detection of appropriate compounds may be of high interest. Okay, we have prepared several types of review papers. So who would like to be more involved in this topic? Yeah, so there is some review just focused on the modification of agriculture and food waste. In another book chapter, we have summarized the information about dye removal using magnetically modified biological materials. So I hope that I will find some cooperation with you who is working with some wastes, so we can try to convert it into second generation waste with promising properties. At the end, I need to thank Tuija, the girl on the left, and all my other girls who helped me during the research during last year, and thank you very much for your attention.
Thank you for this interesting presentation. We have the floor for one question from audience. Yeah. No, in the Zoom, they will not hear you, so it's better the microphone, please.
Thank you so much. Thank you so much, Professor. Very interesting presentation. I'm working on biochar and I have a presentation in 2 presentations. I'm really wondering about modification of biochar. And since I have experience with many biochars, I would wish to ask what kind of properties of biochar are promising to make them modified, perhaps, if there is any chemical or physical property of the biochar that is produced that we can investigate, okay, this biochar will be very good in further processing.
Yeah, go to the expert in biochar. We are not experts in biochar. It's one of the interesting materials we are working, not really experts. We have tested, let's say, 12 different types of biochar produced by us, obtained from our colleagues. In all cases, we are able to find peroxidase activity in different level, yeah. And in fact, we can magnetically modify everything almost, yeah. So as to the biochar, any biochar we can magnetically modify, and using at least one of the procedures I have described, yeah, we are able to modify almost everything. So 2 weeks ago, we have modified nanodiamonds, for example. Yeah. So
may I have one question?
Yeah.
Did you check what kind of physical properties the biochar can, if you have the same feedstocks, perhaps because I'm working on pellets. So if the pellets were subjected to pyrolysis and the biochar is in a pelleted form, let's say, and you have a raw biochar, is there any difference in modification?
Basically, in the case of magnetically modified biochar, which is my main interest, we basically use powdered biochar modified with magnetic iron oxides. And okay, we have used it as an adsorbent, adsorbent for the removal of dyes in combination with the enzyme mimetic property. Yeah, so it was a great advantage that the removal of some dyes was substantially faster, combining this absorption and enzyme mimetic property. So it might be very interesting application we can discuss during the coffee break.
Sure, thank you so much.
Thank you.
Thank you again. I'm happy to invite now Professor Mehmeti that will talk about the application of insect frass for the development of sustainable agricultural production in Kosovo.
Okay, thank you very much. Almost good evening, maybe. However, I will try to be brief in my presentations. Actually, it's my pleasure here today to present an ongoing project which is related to the application of RAS in agriculture, but also a little bit of introduction as well about organic production, which was lead us to this project, then a description of project, and I hope so that in next meetings I will be able to present more results about our project. However, just to give some data about the Republic of Kosovo, which is located in the Central Balkan Peninsula with about 10,877 square kilometers. We have characterized by two main plains. In the west part, Dugajini Plain, and eastern part, the Kosovo Plain, which differs in climate as well, but also in the production systems. However, in the west, we have the climate with the Mediterranean impact, which allows us to cultivate crops a little bit earlier compared to the to the eastern part, which is the continental climate. The most spread soils are fluvial soils, and also we have in the mountain areas some other type of soils which are very suitable for cultivation of different crops. And for this, nowadays, one of the crops that are cultivating and increasing in area are medical aromatic plants. However, main crops that we produce almost are wheat, maize in combination with beans, and then potato, vineyards, which we are very well known about the production of wine as well, some vegetables. But nowadays, also it's quite important to protect forestry because almost 500,000 of hectares belongs to the forests which are very rich with the non-forest forestry products. And for this, we have about 6 million euro import of these products. Then nowadays, as I mentioned, is increasing of the production of medical aromatic plants, and all of this area is certified organic. So we have also producers that produce seedlings of these medical herbs, but also different species of medical herbs, especially oregano and chamomile. So in total, nowadays in Kosovo are about 2,000 hectares of organic products, which in share is about 0.5% of the land. But if you see the data of exports, it's quite a big amount comparing to other neighboring countries, about 6 million euro. But on the other hand, also the government is committed to support farmers, and for that, they have prepared a national organic agriculture plan, which has foreseen some activities to increase the potential of production of organic crops as well. This, for sure, somehow will secure the preserving of biodiversity, natural resources, and to reduce the negative impacts into the ecosystems. However, in 2002, this starts in early stage when was established for first time Organic Association of Kosovo. Then later in 2013, it was established Association Organica, which are mainly focused on the operators of the non-wood forestry products and medical aromatic plants. So the aim is as much as possible to increase the area with cultivation of medical and aromatic plants so to not endanger natural ecosystems. Because in our country, we have like 11 endemic species, which are very important, but on the other hand, also we have some other species which are with high value and for sure that they should be protected. So based on that, afterwards, this sector was emerging every year, and for sure that it's quite important that on 2002, a group of some professors, in this time including myself, we are pioneers of the development of organic agriculture, because somehow everything when you try to change either to farmers is quite difficult to introduce a new production. And in this time, our group starts first time somehow to introduce the first information and also advice related to the cultivation of organic crops. So these activities mainly were focused on the education, not only of farmers as well, also of the students, because we have included in our curricula also the organic agriculture as a subject, and later we start also to use to some equipments as well and to do some research related to the organic production, and afterwards we continue with the publishing, informing as well, and the marketing. However, why I present this introduction? Because our aim is somehow to do same with a new project, and this is about the production an application of insect frass for the development of sustainable agriculture in Kosovo. And for that, we somehow start to map the organic agriculture side stream in the field, and afterwards to feed insects and to produce the proteins as well, but also to produce, as we call it, frass or excrements of the insects. And for that, we start to use the frass in the chamomile production as well, and also in the oregano production. But what are the main objectives of the project? The main objectives of the project are connected to the establishing of the insect frass production in Kosovo. So it's a company that is focused on the producing of such insects, and for that, it's important, first of all, licensing. Today, it was a discussion here about the EU regulation as well. Also, this is quite important, and for the insects nowadays, because some of the insects are allowed to be licensed, but as well also to be used for food, but mainly for the feed of the animals. And then to research and set up the facility where we'll be able to rear the insects. And for sure, it's quite important to do an assessing of local farmer acceptance, because it's quite difficult to convince farmers. Let's say that now in the past many years, they are using artificial fertilizers and organic manure. But nowadays, if you recommend to them to use the frass, for sure, for them it will be something new. And even for us, and for this, we are doing some questionnaire related to the acceptance of the farmers, but also to the consumers related to the using of the mealworms, especially for the food. So another objective which I am much involved beside other member of the group is applying of insect frass in agriculture. So we want to test the mealworm frass on medical aromatic plants, as well to define a guideline for the mealworm frass. So this means that how much amount we should use per hectare to the different crops, as well to map the agriculture side streams. Because nowadays, as we have discussed today, that these losses are up to 20%, but it's quite important that to start from the bottom. So this means that maybe we should be focused to increase the awareness of farmers and consumers how to use these side streams. Otherwise, if we start from up to bottom, I think will be quite difficult, because we did the last year on pilot research about the mapping of side streams, and this year, and the results shows that this year we have less side streams of agriculture. Why? Because the prices are increased. So this means that it's quite important to do this mapping and to have data for different crops. And last but not at least, also we will do the evaluation of impact of frass on the soil nutrients, especially to micro and macro elements, which are very important for the plants. So in this project, we are involved, we as Faculty of Agriculture and University of Pristina, then University of Gizeh, and we have two companies, a company which is rearing insect and called Go Beyond, and we have a company which is producing the medical herbs and is one of the biggest company called Biosource and is located in the northwest of the Kosovo. So the latest innovation, what happens in agriculture, somehow show us that we maybe should think differently and somehow to use the concept of circular bioeconomy using insects, which will be feed in this waste material, or better to say, agriculture side streams. So this is how we use the frass. Is very fine material that you can use, and afterwards, this is a chamomile that was cultivated in these plots. But in other hand, also it's interesting that we are using for first time the frass in Kosovo in medical aromatic plants. As well, I have read many papers, but they are mainly focused in the pots and in the greenhouse experiments, but not really in open field. So that, to be honest, even for us, it's quite challenge, and we are working very hard on results and on the evaluation of the frass into the crops. So the main objective is somehow to explore feasibility of the local frass production. So to evaluate first the sources then by conversation, application, and the company is interested as well to prepare a frass to sell to the farmers. So this somehow maybe it will allow us to increase area of the production of the medical health, but in meantime also to the frass. And another group also is working related to the use of the frass and the impact in the plant growth as well, in the yield and in soil health. So we used for first time Tenebrio molitor mealworm in the chamomile and oregano, and we aim also to license Hermetia illucens, black soldier fly, which is insects mainly used as a feed for animals as well as fertilizer for the crops, but till now not as food for the humans. So the main aim will be focused on that how we to quantify the amount of the frass that should be used for the chamomile and oregano. In meantime, also we did some germination experiment with the frass as well and the compost, but the results were very negative. Not any plant was not germinate, and this was due to some fungus. But we will not stop, and we will try another alternatives and to find the reason why the seeds of oregano and chamomile in this case were not germinating. So the mapping of the amount of side streams in agricultural production is quite important, and we are focused in the main crops like pepper, watermelon, tomato, potato, apple, pear, and cucumber. And for this issue, we somehow want to see that how many waste we do have in our fields then we to use to feed this insects. However, this is what I already mentioned shortly because of the time. So we'll be focused on licensing Hermetia illucens to establish a facility to rear the Hermetia illucens, as well also to evaluate impact of the insect frass in the soil nutrients. And the conclusions, I can say that for this work, for sure, it is quite important to involve stakeholders, producers, researchers, processors, traders, and distributors, because it's as a multidisciplinary sector that is quite difficult to do alone. And this for sure will somehow encourage us to work together with different companies. However, we can say for the time being that the use of frass can be an alternative to be used in future for medical aromatic herbs, but still we need more studies because that we should understand the insect frass role as mineral fertilizer subsidies in the future. So the agriculture side streams can vary significantly depending on specific crop, depending on the agriculture activity, production systems, and it's not so easy to map them and to quantify precisely. But use of agriculture streams for sure to feed insects will contribute effectively to the circular bioeconomy. So the frass also has an impact on the above dry biomass of chamomile and oregano as well, also to the height of the plants. So last but not at least, we have in front of us two challenges. So one is that it is opportunity to use the frass as a fertilizer. Another hand is also somehow to convince people to eat the products which are produced by the mealworms, because nowadays there are many foods, especially chocolates, that they are ready to be used by the humans. And for this, we have two challenges. One is to convince farmers to use the frass as fertilizer, and on the other hand, consumers to use the chocolates or other foods which have origin from the mealworm. I know that in this late afternoon after food is not maybe so promised, but I hope that you can have chance to test such products as we did with our students and our colleagues. Thank you very much.
Thank you very much, Professor Mehmeti. Really interesting presentation and really fitting in this working group. Is there any questions from the audience? Yes.
Thank you, Arben. That was really interesting. So it's curious, but I mean, quite right, I think, that you seem to be treating frass as the primary product and the insects as like a co-product. Would that be right or not? Or at least that's the emphasis that you kind of gave.
No, we are three working groups. So one group is the company which is rearing insects, and they look for proteins as well, but also to commercialize the frass.
Yeah.
And we look only to use frass to the crops.
Yeah.
So somehow we have a divide task in the project.
Yeah. And of course, there's another co-product, which is the oil from defatting, because normally the black soldier flies are defatted before they're used for feed. So that's another product. So just one more question. You said you were looking at the you're going to be looking at the impact of the frass on soil health, but what about on the plants and possible uptake of heavy metals and things like that? Are you going to be examining that too?
Yeah, actually, when I say soil health, it is very wide, but however, our focus for sure will be to soil nutrients as well. But related to that, we'll do also some analysis of the content of the cadmium as heavy metals, also as a lead, and to see if the frass was used will be increased or not. But as micro elements in this case, not really as heavy metals.
Thank you.
Thank you. I have one short question that is related to the last question that we had. As you are choosing maps as a target for the fertilizer frass-based, are you thinking to check also the content of the bioactives of the aromatic plants that might change according to that? And also if those plants are produced for some tea consumption, the sensory analysis also of the final product would be interesting.
Yes, actually, this is a very good question. And already you are thinking next project, because first we are focused only in production, impact on the yield as well, in the height of the plants. And the next step will be related to the chemical content, because this is quite important. Otherwise, if you produce, even if you increase the production or biomass, but if there are less of active ingredients, then for sure the price will be lower. So the farmers then will not be so happy. And we are thinking that next project should be focused related to the impact of the frass as well to the chemical content of the medical herbs.
Okay, perfect. Thank you very much.
Okay, so we can pass to the next speaker is Magdalena Joka-Yildiz. Is from Białystok University of Technology, Poland. So she will present for us bio-waste-based pellets as a promising feedstock for biochar production. Floor for you.
Thank you so much for the nice introduction. Thank you for being here. It's a huge pleasure. So a part of my research that I performed with my colleagues with Edinburgh, I want to show you how the pellets work in pyrolysis, what kind of feedstock pretreatment can we make to produce biochar in the form of pellets. So I will take the opportunity to advertise my university. We are located in Eastern Poland, in the area so-called Green Lungs of Poland. As you see on the picture, the university is surrounded by forests, so that's why we have a lot of woody residues when we are interested in pellets and woody pellets and pellets from agriculture and food waste, because it's also a region that has a strong agricultural production, especially milk-based production. And I want to introduce also myself, as I am new in this course section. So I graduated my PhD in 2019 in the environmental sciences. However, I find myself as a mechanical engineer because of my background. I'm interested in waste to biochar processing, renewable solid fuels. I was working on pellets to combustion. Now I shifted myself in pellets to biochar because they didn't work in combustion so good. Thermochemical processes and solid pretreatment technologies. So pellets, as we think about pellets, most of us were working with pellets or used pellets or saw somewhere. And actually, we call pellets most of granules that we need. Actually, pellets are very well described by some norms, and they are granules of a diameter from 6 to 8 millimeter and a length from 3.15 until 40 millimeters. And pellets are produced in the so-called pelletization process, which is a pressure agglomeration of previously committed materials. And this process can be run in three main different ways. So as you see on your left-hand side, we have two examples of industrially continuously produced pellets in a flat matrix and in a ring matrix. And in the picture C, we have a single pellet press. So it's very commonly used for scientific reports because it gives a huge control of the process conditions. So we can measure the pressures, we can measure the temperature, we can set the conditions as we wish to, and it's a good device to perhaps make some preliminary test of the compaction suitability of a certain feedstock. So what is biochar? Biochar was mentioned today already, which I'm really happy from. Biochar is a porous material. It's a carbon-rich material which is produced in the so-called pyrolysis process. The pyrolysis process is a thermochemical process in the absence of air or any other oxidation agents. So we are just heating up the feedstock and letting the volatiles and moisture to be released. And as a result, we get a high carbon concentration. And by biochar, we also obtain a material that can store carbon efficiently and stably. Biochar, according to the European Biochar Certificate, should not be used for combustion. So the question might be now, then for what? Actually, biochar can be a filter, can be further processed to obtain activated carbons, can be used as a soil amendment, and also some reports declare that it's triggering fungi growth. I'm not an expert on this, but I saw many posters about fungi, so I think you are interested in this topic a lot. So by subjecting biochar to the feed of this, their growth might be improved. So what are the advantages of using pellets? Pellets are having a uniform constant form. They are faster in heating rates and longer residence time in the reactors. They have a high energy density. They're easy to handle and transport. They have some environmental benefits that we can perhaps reuse the waste materials, also combine some waste materials. They are stable and suitable for large-scale applications. And what is important with pellets, we can, as mentioned before, we can mix a couple of feedstocks and then we can engineer the biochar properties. So perhaps we have a certain feedstock that has rich in some contaminants and another is not, but is suitable for compaction it gives a porous structure more efficiently than the previous one. So we can combine that, optimize the process and then process them into biochar. So the most commonly reported feedstock for pyrolysis to produce biochar is wood. 31% of papers published about slow pyrolysis to obtain biochar contain a feedstock as wood. Actually, the feedstock as wood pellets is not that suitable for producing biochar, in my opinion, because first of all, it gives a very low yield compared to agriculture and food waste. And secondly, why should we transfer woody pellets to biochar if they can be used for combustion and they're suitable for combustion? Anyhow, this is the majority. However, people that are writing reports about biochar and describing pellets, they are missing some of important factors, for example, for me as I'm working on pellets. And in order to understand what kind of impact the pellet properties have on the biochar, we need to have a full data set. However, as I marked by the gray points, some of this are missing. So what was possible to do, what it was possible to compare how the temperature of pyrolysis is affecting the biochar yield. And as you see, the correlation did not fit exactly very well. Perhaps it would be better if we could divide this feedstocks into a couple of groups, and then we could understand the mechanisms. So I addressed a couple of questions about biochar and pellets based on the research scan. Detailing the bulk density and particle density of pellets, are there any correlations between them, and what kind of mechanical properties do pellets have after pyrolysis? Because they have some. I designed 10 different types of pellets based on agriculture and food waste using two binders, potato pulp and coffee grounds. The potato pulp is a waste with a high moisture content obtained during starch production. And we have a big deal with this in the region of Białystok. I used buckwheat husk as a main component, hemp hull, giant miscanthus stalks, and hazelnut shells. So 4 different main components, 4 different biomass types. The pelletization took place in a continuous pellet mill with a flat die rotating roll system, and pyrolysis we run in the UK Biochar Research Center in Edinburgh, 2 different temperature scenarios of 550 and 700 degrees for comparisons. We measured the physical properties very easily by using a balance and a caliper. So there was the bulk density and particle density, and also measured the mechanical properties as the stress at break by compression. First of all, I would wish to show you how the pelletization process, pelletization of the feedstock itself, affects the bulk density of biochar. So perhaps if we subject pellets, we have a 4 times higher bulk density of the biochar we receive. What does it mean? It means that it's easier to transport, easier to store, cheaper also to transport and store, and more feasible to use further. We made a correlation between the bulk density of pellets and the bulk density of biochar, and turns out if the denser pellets are, the more dense biochar also we get. So as you see, this fit goes for 10 different biomass types, 10 different pellet types with different binders, and we can state by measuring so many samples that this can be true for most of feedstocks. Also, particle density have been measured, so before and after pyrolysis we have a correlation. And what is the most important thing for those who are interested in biochar production but perhaps don't have facilities that allow to run pyrolysis at a big scale, we correlated the difference in particle density before and after pyrolysis of pellets to estimate the bulk density of biochar. So therefore, you can have a couple of pellets, make pyrolysis in a small reactor, and by measuring with very simple devices the particle density of them, that it can be estimated what kind of bulk density of biochar we can get, which is important for further calculations, perhaps some cost calculations and so on. I have 1 minute, so I will go fastly through the mechanical properties. What I want to say here actually is that depending on the feedstock, the mechanical properties were different. For some feedstocks, for some pellets, the biochar mechanical properties were better, or let's say the pellets were stronger when pyrolysis was performed at higher temperatures of 700 degrees, but it does not fit for all feedstocks. So perhaps for hazelnut shells and buckwheat husks, indeed, but if it goes about hemp hull or giant miscanthus stalks, which are having a lot of fibers, this mechanical properties were same for 2 temperatures test. So to sum up, I'd want to say that Pellets are a valuable feedstock for pyrolysis, and they allow us to improve the bulk density of biochar. And pelletization as a pretreatment step in the production of biochar can improve co-pyrolysis of various biomass types, which is very important. And I want to highlight one more time that pelletization might be a way to obtain engineered biochar. And as mentioned before, we saw a strong fit between the feedstock and the product that has been obtained. Thank you so much.
Thank you, Magdalena, for also respecting the time and for the interesting presentation. We have floor for 2 questions.
Thanks for nice talk. How is the stability of your biochar pellets in water? Could they be used, let's say, for filtration processes? Will they be stable after prolonged contact with water?
I didn't test it, I need to say. I was more focused on the mechanical properties. However, it depends on the biochar. As I tested for other things where water was involved into biochar, so some are more hydrophobic. So I believe that they can be stable until a certain point in water, but I would need to test it, and I can get back to you after performing the preliminary test.
Thanks, we will discuss during the break. Thanks.
Thank you.
Thank you. Other question? So we can pass to the next speaker.
Thank you.
Okay, so I'm inviting Professor Drougakos from the National and Kapodistrian University of Athens in Greece. His presentation will be about natural products in the fight against aging and age-related disease. Please.
Okay, thank you. I would like, of course, to thank Gianfranco and for the opportunity to present some of our data that we think that fit nicely to the purposes of this working group 4 towards the valorization of waste and cyclical economy. Now, just a brief, I am a molecular cellular biologist by training, so this is what we actually are doing in the lab. So we are a group who are focusing on age-related diseases, and we are applying system biology approaches to study the proteome stability in different stages of life, as I will show in the next slides. We try to understand the functional communication of proteome stability with mitochondria, and at the systemic level, how these networks collapse during aging or age-related diseases. Now, we have many different models that we are using for our studies, including normal and cancer human cells, and of course, in vivo models like Drosophila flies and transgenic mice. And we have also a strong translational axis because we are participating in different clinical trials with oncologists, cardiologists, etc. But also we're working very much with natural products, small molecules, and I will explain the purpose of these investigations. So this is just a brief introduction on the genetic information flow. So it all starts with the nucleic acid, then you have the proteome, and there is all this proteostasis network, as we call it, which keeps the proteome functioning. Actually, there's a collection of protein machines and three-dimensional structures. So, of course, you need lots of energy. This is done in the mitochondria, as you know, and we're also working with mitochondria, metabolic pathways, etc. And, of course, you need sensors. Why you need sensors? Because all of us were constantly exposed to different types of stress, and the sensors are transcriptional factors which actually what they do is that they sense the stress, and then they response by changing the transcriptome and the proteome itself. So this is actually what's going on in life. So following the initial period of 9 months, which actually now biological system is the period of construction, there is the period of sexual maturation, which ends up to the period of maximum fitness. This is what's happening in nature, and then it comes this period, the period of aging. Aging is highly unnatural. There is no aging in nature. Aging occurs only in humans and animals in captivity. And the problem with this period of life is that because you have an accumulation of damage in all biomolecules, then we all become very prone to different diseases. And this is what we call the age-related diseases. And you see here some data regarding the epidemiology of some typical age-related diseases. It's also called the non-communicable diseases. So you see that including cardiovascular diseases, cancer, and of course, dementia, all of these diseases appear late in life. And the most emphatic paradigm is dementia, where the risk actually occurs after 75. Now, something which is also clear during aging is that all the pathways and networks which keep our biomolecules in function where we are young, including proteome stability, mitochondrial function, etc., they become destabilized. So, and then it comes the question whether we can do something about that, meaning whether we can decelerate the process. But then again, you have to understand the basics. You have to have a good model so that to start the process, to study the process, and then ask whether this is feasible. So you see here, I don't have time to enter into details. This system biology approach that we apply, we're using, I said, different models. In this case, we are modeling the loss of proteasome activity. Proteasome is a machinery which degrades malfunctional unfolded proteins. During aging, it becomes dysfunctional. So we have been modeling that process in human cells as well as in mice and flies. And just to make a very long story short, we have also found that if you mildly activate proteasome early in life, this has prolongevity effects and also increases health span. Health span is by definition the time of life which is free of disease. And other findings which also relate to this type of approaches is that if you, for instance, mildly overexpress the stress sensors, then you become protected against different diseases like Parkinson's, Alzheimer's, including also cases where you apply therapeutic drugs like proteasome inhibitors, for instance, in multiple myeloma in cancer, where you get some severe cardiopathies. So just to mention that our studies in Alzheimer's and Parkinson's disease were done in humanized flies, as we call them. And these are flies which carry human genes in order to phenocopy the human disease. Okay, and this is where it comes to become relevant to this course-action, and I think also this work in group 4. So, and then, of course, as I said, the question is, can we decelerate the clock? Now, in animal models, and of course in cells, we can apply genetics, transgenics, knock-in and knock-out genes. We can also apply caloric restriction, but this is not, none of these approaches can be done in humans. So we are left only with diet. And diet is very critical because apart from energy, it also provides the organism with many different small molecules, which are, some of them are very beneficial. So we're applying a different approach. So normally we are trying to develop drugs when the disease is there. And this is a bit problematic because diseases, as said, are happening in an aged organism. So we're trying to isolate natural compounds, small molecules, etc., which can be used both for prevention in terms of health span increase, but also for therapeutic purposes when and where this is possible. And we think that natural products, extracts or pure compounds, are a very promising reservoir for these investigations. So I've been very active in this aspect. We have been participating in many different projects. Just to mention, we're not doing that blindly because we have the support of all our basic research. So we know exactly what we're going to modulate. And this is the idea behind all this type of investigation. Just to present, this is a very recent EU project that we are being concluded In this case, we are screening natural products. In my lab, we screen more than 6,000 natural products because we have a very high throughput platform, as I will show in the next slide. So in this case, we are screening, extracts from organisms from the mesophotic zone of the ocean, and this is the pipeline. So you had the collection, of course, extraction, chemistry, and chemical profiling, and then it's us. So in this case, just to mention that this is a process which is full of disappointments, but at the end of the day, you came up, if you know what you are looking for, with success stories. So the idea is to add claim in something in order to increase its value. Claim means that this small molecule natural product can be from a waste, whatever, may have a application in pharmaceuticals, cosmeceuticals, or nutraceuticals. And this is the platform. So we have been developing through the years all these in vitro cell-based, actually, but also in vitro, and also in vivo models where we can model aging and many different age-related diseases. And this is quite high throughput when it has to do with the human cells. So we start with in vitro approaches, then whatever passed the screening goes to cell models, depending on whether we screen for phenotypic targets or phenotypic screening or target-based screening, and then whatever passes the criteria and the threshold, then it moves on to in vivo models, starting from Drosophila, of course. So this is a paradigm of different EU projects who have been involved, including the project with samples from the mesophotic zone of the oceans. Microsmetics was referring to small molecules from microorganisms, and of course, aromatic plants and plants from the Mediterranean diet. This is a success story just to show the pipeline of our work. So it all started with an extract from a mollusk, Hexaples trunculus. So we found this small molecule, 6 from indirubin, and it was quite promising. So at some point we tried and managed the chemists to synthesize the molecule. And by crystallography, we found that a specific inhibitor of kinase, GSK3, this was done in cells. Then we move on to in vivo models like Drosophila, and we found that this 6-bio inhibits GSK3 and also PDPK1. And by doing so, it activates an RF2, it's a stress sensor, and this increases longevity at the in vivo models, including also a delay in the so-called age-related phenotypes like locomotor activity, metabolic malfunction, etc. And these are the papers just to show that this 6-bio small molecule was also very effective in suppressing cellular senescence. So I would like to conclude because it's getting late, and just to thanking people in the lab in this and many other projects that they didn't have time to present, our collaborators in Greece and abroad, our funders, and all of you for your attention. Thank you.
Thank you, Professor, for the nice presentation. Are there any questions? Yeah.
So you really make evident that this natural product are, let's say, promoting healthy lifespan. So if we, in terms of food, I think the ultra-processed food would be the nightmare for the natural product. So if in practical point of view, if you want to give a practical recommendation, so which food can be classified or considered as safe in terms of anti-aging, let's say based on, for example, the NOVA classification of processed food?
Okay, thank you for the question. All foods are safe as far as they are safe, right? So if they pass the regulations, they are safe. Of course, there are studies, for instance, providing data that the Mediterranean food is particularly healthy and has been correlated with reduced risk for cardiovascular diseases, likely health span increase, etc. What we're trying to do is that, we're trying to we're trying to exploit the chemodiversity of the biosphere, right? So it doesn't matter whether it is an aromatic plant, which you collect from the mountains, or something which is post, preparation of a diet, of a food. All these chemistries may have value, right? So because now in Greece, we're also in the mood of developing the so-called cyclic economy, we are very much willing to increase the income also of the agricultural producers. So including aromatic plants, using the waste for, say, producing or screening for something which may be interesting for the economy, etc. So, yeah, we all know what is healthy for eating. Anti-aging is another story. It's very, it's a combination of many different things. Yeah. Thank you.
Any other question?
Thank you. So we can pass to the next speaker is Semanur Yildiz from Sustainable Food Processing Laboratory, Sakarya University, Turkey. We'll speak about optimization of ultrasound-assisted extraction of cold-pressed pistachio meal protein. Semanur, the floor is for you.
Thank you very much for this nice introduction. My name is Semanur Yildiz. First of all, I would like to take a second here to thank to all organization team. It has been a very informative day for all of us, and I feel lucky to have all these presentations which are focusing on sustainable development goals and food loss and waste management strategies. And European Green Deal policies, etc., because they are all, I hope my presentation will contribute to all of those presentations. So my presentation is the optimization of ultrasound-assisted extraction of cold-pressed pistachio meal proteins. I'm working as an assistant professor at Sakarya University. So my expertise is mainly based on non-thermal food processing technologies. Especially recently, we are more focusing on valorization of food waste and byproducts to produce value-added food ingredients with enhanced functional characteristics. So today I will be talking about pistachio meal proteins. So the motivation of this study is based on the environmental impacts of food sector, which are mentioned previously very well. So including the livestock production, there is a need to decrease the environmental impact. And also, as mentioned in the morning session, the world population is expected to reach approximately 10 billion by the year of 2050. So there is an increasing demand to meet nutritional requirements for this increasing population. And there is also changes in the dietary preferences towards vegan, vegetarian diets, or people who prefers not to consume meat or dairy proteins, And another need is to improve the functional properties of the proteins. So these are all the reasons that makes us to focus on alternative protein sources. These are alternative to meat and dairy protein sources, and also alternative processing technologies to develop more sustainable food production strategies. So as we're mentioning alternative protein sources, we can talk about insects, micro and macroalgae, leaves, cereals, oilseeds, legumes, and vegetables. And today I will be talking about the food waste and byproducts, which can be used to meet the nutritional needs, and it will contribute to the efficient use of resources, and we can develop new food ingredients out of them, and it will contribute to the circular economy and sustainability strategies. So residues from oil industry, we are focusing on oil seeds and nuts, which can be used to produce oil by cold pressing. And the resulting residue is called as the biomass or meal, contains a significant amount of proteins, so which can be actually used for the utilization of as an animal feed source. Or another option is to valorize them in the concept of circular economy and use them as a for human nutrition. And the resulting oil will go to the cosmetics, food, or energy applications. So the biomass itself is a good source for proteins. So pistachio is one of the famous nuts in Turkey. Actually, it's the #1 producer is Iran, followed by USA, and then Turkey. The protein content of the pistachio is around 20%. When we produce protein concentrates or isolates, it's very important for us to have an initial amount of proteins at a significant level, because otherwise it may cause low extraction yields. So when we are talking about pistachio meal, then it means that the fat was removed from the pistachio, so the proportion of proteins will be higher in the pistachio meal. As you can see, it will be a rise to 23%, and which makes it a good potential for protein source. So here I would like to summarize how we produce plant protein concentrates or isolates. First, once we obtain the plant-based waste or byproduct, we have the size reduction section, and then we need to remove the fat, because otherwise it will mask the protein extraction yield. So we have to remove the fat. And then therefore, actually, the cold pressed oil meals is a good candidate for this purpose, because it's already the fat is already removed, but still we have the residual fat, so we need to check again for fat removal. And then we prepare the protein dispersion, and the alkaline extraction will start by adjusting the pH to alkaline condition. And then it follows with the centrifugation and collection of supernatant after centrifugation. At that point, I call it as protein extract. Okay, it's in the liquid form. Then we need to adjust the pH to isoelectric point for isoelectric precipitation, and then we will centrifuge and collect the pellets. This time, the solid part, and do the neutralization and drying. And at this moment, we are calling the final product as protein concentrate or isolate, depending on its protein content. And ultrasound process as an environmental friendly technology comes to the picture at this point, where we apply alkaline extraction. So we can assist alkaline extraction with ultrasound to decrease the extraction time and to increase the extraction yield. So here I would like to introduce a little bit the ultrasound process. It's the sound waves that's beyond the human hearing. So usually the food application uses frequencies between 20 to 100 kilohertz. So there are different applications of ultrasound, as you can see from the slide, from emulsification, homogenization, encapsulation, and so on. But the main focus is the extraction for this work. And the main mechanism of ultrasound is actually based on the cavitation phenomena. So when we apply ultrasound, there is going to be bubble formation. So there are different pressure levels and temperature levels, which are changing locally, you know. So it helps the bubbles, the ultrasound bubbles, to grow. And once the bubbles reach to a critical size, they will be collapsing. So at this point, there will be locally produced heat and, you know, shock waves, which cause the extraction process easier. So the research question for this work is that can ultrasound-assisted alkaline extraction improve the protein extraction yield from cold-pressed pistachio meal? So we are trying to optimize ultrasound-assisted extraction, ultrasound-assisted protein extraction for maximum protein yield. Since the shock waves and ultrasound waves will affect the tissue, it will be easier for us to extract the protein from plant matrix. So here you can see the materials and methods. We have grounding first, once we receive the meal, and then we are removing the fat. Once we have the defetted powder, there's a rehydration stage, and then the alkaline extraction or ultrasound-assisted alkaline extraction starts. So this is a drawing for the ultrasound extraction. We have a probe system here. It goes inside the treatment medium, and this is a double wall sample container, which allows us to control the temperature because we are circulating, you know, cold water around these double walls, because we do not want the temperature to increase too much. That will cause protein denaturation. And then the following stage is the centrifugation and isoelectric precipitation. And finally, we have the freeze drying to obtain the final protein powder. So now I would like to share our results for the optimization of extraction conditions. We use Bugs-Bunkin experimental design based on response surface methodology. So we have power, time, and pH, extraction pH, as the independent variables. And the response variable will be the protein yield. So we are trying to see if we can improve the protein extraction yield. And the solid-liquid ratio is constant for both alkaline and ultrasound-assisted extraction. So we use alkaline extraction only at the optimal conditions to compare with the ultrasound-assisted extraction if ultrasound-assisted extraction had higher protein extraction yield. So you can see the variance analysis results. The main effects, power, time, and pH, and also the interaction between power and pH were found significant. So we can estimate the protein content by this equation. And you can see how protein content in the protein extract is increasing depending on pH. Let's see, here we have different pH values. So as the pH goes to alkaline levels, the protein extraction yield is increasing. Here you can see the red color. And also you can see, you know, the effect of time and also power. So as the time increases, there's more release of protein to the extraction medium. And when we compare the power, as we go to the higher power levels, the protein extraction yields increases. So finally, we are reaching the optimal extraction conditions in terms of power, time, and pH, as you can see, with a higher desirability. And we validated these results with repeating the experiments at the optimum conditions. So we can say that there is a 45% increase when we apply ultrasound-assisted alkaline extraction compared to conventional alkaline extraction. So this study contributes to circular economy by valorizing the byproducts from food industry, and it contributes to development of environmentally friendly and sustainable production strategies. So the next stage actually would be investigation of functional and structural properties of proteins, because once we produce the protein concentrate or protein isolate, it's important to identify their techno-functional properties in terms of like solubility, emulsion forming properties, or forming capacity, in order to identify which type of foods could be a suitable candidate to use that protein as an ingredient, in that food formulation. So we are working with food waste and byproducts to convert them into valuable value-added food ingredients, and we are open to collaboration. So I would like to take this opportunity to say that we are open to collaboration for, let's say, Primacol, which is focusing on the Mediterranean area, and also European Union calls. And I would like to thank to my colleagues from Sakarya University if they are watching this presentation right now. And thank you very much. I hope I made it on time, and everyone, I think, is looking for the dinner. Thank you.
Thank you, Simanor. So we have the floor for one question. Okay. So otherwise, I guess that it is all for today. Just to note that for the participant that will be not able to come tomorrow, they can collect a bottle of wine offered by the company Moncaro that support us in our meeting in the desk of the registration. For the other participants that will be tomorrow, they can collect tomorrow. See you in the Giardino restaurant.